Good morning. Friends, welcome back to Science at the Shine Dome 2024. Thank you for being with us today. Over the course of today, we will hear some additional presentations from our fellows elected in 2023. And this afternoon, we will be admitting to the Academy our 2024 fellows in what will be another lovely and historic ceremony. May I begin, as I did yesterday, by acknowledging the traditional owners of the land on which we meet, the Ngunnawal people, and pay my respects to their elders past, present and emerging, and to all traditional owners of the lands where the Academy and its fellows operate, live and work. Our sessions this morning will be facilitated by two of our Executive Committee members. Firstly, by Professor Lynn Beasley, our Secretary for Education, and Professor Jim Williams, our spokesperson for Integrity. I introduced Lynn Beasley to you yesterday as the person holding the Academy's most contagious smile. It's still there. And as a wonderful, wonderful scientist with expertise in neuroscience and an enormous advocate for young researchers across Australia and across the world. You may not have met Professor Jim Williams, so many of you have. Jim Williams' research has been in diverse areas, in materials science, in nanotechnology, ion-solid interaction, semiconductor processing, and in high-pressure research for some 50 years. He's pioneered novel methods and has made discoveries that have helped shape the semiconductor and quantum research of our nation. And he has founded two spin-off companies. Jim's our spokesperson for Integrity, a recently created portfolio that is absolutely required to ensure the highest standards of excellence in the Academy and to bring a neutral and methodological approach to misconduct examination. As you can see, we give Jim all of the easy jobs. So to get going today, I would like to welcome to the stage Professor Lynn Beasley. Thank you.
Good morning, everyone, and let me add my welcome to this second exciting day. And thank you, Anna Maria. It is a real privilege for me and a delight to introduce Professor Linfa Wang, who became a corresponding member in 2023 and has come to join us from Singapore, but I know Australia is your second home. Professor Wang is globally acknowledged for his contribution to bat viruses and bat biology and the successful translation of his discoveries into biomedical innovations that have helped combat viral outbreaks from Hendra virus in Australia to COVID-19 pandemic. Please join me in welcoming Professor Wang.
Thank you, Lynn. First of all, really thank the Academy for this prestigious recognition. And I'm also extremely lucky and fortunate to have Peter Doughty as the nominator. To have a Nobel laureate believe in your science and support your science for the last 2 decades, it's really a privilege. So I start my career studying backbone virus September 94, exactly this day, 30 years ago. And so for you guys, it's clear that, bats in the media is all associated with the virus they carry. And some people even blame it's my fault. Because I was the only one in the world now, really studied all 6 major backbone zoonotic viruses from Hendra 94, as I say, in Australia, all the way to COVID-19 in Singapore. And I'd like to thank, in addition to Peter, there are many mentors, and I'd like to mention 2 people. Professor John McKenzie, who gave me the chance in 2003 to join the WHO mission and led an international team of Australian scientists, American scientists, and Chinese scientists. We discovered that bats are the reservoir of SARS-1 virus. Fast forward to 2019, during COVID-19, I worked very closely with Professor Eddie Holmes, who was the one upload the genome sequence to the data bank. That is just very, very important for us. So, and then Eddie was the supporter for this nomination. But today, I'm not going to talk on the virus they carry or the pandemics. Instead, I think I want to really give the impression is bats are good for us. So I want to really show that we can learn from bats. And that was made possible by a CSIRO OCE Science Leader Award in 2007. And I thank Jim Peacock, the visionary scientist, who was the ex-president of this academy. He established this OCE Science Leader program within CSIRO. Basically, he trusts the scientists, gave you $2.5 million, do what he says, whatever you like, for 5 years. And every year is a half-page report. So with that, I was able to transform and evolve. Now I have the nickname of Batman. I used to say I was only a bat virus man for the first 15 years because I was focused on bat viruses. Jim allows me to go into bat genome, bat biology, bat immunology, and bat everything. So I just want to show you these slides, I don't have the time to go through. So bats are fantastic, very, very biologically powerful mammals. They represent 20%, so every one of the 5 mammals by species is a bat, and they are absolutely the most abundant. And the last 3 is the most important. They have a long lifespan and a long health span. And they don't suffer cancer, and they carry all the viruses. They get an infection without disease. Now, we human beings can only dream that about these kind of biology features. But even more fascinating is during flights. Their metabolic rate can go 30-fold of their base rate. Their heartbeat goes over 1,000 beats per minute. We just finished the Olympics. I said, any athletes, if you are like bats, you can win gold medal with no trouble. And their body temperature is 38 to 42 degrees. These are normal body temperature. We all had a fever. If we're 40 and above, you will be in ICU. You will not survive a day. But bats do this every day and a few hours. So bats diverged from us 65 million years ago, and they have to be able to adapt to that. Okay, so why bats are such a good reservoir of virus and also such a healthy mammal? I can spend hours on this, but I don't have the time, so I just want to summarize. 30 years later, we have this working hypothesis. 65 million years ago, for whatever reason, one group of mammal acquired the flight capability. And they face a lot of stress, high metabolism, high oxidative stress, and that leads to DNA damage and all the other things. So they have evolved a much more efficient DNA damage response and also more robust, the innate defense and the tolerance. That's the key. I will come back later. Because these features now, the 3 features I said unique to bats, they live long without suffering cancer, and they harbor virus without any clinical symptoms. So the ability of bats harbor viruses is really an evolutionary byproduct. They don't evolve to harbor virus, they evolve to handle the stress that as a result of flight. Okay, so, there are many, many groups, and our group's the leader in these studies. So after 20 years, we had this review in Nature, but I just want to summarize that. The lessons from bats is clear. They can do the defense and the tolerance better than any other mammal, including human. So on one hand, on the left hand, they have elevated defense genes. We call them battle ready. They can fight all stresses, including viruses. But equally important, on the right arm, they have many mechanisms to dampen the overreaction or the over-immune response. From yesterday's talk, we already learned, we humans suffer a lot of diseases, especially infections. It's not due to the pathogen, it's due to our immune response, over-response. So we have a cliche in our field, say, very virus kills us, we kill ourselves. And bats don't do that. So now, I started my career in Australia and by discovering hantavirus, and so we use bats as a model to really study the virus infection and the pandemics. But my group in Singapore now is really 80% on non-infectious diseases. So we go all the different disciplines of human health, basically. We do metabolism, DNA damage, senescence, inflammation, cardiology, and heat shock stress is another really interesting area, and we're still trying to figure out how we can translate into human health. But today, it's not a science talk, but I just want to give you one example of our latest discovery. As I said, inflammation is very important. Scott O'Neill is the pioneer of the field, and he claimed in one of the meetings in Sydney to say, the roots of all human diseases is inflammation or over-inflammation, especially if you're 50 years or above. Inflammation is absolutely required. When it's balanced, it's protective. When you have too much, it's pathogenic, okay? So bats have evolved in many, many different mechanisms. So this is an inflammasome activation. So inflammasome is a very important arm of inflammation control. So as you can see, you have many different sensors, and then go down to this S as a transducer, and then the effector molecules. We discovered that bats has a new mechanism to dampen the inflammation at the very important, it's like a funnel, right? You have different sensors all through S and then go to caspase and R1 beta downstreams. So this X2 molecule is really interesting. I was just talking to Lin, It's only present in primates and bats, and absent in other mammals like the rodents, pigs, cows. And in humans, it's like an appendix. It's a leftover. It's no longer functional. It does not express. Even if it's expressed, it's not very active. But in bats, it's ubiquitously expressed and very active to control the over-inflammation. So that's a very important discovery. And now from there, we have a translational aspect. We have several patents on this. The question is, can you really learn from bats to translate it into human medicine? And I was questioned on this, by a Nobel laureate 10 years ago. And I said, we should be able to if you gave me 10, 20 years. So now 7 years later, we made it. So there's a new company called Parachute Sciences. It was founded, by VC investment in 2022. During pandemic, it's very hard to raise funds for new biotech company. So this company's headquartered in New York and has a branch in Singapore, raised $100 million doing nothing but bat immunology. I was inspired by my research, and the company's first 2 license, is from my patent. So, and the Nature Biotech also believes we can do that. So this feature came in July 2023, and the title is interesting, Bat Biotech Takes Flight, right? So, and it's a dream, 100 years dream, So the Nobel laureate immunologist 100 years ago tried to use the bat microbe to increase human health. Of course, the technology was not there, and said we had to wait for another 100 years for myself and Emma Tilling to really start this field. So Emma Tilling is an evolution biologist from Dublin. So not only the private investors, but also the public funders, NIH, 2021 had a grant call on bat immunology. In the history of NIH, this is the first species-specific grant call. And ERC, the European Research Council, followed up. They awarded us, Emma Tilling is a leading PI because it's an EU grant, only allowed 1 non-EU member. And they awarded us around 12 million euro to do a project learning from bats and to extend lifespan and improve health. So another indication is the medical doctors. I have 7 doctors doing PhD with me. These are the doctors believing in bats, okay? And consider I'm not a doctor, so that's, but even movie makers, the Hollywood made a movie on contagion, and there was just like COVID-19, this is a movie, bat transmit to pig and killing 8 million people, and the people were raising to raise a vaccine. That's before the mRNA days. You have to grow the virus and make a vaccine. Nobody can do it until they use a bat fetal cells from a lab in Geelong, Australia. That's my lab. So I always say, joking about, I have saved the world in Hollywood movie, and now I try to do it in real world. I'm really lucky now. I have a brand new sort of tailor-made facility for our bats. These are the guys, they are not only cute, really powerful. We have a lot to learn. And then to mark the opening, we have a big celebration, and of course with a bat cake, So to conclude, I'm really excited. I'm really lucky. I think as a scientist, we all dream about doing many things. We want to do good science, publish high impact papers. We want to train the next generation of scientists. And if we're lucky, we can influence policymakers so that, have science-based policies during COVID. That's very important. And you want to go further, have product and commercialization. And I have done all of this, and on top of that, I have a Hollywood movie. And for that, I really thank, Australia to adopt me, and so I was able to pioneer this work 30 years ago, and I thank you for that. Thank you.
Oh gosh, we're all taking flight in the battle, sorry about that, against human disease. I'm just opening it for questions. I can see hands are up straight away, please. Right down the front here, and then a second question up there towards the back. Here we go.
Thanks so much for such an engaging talk. I was wondering, are there any fundamental differences between fruit bats and microbats in these immunological traits?
Yeah, so that's interesting. There are obviously over 1,000 bat species, so you do have a species-specific, traits. But what we're interested in is the immunological traits that is adaptation to flight. So that across different suborders. So the fruit bats and the microbats, the insectivorous bats, are identical. For example, the inflammation dampening pathways is all same.
Wow.
Yeah. But in terms of diabetes, for example, the sugar control, then the fruit bats has certain genes that the microbats does not need. Yeah.
Do the microbats have torpor the way the microbats do?
The bats, the torpor and the hibernation is not to do with their food intake. It's more to do with geographic location. So if they're in the northern hemisphere, then they go to hibernation. Whereas in like Singapore tropic, we have both fruit bats and microbats. They don't go through this process. So it's a geographical location. It's not diet specific.
Thank you.
Oliver.
Delightful talk, Linfa.
Thank you.
Thank you. But my question, you're an Australian. Have you looked at the Australian non-terrestrial mammals? They don't fly, but they glide. And I just wonder if they're intermediate in some way.
Yeah, so that's an interesting question. By the way, Oliver was my boss in CSR, so I thank CSR for that, So no, I haven't, because gliding is not considered as a powered flight. So we would suspect that they will have a very different mechanism, but no, I don't have the bandwidth to do that here.
Time for one more question. Francis. And I know lots of people will want to talk to you in the breaks.
Thank you. Yeah, thank you.
Thank you. That was wonderful. I've become a bat fan.
Thank you.
But what I wanted to ask was about bacteria. How do they react to bacterial infections?
Yeah, so inflammation obviously controls more than just viral infections, so that, bacterial infections are not as well studied as virus, because the virus, due to the pandemic and the funding, but bacteria, even parasites, there is a group in Germany discovered that bats carries more malaria parasites than the whole mammal kingdom. So they do live with pathogens and coexist. So, but again, the bacteria side is not as well characterized, but I assume that they can handle that, yeah. Thank you.
That's wonderful. Thank you so much. And as you know, to become a corresponding member is a special honor from the Academy, and one can just see how appropriate this award is. Thank you so much. It's now my pleasure and honor to introduce Professor Tim Brodribb. Tim is an evolutionary biologist. His work explains why leaves die during drought, why some plant species are more vulnerable to water stress than others, and how flowering plants evolved the capacity for rapid photosynthesis and growth. Please welcome Professor Brodribb.
Thank you, Lynn. I'd like to start by saying what a great honour it is to be here and acknowledge the nomination by Christine Beveridge, who is very inspirational character who's the head of our Centre of Excellence and a laureate fellow, so great inspiration. I want to talk today about some interesting discoveries that I've made during my career. I've just spent the last month writing grants, so I have very finely tuned sense of self-promotion at the moment, so I'll probably need to be dragged off the stage after 10 minutes. I'm going to talk about plants under pressure now. Next is that way, yes. My career has been sort of built around this concept of plant hydraulics. I'm a plant hydraulics person, and I really need to explain that because it's got nothing to do with being a diesel mechanic. It's more about being a sort of plumber and looking at trying to understand the way that water moves in plants. It's a pretty simple sort of biophysical question. Plant pulls water up out of the soil using pressure to pull water through the system of pipes that conducts water through the plant in a very elegant vascular system. And I guess my interest in this was really peaked. I sort of started working on stomata and photosynthesis, but stomata are these little valves that sit on the surface of the leaves. They regulate how gas exchanges between the atmosphere and the internal parts of the leaf. And these fantastic little valves, the interesting thing about them, and the thing about photosynthesis itself, is that it's a very simple question that plants have sort of evolved this system to facilitate carbon dioxide going into the leaf so they can photosynthesize. But it develops this counterflow of water, and the counterflow of water is extremely rapid and can be 100 times faster than the flow of carbon dioxide. So vascular plants have developed this system of plumbing to draw water out of the soil and to feed these stomata and ensure that they stay open, because the moment they dehydrate, they close. And I guess my interest in this was really a pretty simple question, which was that if carbon dioxide and water sort of were linked in this way, was there a chance that you could sort of explain the behaviour of the photosynthesis system by understanding the vascular system? And it was a very simple sort of concept, was that if a plant could build better pipes, could it do more photosynthesis? A pretty simple kind of hypothesis. And so that was kind of my inspiration for my first major experiments after my PhD. And the idea was that more pipes, plant could make more sugar, it could grow faster, and this is what a great desire for how we want plants to behave. And so I went out with a colleague into Taylor Fields in beautiful island of New Caledonia, and we pushed water through sticks basically, and we measured photosynthesis, and we found this really nice evidence to suggest that the better the vascular system in the plant was, the more photosynthesis that the downstream leaves could do. And this was kind of the first work that was really suggesting this relationship that the anatomy of the wood, if you had bigger pipes in the plant could do more photosynthesis. And this was this relationship between the efficiency of water transport in the stem would be correlated with the photosynthetic capacity of the plant. And if you had these great big tubes in the stem, you could do more photosynthesis. And so this was a really interesting idea that I pursued kind of later, moving on to the leaves themselves to see whether that same sort of vascular constraint worked at the level of the leaf. And again, found pretty convincing evidence that the better the leaf transported water, the more photosynthesis that the leaf could achieve. And what's more, we found these very interesting patterns within the sort of phylogeny of land plants, which suggested that these sort of early branching groups like the ferns and the lycophytes had quite poor vascular systems, and that it was really the angiosperms that were the plants that had these very well-developed vascular systems, could move a lot of water and could do a lot of photosynthesis. And this was linked to the branching density of the veins, because it was the veins that were carrying the water in this case, rather than in the case of the stems, it was the size of the pipes. In the case of the leaves, it was the branching density. And we found this lovely pattern sort of through evolution that suggested that the ability of plants to branch their venation was really strongly correlated with their ability to photosynthesize. And we kind of addressed, it's always great to draw attention to yourself by addressing one of Darwin's great questions and the question of why, where angiosperms came from and how they became so successful. We proposed that it was only the angiosperms that were actually able to produce this high branching density leaves, high vein density leaves, and we suggested this was a key part of the evolutionary success of angiosperms. But you're sort of left with the question: okay, well, all plants have to do well is to make efficient vascular systems and photosynthesize. But there is a catch. Water transport's not just about moving water; it's about moving water safely. Plants have to develop very high tensions in their water transport systems to pull water, and this exposes the water, makes the water unstable, and makes it likely to cavitate, which is, it's not quite the right word, but it's where the system, because water's metastable, any nucleation will cause the vascular system to break. And there was this sort of connection between how well a plant could transport water and how well it could resist this process of cavitation, which is the thing that seems to kill plants during drought. But at this time, the way we measured cavitation was very sort of third party. We had to put plants into centrifuges and spin them at high speed and hope the centrifuge didn't disintegrate with the plant in there. And we had to pressurize plants, and it was very difficult to really understand. There was a lot of debate and a lot of quite acrimonious scientific debate about what was actually going on during cavitation. So I invited a colleague, Philippe Marmotton, who's a bubble physicist in France, and I thought we should be able to see the process of cavitation. If we could actually watch it happen, then it would be really hard to argue about whether it's important or not. And we developed this technique where we could actually, I wonder if this movie will work. This is a, oh, goodness me. Oh, there it goes. So this is a tomato leaf. It's in a cuvette. It's being exposed to very high humidity, very high evaporative demand. And at some point, using this sort of image difference technique, we could actually see that those red marks are cavitation spreading through the distal part of the vascular network. And so we were able to actually see the process of cavitation happening, in real life in the plant, and we could use that to connect, cavitation with downstream damage in the plant. We could see where cavitation was happening, when it was happening, and we could look at the leaf afterwards, and we could see, okay, well, the cavitation seems to be linked to where the leaf is dying, and we could really make a nice clear connection between these processes. So that was all very exciting, and I guess now we've sort of got to the point where we understand that water pressure is this really key part of understanding how plants work. It determines how fast water flows through the plant. It determines this damage, where plants are damaged. It determines growth. And what I'm really keen to do is to find a new way to understand this pressure, because the tools that we have to measure this pressure are just horrible at the moment. This sort of looks like it belongs in a medieval dungeon or something. You put the plant into a pressure bomb and you wind the pressure up until water spews out of it. It's very basic and very difficult to see, precisely what's happening. And so what I got together with Chris Lucani, who's a PhD student of mine, and we developed these cameras to see cavitation and see this process of cavitation, but we realized that we could also see a lot of dynamic movement in the plant itself during the process of dehydration. And we could sort of record this shrinking and swelling and convert that into a beautiful sort of heartbeat of the plant. We can see this diurnal shrinking and swelling of the plant. This is in a day. And, it finally, this is the first time that we could sort of see this dynamic pressure relationship in plants and understand this sort of the dynamic challenge of the plant to the things that are happening to it during the day. And we could, the moment I sort of discovered this, I've sent all my students out, and PhD students and postdocs out to put these sensors on equipment, on plants all around the world. We've got wheat plants, and we've been putting plants in the top of eucalypts. Because, I feel like finally, we can see this behaviour, and we can convert this behaviour into a quantitative representation of what plants are actually doing, how they are responding to their environment, and how different plants respond differently to each other. So it's this is a really exciting period now where we're sort of getting all this information and hoping to sort of convert this into an understanding of how plants use water in the field, how they store water, how sensitive they are to drying and atmospheric heat. But also in the same respect, we've got this a great application where we can because we can see how stressed the plant is from minute to minute. The applications into understanding crop water use and crop water demand are I think really very exciting. And so I would just like very quickly thank the future scientists who have been my PhD students and postdocs who've been a fantastic community at UTAS, and thank my wife who's been a very tolerant collaborator in the whole process. Thank you very much.
Thank you so much. I think we all share your enthusiasm, and as the driest inhabited continent, we so need your work. Opening it for questions, please. I've got a burning one. I can see Francis has a question, but if I'm allowed to ask the first one, what controls how many stomata there are, and do they all die and new ones pop up?
No, that's controlled sort of very early during the development of the leaf. The number of stomata are determined by a genetic program. It seems to be pretty largely genetic. And so that sets the number, and then it can be sort of diluted as the plant, if the leaf grows extremely large, the number of stomata might be diluted and dynamically responsive. And they sit there for the length of the leaf, so they'll function until the leaf dies.
Right, thank you.
Population.
Please, Francis.
It's good to see that you can do this in the field. So I wanted to know if there's a general statement you can make comparing, say, desert plants to tropical plants in the rainforest or something, in the vasculature, or is it just much more species dependent?
Yeah, I mean, I think characterising the vasculature, certainly species in the desert, woody species in the desert, we can very easily identify the characteristics of the xylem. It obviously needs to be extremely resilient to cavitation. And the same way the stomata, we can probably characterise the way that the stomata will behave. And this is part of one of the challenges of the Centre of Excellence that we're working in, is to try and understand, what are the constellation of traits and behaviours that really are successful in those sort of environments, whether they be desert environments or rainforest environments, and try and, the dream really is to, and I think it's a reasonable idea, is to translate those into crop behaviour and try and improve the way that crops perform under those sort of conditions as well. So thank you for the question.
I guess this is a related question. Can you say, is the cavitation pressure of -3 megapascals quite universal, or does it depend on species? I'm thinking of like giant redwoods, for example. Do they have a same value of cavitation?
Yeah, no, that's a great question. The range is enormous. So you've got, wheat plants that will cavitate at 2 megapascals. But then I'm very proud of our Australian Calitris. We have an endemic conifer which has the world record for being the toughest of all the plants on Earth, and it cavitates at around about sort of 20 megapascals, which is almost where you get sort of spontaneous nucleation of cavitation of water. So yeah, that plant has broken an enormous amount of equipment, but just by trying to spin it hard enough to make it break. So it's a really interesting trait, and a trait that's obviously very strongly evolutionarily selected. So yeah,
though. Thank you so much.
Yeah.
I think a wonderful example of research based in Australia, but with colleagues all around the world and calling many different disciplines into play. Thank you so much. I'm now going to hand over to my co-presenter, Professor Jim Williams.
Thank you, Lynn. Our next presenter is Professor Richard Payne. Richard is an international leader in the fields of organic chemistry and chemical biology. He is recognised for pioneering a number of technologies for accessing precisely modified peptides and proteins for applications in biology and medicine. Please welcome Richard.
Thank you. Well, first of all, I'd just like to say what an honour it is to be here, and also to thank my nominator for the Academy, who was David Craik, who's here and sitting up the back today. So thank you, David. So today I'm going to be talking about work that has spanned my entire career, looking at a particular family of natural products. And, you know, there are plenty of natural products out there that everyone will be aware of. They're very important drug molecules, and they get broken down into subclasses. So we have terpenes, such as the anti-malarial drug artemisinin shown here. We have polyketides that include antifungals and antibacterials. Alkaloids, so our cup of coffee this morning, of course, contains the very famous molecule caffeine. But, you know, my lab is really focused on a particular class of natural products called peptides and proteins. Peptides and proteins are built from amino acid building blocks, and we tend to classify these in different ways, but for the sake of today, a peptide will be less than 50 amino acid building blocks, and proteins more than 50 amino acids. And again, there are many peptide and protein drugs that have become very, very famous, you know, insulin being one of those, the antibiotic of last resort, vancomycin. And down the bottom here, I've got a couple of peptides and proteins that my lab has worked on, and you might see these a little bit more today. Now, the big thing for us is that these molecules are very challenging to access, and they're very difficult to make. And my lab has been particularly interested in making highly modified peptides and proteins, so things that you can't necessarily extract from the organism or make in bacterial culture. We really need to make these de novo in the chemistry lab. And, you know, I think this really exemplifies why we need to do this, and it's not just for the peptides and proteins we work on, it really is for many of the natural products that we want to work on. If we want to access the natural product from the natural source, we're only going to get very, very small quantities from quite a lot of the natural source. And here's an example from my lab. I was thinking, you know, Batman, tick guy, I don't think it really works, but we've done a lot of work on ticks over the years, and we've actually done these experiments. We've gone out to Terry Hills and the Northern Beaches, we've collected ticks, hundreds of ticks, to try and extract their bioactive proteins. And in one particular experiment, you know, 200 ticks gave us 50 micrograms of the protein that we wanted to work on. So, you know, you can't do much with 50 micrograms. You certainly can't be using this as a drug. And so the really nice thing about synthetic chemistry is that we get to build these natural products from scratch. That is an art form in itself. But more importantly, we get to add different modifications to these proteins that nature doesn't make. So we call these new to nature molecules. And if you think about this, I guess like the foodies talk about these days with their meals that they order from fine dining restaurants, you can really start to deconstruct these molecules and then reconstruct them again or reconfigure them to make new to nature molecules. And you know, that's the way I explain it to my colleagues that are not from the field and my daughters. I explain it like Lego, right? We've got all these Lego blocks that we've got in the lab that we can play with. And of course, they can be assembled in lots of different ways to make different functional biomolecules. And so how do we make these peptides and proteins? Well, it's actually a Nobel Prize winning technology that we base everything that we do on, and it's called solid phase peptide synthesis. We actually make our peptides and proteins on polystyrene resins using robotics. And we have these jars of amino acids that you can buy. Now, these are the proteinogenic amino acids. There's 20 that make up peptides and proteins, but more importantly, we can in the lab make these reconfigured amino acids, these new modified amino acids, and slot them in and grow these peptide chains using robotics on polypeptide chains. And if we want to make bigger things, we can actually take these peptides and start to join them together to make fully fledged proteins. And that's been a huge area in my lab that I won't be talking about today, but a very, very important area for making fully fledged proteins. So these are some of the natural products that my lab's made over the years. Don't need to dwell on the details of this. Needless to say that they come in lots of different shapes and sizes. This has been a program in the lab that spanned more or less since I started my lab in Australia. And I just want to give one example today of these smaller peptide natural products. This is a family of natural products called the sansamycins that were discovered from Streptomyces species from Japanese scientists. And what really attracted us to these molecules is they're kind of part peptide and part nucleic acid. So they had very beautiful structures and they'd never been made before. But more importantly, our lab was very interested in making anti-infective molecules to target TB. So the bug that causes TB is called Mycobacterium tuberculosis. And before COVID, it was the greatest infectious killer of humans and still is responsible for about 1.3 million deaths a year. So a real problem, not in Australia, thankfully, but certainly around the world, still a big problem. And so we use this sort of deconstruct, reconstruct type of approach here to build a very large number of analogues of these natural products. We could make the natural products themselves, but we could also build things that nature wasn't making. And again, you can see we can basically break it down into these smaller building blocks, and then using that polystyrene resin and building things up, we can start to make these structures using the robotics that I show you on the left-hand side. Whoops. And so we ended up finding a whole bunch of lead molecules. We screened these against the bacterium, Mycobacterium tuberculosis. They were potent against drug-sensitive strains of the bacteria. Importantly, very potent against drug-resistant strains. The big problem with TB at the moment is there's a lot of drug-resistant bugs out there, and we need new drugs with novel mechanisms of action to target those. And these molecules were also efficacious in TB models in vivo. And so this was a really great starting point for a drug discovery campaign. And the really important thing about this work was that we also were able to discover that these had a novel mechanism of action. So they actually target cell wall biosynthesis in Mycobacterium tuberculosis. And there are drugs out there, of course, that already target the cell wall as antibiotics. Things like penicillin and vancomycin that I showed earlier target this final step in peptidoglycan cell wall biosynthesis. And what we're able to show is that the sansamycins target an intracellular step where a cytosolic precursor of the cell wall gets transferred to the inner leaflet of the membrane. And this enzyme's called MERX, and this was the first time that people were able to show that this was really a candidate for TB drug discovery. And our colleagues in the US solved a beautiful crystal structure showing exactly how the molecule works here. It's bound in the inside of the cell to this transmembrane protein called MERX. It's a very complicated structure, and this was solved by X-ray crystallography with these analogues bound at the center. So I'm now just going to move on the last part of my talk on these modified protein natural products. And now we're talking about bigger things that we build by chemical synthesis, so more than 50 amino acids. And the example I want to give today is from blood feeders. So I talked about ticks earlier, but most blood-feeding organisms, or I think all blood-feeding organisms, including the vampire bat that we heard about earlier, they need to feed on blood to survive. And to do that, they produce very potent anticoagulant molecules to stop our blood from clotting when they're having a blood meal. And so the way that most of these work is they target an enzyme called thrombin. Thrombin is responsible for activating platelets that are part of the blood clot. But importantly, what they also do is they convert a soluble protein called fibrinogen, and they clip it up into this insoluble form called fibrin. And together, fibrin and platelets make up the blood clot. So that's how these molecules work. And so by blocking thrombin, of course, you can prevent a blood clot. So if you're one of these mosquitoes or flies or ticks that's feeding on blood, that's how they prevent your blood from clotting. And over the years, we have produced Proteins, modified proteins, these molecules are all sulfated. You can see there's a sulfate group out here. This is absolutely crucial for their biological activity, and we're able to show by isolating these from the organism and doing mass spectrometry that this sulfate was there, and then using biological assays that it was important. So these are very potent inhibitors of thrombin, so this Ki stands for inhibitory constant, so very, very potent molecules from leeches, from ticks. This is the tick from the northern beaches I talked about earlier. This is a South American tick, and you can see all of these contain these sulfate groups. And we've built all of these molecules de novo in the lab using those technologies I showed earlier. Mosquitoes, like the Anopheles mosquito that carries the malarial parasite, is sulfated. The tsetse fly also gets sulfated, and we've just started working on these flea proteins, which for some reason don't seem to contain sulfate at the moment, but we think there might be other anticoagulants out there. So just in the last minute, I want to tell you where we're going with this. So we've made hundreds of these anticoagulants, and this has actually led to molecules now that we're starting to try and translate towards the clinic. And the particular molecules that we're looking at, we're looking to use for stroke. So stroke is a big problem, as everyone probably knows here. And it turns out that there's only one therapeutic recourse for stroke, and it's called tissue plasminogen activator. It's a protein that gets injected after you have a stroke, needs to be delivered within 4 hours. It basically converts plasminogen to plasmin, and then plasmin lyses the clot. This drug works okay, but not particularly well. So 30% of patients, their vessels will reocclude and the clot will reform. It's known that anticoagulants can be used with TPA effectively, but all the anticoagulants on the market, unfortunately, have a serious risk of bleeding on the brain, and so they're contraindicated for use with TPA. And so from these tick molecules, we've actually discovered 2 lead candidates, and these are molecules that we're pursuing towards the clinic. And so very, very quickly, what we've been able to show so far is that if you take Herodin or Agatroban, which are clinically approved anticoagulants, you start to dose, and these just go through the roof, and they cause very serious bleeding. The 2 molecules we've got, you can use very high doses of our molecules, and we never go above a safe window. They can dissolve clots in vivo, so we can treat stroke, and we can treat thrombosis in a mouse model. And just lastly, we've been able to show that these molecules, when they're used with TPA, can actually stop these vessels from reoccluding. So here is tissue plasminogen activator. This is Doppler flow, so when you get this big long blood flow here, you can get blood flow to stop when you treat with your compound. And then what happens is, unfortunately, the vessel reoccludes. And when we treat with our drug molecule, we get opening of the vessel and it's stable recanalization. Whoops. So that was my last slide. I don't know, the acknowledgement slide went away, but I think it's time to finish anyway. I just want to thank my group. I've had a wonderful group over the years working on these programs, and all you for your kind attention. Thank you.
Thank you, Richard, for a wonderful presentation. And your analogy of playing Lego with molecules is a terrific one. Thank you. Now, we're running a little late, so we'll just take one question. So a burning question from the audience, and then you can also catch up with Richard later. Now, Lena, I'll just see if there's another question other than the presenter here. We've got one here. Yes, up here.
Thank
Andrew.
you, Richard. Brilliant lecture. 2 questions I'm going to squeeze in, Jim, if you don't mind. One is, Richard, can you tell us what scale you can make these molecules on in terms of taking them into the clinic and for clinical trials? And secondly, I know you're very much the person who takes the problem from the chemistry lab to the biological lab. So could you give us an example of that? I think the TB example was one where you've done that.
Great. So the answer to your first question, AIS 109 and 127 we've now made on multigram scales. Now, they're very potent molecules, so we don't need to use very much. For this to be viable for real translation, it would need to be made probably on kilogram scales. But, going from grams to kilograms, I think, is not going to be a problem there. So we think it is feasible. Usually more expensive to do than small molecule drugs, so to be honest with you. But the robotics that we have now and the scale that we can do things on, things are starting to get cheaper. And so I think that's why biologics, are sort of going through the roof in the clinic. The second question was around going from the chemistry lab to the biology lab. So more or less everything that we do, goes into a biological assay, whether it be in vitro and in vivo. Many of those things we do in-house in our lab, so students in my lab learn how to do the synthetic chemistry, but they also learn how to do the biological assays. But some things get very specialised, and of course we have a great bunch of collaborators that we work on, particularly for specialised in vivo models. We tend not to do them in-house.
Well, thank you very much, Richard. That's wonderful presentation. Our next presenter is Professor Xinghuo, and Shizhang is recognised internationally for his landmark work in developing catalysis principles and next-generation catalysts for clean energy. His work has reshaped materials development, pathways for catalysis, energy conversion and energy storage. Please welcome Xinghuo.
Good morning, ladies and gentlemen. So it's my big honor to give a presentation in this very beautiful building. So I'm Shizhang Qiao from University of Adelaide. Currently, I am leading the Center for Material in Energy and the Catalysts in University of Adelaide. I'm a director of ARC Training Center for Battery Recycling and the Deputy Director of ARC Center of Excellence for Carbon Science and Innovation. This center director is Professor Li Mingdai. So today, I would like to introduce my two research projects I'm working on. So first one is electrocatalytic energy conversion. The second topic is battery technology for electricity energy storage. So before I give my presentation, I would like to introduce a e-refinery concept initiated by my research group several years ago. So from this slide, you can see the black line is a traditional chemical refinery process. You can use natural gas or fuel or oil. You can use gas and oil as a free store. We can take a high temperature and high pressure. We got the fuel and the chemical and the fertilizer. Green line is the e-refinery process. You can see we can use a small molecule as a free store. So we take the reaction in the electrolytes at low temperature and atmosphere. So you can see we can take same or similar intermediate, and we got the same or the products. So this reason we summarize and initial this the concept. Actually, the e-refinery include many different reactions. For example, water splitting produce hydrogen involve the hydrogen evolution reaction and oxygen evolution reaction. So for carbon reduction reaction and some complex reaction. So today, I give an example. So for the water splitting. So here we talk about seawater splitting produce a green hydrogen. So why we use seawater? From this slide, you can see if we got the 1 kilogram of hydrogen, we need to 20 to 30 kilogram, the fresh water. So it's impossible in Australia and many countries to, use this electrolytes method to get the hydrogen because we need to a huge number of free water. Second problem is if we got the 1 kilogram hydrogen, we need to 50 kilowatt electricity. Yeah, from this slide, you can see the red zone. So, the face of ocean. So we have unlimited, the seawater. So in this area, so electricity can come from solar and wind energy. So it's have a lot of advantage in this area. Use seawater splitting produce green hydrogen. For seawater splitting process, we can take two the technology. One is the inductor, second is the director, the seawater electrolysis process. For the inductor seawater process, we need to use the RO process to get the free water. However, that's the RO water quality purity is not good enough for the PAM, water electrolyte, because, there are too many ions in the water. So we must pass a column for, the deionization process purifies the water. In that case, the hydrogen price increase significantly. So second problem is, the RO capacity is not enough. So we must build up a lot of RO plant, achieve the carbon zero target. Okay, so in my research group, we develop dialectal seawater electrolyzer process. We, use two technology. One is we use, the neutral seawater as a feedstock. So second one, they use alkaline treated seawater, for the water splitting. So actually, for the seawater splitting process, we face, two problems, it's two challenges. First one is chlorine and chloride produced on the anode. So that is dangerous for human body, and is, byproduct, the damage device. Second problem is calcium and magnesium hydroxide precipitate produced on the cathode. So, this cover on the surface of catalyst, lets the catalyst lose activity very quickly. So, yeah, so several years ago, my group initiated another, the concept we call the local environment effect on the electrocatalytic performance. In this study, we use this concept. We combine carbon oxide and copper oxide together to generate local alkaline, the local environment on the catalyst surface. It means on the, the double layer, so several molecular layer on the surface, we can make the pH value is 14. If that case, we can prevent, the set reaction on the anode and cathode. So, yeah, this, is a device test. From here, you can see this green line is, the commercial device performance. We use, very expensive platinum rhizome oxide, as a catalyst. We use pure water. So in my, the experiment, you can see we use a very cheap the catalyst. We use seawater directly. So you can see stability quite good. So currently we finish, scaling up study for, 100 watt, device and 1 kilo, test. So next, the company gave us $8 million to for commercialization study for this technology. Next, I would like to introduce, another research product is battery technology for electricity storage. So, yeah, in here we study. You can see we study the aqueous battery. So actually, compare with lithium-ion batteries, aqueous battery have a lot of advantage. For example, the very safe and price is low and power density is high. It means we can use a very short time, the finish charge and discharge process. Definitely, this the production process is simple, so save the money. So for aqueous battery, we can use that battery for the smart green and household solar panel system. So, however, in this system, we have a lot of problem on the cathode side and anode side and electrolytes. So, in my research group, we develop zinc iron the battery in cathode side, the different from traditional intercalation working mechanism. In also new battery, we take conversion reaction maximum, so we can make a battery, the cycling longer. So we also develop new electrolytes and add some anti-solvent, so we can prevent, the water decomposition. So, it means, we can prevent the hydrogen evolution reaction. So we can, increase energy density also. In anode size, we, built a solid and electrolyte interface layer, so we can, prevent the stability of anode. Currently, we hold for the patent. So this, all the industry partner, Iron Drive, the company, gave me the $1.5 million support my ARC industry Laurel Fellow, so for commercialization of this technology. Okay, so yeah, it's time over. So summarize my study. Actually, in my research group, so we focus on the material synthesis for different application and commercialization. So finally, I would like to thank financial support from my university and the ARC through the Laurel Fellowship and the Industry Laurel Fellowship and the ARC Center Training Center for battery recycling and ARC COE Center. So we also would like to thank the industry partner. That's my presentation. Thank you for your attention.
Thank you, Qiao, for a wonderful talk. Just before we open for questions, with your aqueous battery, how do you stack up in terms of the numbers of cycles you can charge and discharge compared with the solid state?
Yeah, so if we increase cycling time, we must solve the challenger in different the anode side, cathode side, and the electrolytes. So for the cold cell is easily, but for the the pouch cell, it's very difficult. So in my research group, so we can make a very big pouch cell, so we can run the 10,000 the circle already. So these reasons a lot of industry partner interested this product and give us money for commercialization. Thank you.
Questions from the audience? Well, it seems that Andrew and Lynn are asking questions. I'll take one from Lynn this time, Andrew.
Thank you for a fascinating talk on clearly something that's really futuristic but can change the world. I noticed you had the synchrotron up as one of the facilities that you use. Could you explain a little bit more about the role of that in your research, please? Because it's so central that we have that facility here in Australia.
Yeah. Actually, we use a lot of synchrotron, not only in Australia, but also in Japan and China. So yes, that's very powerful, the tools, because, if we want to study, the working mechanism, we must know the detail about reaction and reaction pathway. So, yeah, so this reason, so we use this as an instrument. So, and yeah, so I think that is a very benefit. So if we want to design the catalyst or material, we must combine the first experiment. Second is advanced characterization. It means the synchrotron. So third one is simulation. It's a computation. Currently, my group, study the machine learning and AI using the designs of catalysts also. Thank you.
Thank you. Andrew, Andrew, a very quick question. and a quick answer. And then I think we're...
Thank you, Jim. I appreciate that. Is it a limitation that we can only really in Australia get our water from the sea, and that's, of course, quite geographically limited? Alan Finkel told me electrons are cheaper to move than water.
Okay, actually, now, so we produce a very small number of, the hydrogen from free water, no problem. But if we want to achieve carbon zero, most of hydrogen come from the water splitting. In that case, we need a huge number of free water. So in Australia, I think the government never approved, this proposal, use free water. We must consider use seawater. Thank you for your question.
Thank you again, Shizhang, for a wonderful talk.
Thank you.
I'll now hand over to Lynn for the next presenter.
Thank you, Jim. It's my honour now to introduce you to Professor Brajesh Singh. Professor Singh is a world-leading authority and a soil ecologist. His work has provided direct evidence that loss of microbial diversity leads to direct loss of ecosystem functions on a local and a global scale. In addition, his global studies have demonstrated the central role of soil microbes in predicting, mitigating, and adapting to climate change. We welcome to the stage Professor Singh.
Good morning, everybody. What a wonderful presentation we have this morning so far. Can I start with saying what a privilege and honor to stand here and share some of my journey of the science. And I also like to start with thanking Professor Barry Pokshins of ANU. He's not here, he's in Finland, I think, for nominating me and giving me this opportunity to stand in front of you and talk about science. I should get on with my presentation because I don't want to be standing between you and coffee, so let's get with the talks. I'm going to talk about soil biodiversity, the unseen majority of the life on the planet Earth. Let us remind ourselves why healthy soil is important. Healthy soils is for basis of all the planetary functions, from primary productivity to the cycling of nutrients, carbon, and water, and provides habitat for other organisms. From the human health, if you look at these slides, from the human health perspective, 90% of our food, still today, 90% of our calorie intake come from the soils. And majority of medicine, we just thought about streptomyces and mycobacterium, they all soil organisms. They provide the medicines and all the biotechnological product and enzymes we're talking about. And this is why, this is no surprising, almost every sustainable development goals of United Nations requires healthy soils. Why healthy soils is important? Because it provides critical ecosystem function and services. What I'm going to talk about that, those services are carried out by the soil biota that lives in those soils. Let's remind ourselves first things that the soil biodiversity accounts for 59%. Majority of the global biodiversity live inside the soils, and that's why their activity is crucial. In simple words, there is no terrestrial life is possible without healthy and functioning soil biodiversity. And this is not something new we are talking today about. Through the civilization, human civilization, so the oldest civilization of Mesopotamia and Indus Valley civilization, they all talk about how important is soil health for agriculture productivity, so for the civilizations. For me, the most interesting one is that when Cleopatra was the queen of Egypt, which is 50 years before Christ, she forbidden the people to touch the earthworm. And the touching the earthworm was punishable by death, because they thought if you touch the earthworm, you will offend the god of fertility that will cause the less agriculture productivity, ultimately famine and the war. My journey wasn't that early, honestly. I did not know anything about soil biology until I went into my master degree, where I got into looking at the impact of the pesticides on the soil biology. And since then, I have been trying to answer, I put it another way, I'm trying to tackle two long-held assumptions in the science that we cannot describe soil biology, soil biodiversity, because it's so diverse and so heterogeneous. You have microbial diversity here and here, that's really far apart, so we can't look at the distribution as a driver or map them. And the second thing is that soil biodiversity is so immense that losing some of the biodiversity will not result into the loss of ecosystem functions. And those are the challenges we have been trying to address throughout my career. So this is just some of the results that we did make some progress. On the left side of the right side, I think, you're seeing there's a global atlas we produce for the bacteria, fungi, cyanobacteria, lichens, some of the small animals, looking at where they are distributed, so what are their driver, and what functions they do in the different environments. People ask you, what is the importance of that? Importance is on other sides of the things that we need to know what are the hottest spots of the soil biodiversity. If you want to conserve the soil biodiversity, if you want to harness soil biodiversity for ecosystem services, we need to know where are their hottest spots are. And that's what allows the policymakers to develop policies to protect those biodiversity hottest spots. I should have just mentioned in one word that initially, it was presumed that where you have a higher plant biodiversity, you have a higher soil biodiversity. That's not the case. 40% mismatch across the globe between the plant biodiversity hotspots and soil microbial diversity as hotspot. So if you want to protect the soil biodiversity, you need to protect those regions. The second question is good. I'm going to be very fast, as I say, that don't have a lot of time, was to tackle what is called the functional redundancy. There's too many species, losing some species will not result. Obviously, I'm a microbiologist by training, so we work at the microscope level, but for the policy decision, management decision, it has to be a scale. It has to be plotted regional and global scales, and that is what we have been working through all these scales. This is one example of the global scales. What I want to look at you, the ecosystem multifunctionality and the soil biodiversity. The things that take-home message from me on this diagram is all the relationship is linear. What it suggests that the loss of biodiversity will have proportional to loss of ecosystem functions they provide across the globe. But those are the, I guess, directly impact, you can't say that through the food and through the ecosystem services, they impact the human society, but there is other way that they directly impact human societies. We talked about it yesterday and today about the pathogens. Most of beneficial pathogens in our gut, in our airway, in the skins, it have a soil's origin. So the beneficials are also the soil provides some of the seeds. In fact, in pathogens, there are some nasty pathogens that lives in the soils, anthrax, E. coli, Salmonella typhimuriums, and Listeria, they live in the soils, and they find the way through the different factor to infect the humans and cause the human health. Antibiotic resistance, majority of antibiotic resistance is origin in the soil, and then it goes in transfer into the clinical settings. We have been looking at the distributions, the human pathogens in the soils, all the soil inhabiting human pathogens across the globe. This is just one of the example of the opportunistic pathogens, is Aspergillus niger. It's caused aspergillosis, and it kills around 2.1 million people every year, mainly in the immunocompromised people, but mortality rate is pretty high, more than 50%. And so what we are looking at, where they are distributed, what are their drivers, and how they move from the soils to the clinical setting, and what are the correlations between the abundance in a particular area with the infections rate between different regions of the world. Human pathogens in one aspect, so plant pathogens, I will argue, have even more impact on humans throughout the human civilization. There has not been an era where the human plant pathogen has not caused famine and war. This is just one example, is the Irish famine in 19th century that killed 2 million people, and just because of one pathogen, Phytophthora infestans, which caused the potato blight. So we are looking at the distribution again and what is the relation between the distributions and abundance with the disease outcomes. But one of the things I wants to take the message from here, the critical things what we found that everywhere you have a higher soil biodiversity, you have a lower plant pathogens, animal pathogens, and human pathogens. Simple rule, they occupy most of the niches, and the pathogens finds it difficult to find a niche within the soils to occupy. So lower abundance means lower infections rate and ultimately lower disease incidence. So this is just a global map of Phytophthora and Pythium, two of the plant pathogens. So we are not only looking at how they are currently distributed in the globe, but we are also looking what it looks like in 2050, 2080, and 2100, so that policies and the management policy and solutions being developed to tackle the issues. Because as I said, that we lose around 3 billion worth of crop everywhere just through the infections by the plant disease. Given it's a central role of everything for the human society, one will expect that there will be a very strong policy to protect our soil communities, soil biodiversity. Unfortunately, this was not the case until very recently. Along with working with providing the scientific evidence, we have been working with many global organizations, particularly to develop the policy papers to why we need a high conservation of soil biodiversity and how we're going to do. We had a small win in 2020 Montreal COP United Nations meeting where soil biodiversity is now included. Each and every country of United Nations from 2026 has to include soil biodiversity among their national reporting. I said a small win because it is a small win. Big challenges is, once this resolution was adopted, the United Nations passed that onto CBD, the Convention on Biological Diversity, to implement it. CBD, that's delegated to FAO, the Food and Agriculture Organizations of United Nations, to the NETSO, which is International Network of Soil Biodiversity, which I chair. I can tell you that significant number of countries does not have basic infrastructures and expertise to measure the basic soil biodiversity. So this is a global problem, and the solution needs to be found in the globally, and we have to make them, enable those countries, the poor low economic country, to meet these goals of the soil biodiversity conservations. Any work we do is the work of a number of people. I simply do not have a space to talk about. As you see, most of my work is global in nature, so all my work is done in global collaborations. But these are the peoples on the bottoms are from my lab. They have graduated, most of them, and becomes their independent science themselves. Some of are still in my lab. They have made all these science contributions we are talking about. Two organizations I should mention that most of my national work is supported by the Soil Science Australia. They have been incredibly helpful. And the global scale is a FAO that has been incredibly helpful to provide me all these opportunities to work together. I should not forget my wife and my 9-year-old daughter sitting beside me for their supports and their tolerance of most of the times I spend on the computer. So thank you very much.
What a wonderful advocate for a better world. My gosh, such important work. I'm very happy to open for questions, if I may, please. Yes, please.
Yeah, thanks for that really interesting talk. So the practicality of increasing biodiversity, I guess if you're, would you advocate for irrigating with a soup of biodiverse bacteria? And if you did that, do you think you would improve crop performance, or would you just increase pathogenesis?
You answer question yourself, thank you. Look, I am a big fan of precision interventions rather than the shotgun interventions. What we need to is to look for manipulating the targeted organisms rather than bringing soils from one area to other area. Restoration biology, that can work fantastic, because if this area is polluted, I can take those sites from there, which is nearby, or all microorganisms and beneficial and pathogenic is here and there. So I can bring that healthy soils here, but I will not suggest that we take something from the Western Australia to the Victoria or the Victoria to the Queensland, at least at this stage. Yeah.
Wonderful. Please, Francis.
Thank you. There's so much work to do. But I wanted to ask about the effect of fire and floods. So after fire or floods, do we see a change in the biodiversity? Do we see an increase in plant pathogens, perhaps?
So we see two things. I guess there's a PhD student who is working exactly the questions you have just put it through. It depends upon how severe the fire was. If the fire was severe enough to burn the top layer of the soil, you will see a drastic decline, and then it slowly is built up with a period of time. In some cases, that is possible. If the pathogens comes early on, they can settle in, the priority effect, settle in more early and cause diseases. But it will depend on a lot of factors, as you can imagine, and in any ecosystems, is that how much burn has been done and what is the recovery rate, and if the pathogens arrive first or if there is beneficial there before.
Thank you so much.
Thank you.
Again, really insightful work to bring about, I think, a better world. Congratulations to you. Now, it's almost the end of our session, but I think before I do the wrap-up, can we have a round of applause for all our presenters? They were so marvellous. And thank you to all those who asked questions. But we're about to have our morning tea break. I must remind the new fellows who haven't had their photo taken yet, will you please go to the Becker room to do so? We've had a marvellous session. It's going to be morning tea, as before, in the Jaeger room, the Dorothy Hill room, and outside in the marquee. And for those of you with dietary requirements, options are going to be available in the Dorothy Hill room. We will reconvene at 11:15 to hear more exciting presentations. Thank you all for a great session.
The human body houses trillions of microorganisms known collectively as the microbiome.
We're more bacteria in terms of our DNA than we are human.
Professor Elaine Holmes has spent much of her career decoding the chemical dialogue that happens between us humans and the bacteria in our gut. These gut microbes contribute significantly to our metabolism. They process our food and produce compounds from the nutrients we consume. Some are beneficial to our body, but others could be potentially harmful. Professor Holmes pioneered computational approaches to capture chemical fingerprints that are biomarkers of disease.
Every disease has its own characteristic fingerprint. So, for example, people with liver disease might have increased bile acids or increased acylcarnitines.
More recently, her team has been on the hunt for biomarkers of risk that indicate if a person is predisposed to neurodegenerative disorders such as Alzheimer's. It's research that could lead to earlier detection and even prevention of these conditions.
If we know which metabolic pathways are associated with neurodegenerative conditions, then we can try to optimize the performance of those pathways or modulate them by lifestyle choices earlier on in life.
Lifestyle choices including diet. But what does a healthier diet look like? Professor Holmes says there's no one-size-fits-all answer.
If everybody eats a particular healthy diet, like the Mediterranean diet, does it have the same effect on everybody? And the answer is no, because everybody has a unique gut microbiome, and they have unique genetics, unique set of environmental lifestyle circumstances.
So another strand of her research is looking at how we can personalise nutrition. That could extend to modulating food itself.
The same instrument that I put my blood sample into in the lab, I can also put in, an artichoke or an apple or some other carbohydrate. We can measure foods just as easily with the same technologies we can measure human body fluids.
Originally from the UK, Professor Holmes moved to Australia in 2019, along with several members of her Imperial College London research team. They were lured by the world-class facilities at the then newly built Australian National Phenome Centre here at Murdoch University in Perth. Since then, their team has continued to expand.
Science is really multidisciplinary, and because of that we have to search for expertise from different communities. So here we're really a big international family. We have, I think, 17 different nationalities here.
Now, as a Fellow of the Australian Academy of Science, Professor Holmes hopes to encourage even more bright young minds to join their mission in unlocking the secrets of the gut microbiome and its impacts on our health.
You're looking at the largest single dish radio telescope in the world. It's called FAST and located within the mountains of southwestern China.
And that is by far the most sensitive radio telescope in the world. I first went there while they were putting the first few things on the ground, and it was amazing to see how quickly in China you'd come back a year later and the whole dish would be built.
At the heart of FAST is a primary multi-beam receiver designed and built by Australian scientists. China chose to collaborate with Australia because of our experience with the Parkes radio telescope in regional New South Wales.
The Parkes radio telescope is by far one of the greatest telescopes in the world. It's been upgraded many times, so it keeps getting upgraded. It's not a 60-year-old telescope. The most recent upgrade was just a couple of years ago with a very wideband receiver system that allows us to observe the radio waves over a much wider bandwidth than any other telescope on the Earth.
Parkes has special significance to Dr. Shi Dai. His first visit was as an international student.
I mean, the second you see the 64-meter big telescope there with wonderful weather, blue sky, I was really shocked. I saw this is the way to do astronomy.
He later returned to Australia to complete his PhD under the supervision of George Hobbs, before accepting a teaching position at Western Sydney University, where his research focuses on rapidly spinning neutron stars known as pulsars. The Parkes radio telescope has detected more pulsars than any other facility in the world.
Radio astronomy in Australia is really strong. The experience and expertise here are really sort of the most attractive for a student in China.
That's thanks in part to the work of people such as Professor Elaine Sadler. For more than 30 years, Professor Sadler has been at the forefront of research into astrophysics. Her team discovered a 5 billion-year-old galaxy using the ASCAP radio telescope in Western Australia.
Now, 5 billion years ago, the Earth didn't exist. So in the time that signal's been travelling to us from this galaxy, the sun has formed, it's made planets, the Earth has settled down, it's got oceans, life has evolved, we've got a technology, we've built telescopes, and there we are just in time to catch this signal.
Professor Sadler started working regularly with Chinese scientists 7 years ago. She says international collaboration is critical to understanding our universe.
I think one of the great things about working collaboratively with people is it allows you to build up the trust and the common interests that allow you to make advances in science.
Astronomy is a very collaborative science. You can't just do your own research simply on your own.
It's about actually exchanging your idea with different people, different background, different expertise. If we don't have that, we are not doing the best science.
He's often referred to in the media as Batman. Professor Linfa Wang's superpower is his vast knowledge on all things bat-related. He's one of the world's foremost experts on bat-borne viruses and led efforts to trace the origin of outbreaks, including the Hendra virus in Australia and SARS in China. So when COVID-19 hit, Professor Wang, who had been considering retirement, didn't hesitate to step back into the action.
I use this, a quote of, success favors the prepared mind. I've been doing this for the last 30 years before COVID. And when COVID came, I feel like I'm so ready.
His team here at Duke NUS Medical School in Singapore was the first in the world to develop a test that can rapidly detect neutralising antibodies against COVID-19 in blood samples, an indication of prior infection and protective immunity.
We made a beta test kit, and we got a HSA, the Singapore FDA approval, and we launched as a commercial product in Singapore. All of that, 70 days.
The test, known as CPAS, could help untangle the starting point of the pandemic. In other words, identify how the virus first jumped from animals to humans and the early human-to-human spread, especially between those without symptoms. That's not the only mystery Professor Wang is working to solve. His research also offers novel insights into bat immune systems and why these creatures are reservoirs for many emerging viruses.
Bats, as a flying mammal, they have evolved a different immune system. Put it simply, that the immune system have a much good balance of defense versus tolerance than human. So if we get a virus, we fight very, very hard. Whereas the bats get a virus, they just fight hard enough so that they don't cause disease, but they don't necessarily clear the virus. What we want to learn is this anti-disease aspects that bats, can do better than a human.
Learnings that could help inform the response to future outbreaks of zoonotic diseases. In 1989, an unexpected phone call brought Professor Wang from California, where he was doing his PhD, to Australia. Without his knowledge, his close friend had submitted his CV for a position at Monash University.
The head of the department saw my CV and he was so excited. He called me that afternoon. So from Australia to California, and offered me a job, so the rest kind of is history. So I went to Australia purely by almost an accident.
After a couple of years at Monash, he joined CSIRO, where he made his first big breakthrough. The deadly Hendra virus had just struck Australia and infected and killed many horses. Professor Wang led the team that sequenced this virus and developed the Hendra vaccine veterinarians use on horses to this day. Today, he lives and works in Singapore. But Professor Wang says Australia will always have a special place in his heart, and he's honoured to now be elected a corresponding member of the Australian Academy of Science.
Australia means to me is my second sort of homeland. I was born in China, but my career in infectious disease was 100%, started and developed in Australia.
The scientists say this probably means they are dealing with some new deadly sexually transmitted disease.
It's been 40 years since the first description of AIDS-related illnesses. We still don't have a cure or even a vaccine, but experts in the field say that doesn't tell the full story.
40 years seems a very long time. However, the progress in HIV has been extraordinary. I mean, in many ways, it's to me the biggest and most important scientific advance in the last century, because we developed a way of managing a virus that had a universal death sentence and make it a completely manageable chronic illness, all through science and the availability of antiviral drugs.
HIV is a virus that gradually destroys cells used by the body to fight off disease. It can severely damage your immune system, making it much easier to develop diseases such as tuberculosis and some cancers. The most advanced stage of HIV is known as AIDS.
Even in Australia, about 1 in 5 people diagnosed with HIV are diagnosed so late their immune function has deteriorated to a point where they have AIDS.
But overall, new cases of HIV in Australia have halved over the last decade. In fact, Sydney is on track to become the first place in the world to end community transmission of HIV by 2025.
It's time to make history.
Effective elimination is defined as a 90% reduction in new cases over a decade. The big improvement is largely due to high testing rates and preventative measures taken by gay and bisexual communities. But infection rates in the heterosexual population haven't fallen as sharply. The Kirby Institute's Professor Rebecca Guy says the data shows why regular sexual health checkups are critical.
The test is like the gateway to a whole range of things that are important for a person. So you think about HIV, if you have a test and you find out it's positive, you can start treatment straight away and you can live the same longevity of life of everyone else and remain healthy.
While accessing an HIV test is relatively simple in Australia, the same can't be said for many places in the world.
People at risk of HIV, men who have sex with men, for example, there are still countries that criminalise that activity. And so how can you get a public health message out if the activity is considered criminal?
Even where it's not illegal, the stigma around HIV can sometimes drive down testing rates. That's one reason the Kirby has led the push for more screening options, including self-testing kits for people to use at home.
They're not quite as accurate as a lab test, but I guess the way I always think about it is having a test, even if it's less accurate, is better than no test at all.
Professor Sharon Lewin is a Fellow of the Australian Academy of Science and President of the International AIDS Society. As head of the Doherty Institute, she played a key role in fighting COVID-19, a virus that we managed to have vaccines for within 12 months. So why haven't we been able to do the same with HIV?
Well, HIV is a very different virus to COVID or influenza, which are viruses that can be completely cleared from the body. HIV is unable to be cleared from the body, and that's because it's a virus, part of a family, that's able to become part of a person's DNA.
And that happens within hours of a person becoming infected. Treatments allow the immune system to recover and can even stop the virus from being passed on. But the virus doesn't disappear. It's just hiding and will re-emerge if treatment stops. Professor Lewin has dedicated her career to understanding this phenomenon, known as HIV latency.
I've been on the hunt for understanding where the virus hides, how to mimic latency in laboratory models, and ways to coax it out of hiding.
While we don't yet have a vaccine, we do have access to a medicine that can help prevent HIV, known as PrEP. If taken correctly, the daily tablet can lower the risk of infection by 99%.
In Australia, we have a public health system that makes medicine free, essentially. We have a fantastically mobilised, vocal and articulate gay community that have done a lot of the education around accessing PrEP, and our uptake of PrEP is one of the highest in the world.
PrEP doesn't offer protection from other sexually transmitted infections, so doctors recommend the use of condoms. Professor Lewin says PrEP is just one of many advances in HIV research that goes to show the power of science. She believes it's only a matter of time before HIV is cured once and for all.
It's become a very important, high priority area of research because we now have treatment, we now have widespread access to treatment. Treatment's relatively simple. the next big challenge is finding a cure.
Deep thinking, where every move counts. That's a skill set Professor Andrew Wilkes has refined through chess tournaments like this one.
Chess taught me how to think tactically and strategically. In the sciences, those sorts of skills are incredibly important.
He was amazed in 2017 when Google DeepMind's AlphaZero AI program triumphed at chess against world-leading specialist software within hours of teaching itself the game.
If you could apply these sorts of tools to a similar process, drug discovery, it's a process that has some rules, many unknown unknowns, then you might be able to advance your programs that much quicker, that much more effectively.
And that's what brings us here to the Bio21 Institute at the University of Melbourne. Professor Wilkes co-founded a company based at the institute called Synthesis MedChem.
There are big machines, big toys, big equipment that can do amazing things here. And the sort of information that comes out of that, structural biology, analytical chemistry, those sorts of things are incredibly important for us as we develop our drug discovery programs.
Professor Wilkes is a molecular and cancer biologist. During his time at the Ludwig Institute for Cancer Research in the 1980s and 90s, he discovered and named the JAK family of kinases, enzymes that play a critical role in how cells interact with their environment. That discovery significantly advanced our knowledge of immunity, opening new avenues for therapeutics. Professor Wilkes is also very much an entrepreneur. Before Synthesis, he founded one of Australia's most successful biotech companies, Cytopia.
Making the connection between the science and cash or investment, that's a little bit of a skill. There's still a translation gap in some way, where the potential value of a piece of science is still somewhat opaque to the people who have cash to invest.
Professor Wilkes' mission these days is to generate enough investment for early phase drug discovery before then passing the projects onto larger pharmaceutical companies to complete clinical development. That mission is enhanced by machine learning technology.
The computer will develop an algorithm by which it can improve the way the molecules behave, and you kind of go along for the ride.
It's not all left to the computer. Ultimately, each hypothesis must be subjected to biological validation.
But you can get a long way down the path much more quickly, simply using these AI programs and computational chemistry. We're all cyborgs now, in many respects.
Professor Wilkes says becoming a Fellow of the Australian Academy of Science is a massive honour, and one he hopes will encourage more investment in the complex chessboard of translational science.
Our research and innovation is generating ideas and impact to deliver positive and lasting change for a better future.
As creators and innovators, together we work across diverse disciplines and 5 impact themes to solve the world's greatest social, economic, and environmental challenges.
We ensure the innovations of the laboratory and state-of-the-art technology translate to the ward and treatment rooms on health's front line.
We place sustainability at the centre of our research, prioritise the protection of all species, advance a circular economy, and support the transition to renewable energy.
With world-class research teams and resources to match our ambitions, we create smart and ethical technologies to improve lives and our economy.
We strengthen society, culture and commerce through creative, contemporary and intercultural approaches to education, the arts and business.
From increasing community resilience to cutting-edge cybersecurity, when we see problems, we build ethical and innovative solutions.
At Deakin, we're delivering positive and lasting impact for a better future.
We're in Chile's Atacama Desert, said to be the sunniest place on Earth. It's certainly one of the driest. But while it rarely rains here, the hills are often cloaked in fog that drifts in from warm oceans nearby.
So the question is, can I harvest it? Can I find some way of sucking that water out of the air?
Professor Gareth McKinley studies the rheology of complex fluids, or the science of how these fluids flow.
And really, we're surrounded by examples of things like that. So if you go into the kitchen, open the fridge door, anything like a mayonnaise or Vegemite or something like that is going to flow in a very strange way. It still flows like a liquid, but it may look like a solid to start with. You have to push on it to a certain speed before it'll actually start moving.
In collaboration with colleagues in Chile, his team used fluid mechanics to figure out an effective way to harvest fog in the Atacama Desert for much needed water. It wasn't a new idea, but their approach was. The fishing nets and meshes commonly used to extract water from fog tend to be far from optimal. So Professor McKinley's team undertook extensive lab testing to design an optimised mesh consisting of stainless steel filaments about 3 times the thickness of a human hair. They also coated it in a special polymeric substance that helps trap the water droplets blown across the mesh in the air, but also allows them to drain down into the collector.
And you could literally collect drinkable water at the bottom of it. In fact, you really don't have to do any purification at all. It's naturally desalinated. You can turn it into just bottled water almost instantly.
Their desert deployment successfully yielded a few litres of drinking water per day from each square metre of mesh, which is roughly the daily requirement for a single farmer. But they think they can do even better with different configurations. It's the sort of challenge Professor McKinley thrives on. One of his career highlights was working alongside researchers from NASA to better understand the spinability of strong, stretchy materials such as spider silk.
So with a spider spinning its thread, it goes from a viscous liquid towards a soft solid that can hold an entire spider's weight. So we were really interested in measuring the properties, or the stickiness or the stringiness of this. But of course, if you try and do that on Earth, then gravity dominates and your thread just sags to the ground.
He helped design special hardware known as a filament stretching rheometer that could be used to experiment with stretchable liquids on the International Space Station. Professor McKinley's other work is more down to earth. His insights on a range of complex liquids have been used in a variety of applications, from 3D printing to food manufacturing.
If I take the fat out of a product and try and replace it with something else that's vegan or vegetarian, then can I still make something that has the same taste? Or as what's used in the food industry, they call the mouthfeel. And mouthfeel is much more than taste. It's how does it feel in your mouth? Is it slippery? Is it slimy? Does it get stuck to the roof of my palate? And trying to match those kinds of properties is really what rheology is all about.
Professor McKinley first visited Australia as a student in 1985 and has worked closely with scientists here ever since, particularly at Monash University and the University of Melbourne. He says Australia has a strong reputation in rheology and calls becoming a corresponding member of the Australian Academy of Science a genuine honour.
There's very few corresponding members elected each year, and when I told my children this and they heard that David Attenborough was also a corresponding member, they were amazed. To me, if I can be mentioned in the same sentence as David Attenborough, then that's wonderful. But more importantly, it's really a great reflection of the collaboration and the science that I've been doing with colleagues in Australia for more than 25 years now.
This is where it started, and where you start. This is where you find a top 100 university, world-class research, and these little guys. This is where you study crashes and become cancer smashers. This is where you crush it, cram it, glam it. And this is where you grow food. Space. This is where your time begins. Make history at the University of Adelaide.
Professor Louis Moresi has some planets on the boil.
It's a big block coming down. Watch.
He often encourages his students to make lava lamps like these ones as a way of demonstrating Earth's evolution.
The density of the hot versus cold liquids, they play off against each other in these really complicated ways. So the blobs would be maybe continental crust,
And the background liquid, we think of that as maybe being a little bit like the mantle of the Earth.
Professor Moresi is an expert in planetary scale geodynamics.
My interest is in how the Earth formed and how it became how it is today, with plates moving, turning itself inside out, creating mountains, creating the oceans.
Earth is covered in tectonic plates. Interactions at their boundaries can lead to major geological events such as earthquakes and volcanic eruptions. Professor Moresi builds computer modelling tools to better understand the formation of those plates.
Now, the reason we're interested in that is because the process of plate tectonics stirs the whole Earth in 3 dimensions. So the plates going round into the interior and coming back out again, that recycles a lot of interesting elements, and those might well be elements that are important for sustaining life in the long term.
He also wants to know why Earth appears to be the only planet in our solar system that has tectonic plates.
And the really interesting question, I suppose, is if you look at exoplanets, planets around other stars, which might be a bit larger or a bit smaller, or maybe have a different history, maybe they have a different heat content, maybe they're made of something very slightly different. Would those planets potentially have plate tectonics? So the study of plate tectonics has very big implications for understanding whether distant exoplanets may be suitable for life.
One of Professor Moresi's software creations is called Underworld, which can be used in both laptops and supercomputers to study geological problems. He insisted on the source code being available to researchers everywhere.
It's very important to me because I really do actually believe in open science from that perspective, that if you want to do something, you want other people to be able to reproduce your work really quickly. You want other people to build on your ideas. So if they have access to the best tools, and I have a bit more time to think about how to improve those tools, then it's a win for everybody.
Born in London, Professor Moresi moved to Australia more than 25 years ago. Today, he's based at the Australian National University, which is also home to one of Australia's most powerful supercomputers.
It is sort of the national university, which makes it a little bit special. And, although we can access computers from anywhere, we can also access the people, we can talk to people. There's natural collaboration that develops with other people who you run into.
He sees becoming a Fellow of the Australian Academy of Science as valued recognition of the earth sciences.
It also gives a sort of an opportunity to let people know about the importance of the Earth as a planet, the planetary evolution, to be able to explain a little bit how the Earth evolves and why it's important to understand that.
Professor Donna Strickland's love for lasers began while attending an engineering physics course at university.
1/4 of the program was called Lasers and Electro-Optics, and I went, oh, that sounds so cool, let's do that. Because this is the 1970s, it's not now where everybody sees lasers everywhere.
Professor Strickland never expected to make such a groundbreaking discovery during her PhD that would see her recognised in 2018 with a Nobel Prize in Physics, making her only the 3rd woman ever to receive the honour. She found a way to produce ultra-short high-energy laser pulses, a feat that hadn't been possible because ramping up intensity creates pulses so strong that they destroy the amplifying material, similar to hitting it with a hammer.
We just figured out this very simple thing. Take your short pulse, stretch it and make it a long pulse, safely amplify the long pulse, and then when it's energetic, you can make it short again when it's outside the laser, ready for the application.
The technique, known as chirped pulse amplification, has many uses. It enables clinicians to...
This is the last session of presentations from our fellows elected in 2023, and I think you have probably all come to the same conclusion that I have, that this is Australia's biggest brain tickle. In this room you have heard the breadth of science delivered from the best in science, and it is such a treat. So I'm looking forward to this session as well as presentations from fellows elected in 2024 tomorrow. So our session today will be facilitated by two council members, Professors Glenda Halliday and Julian Gale. Thank you, Glenda and Julian. Professor Glenda Halliday is a pioneer in the field of neuroscience and is a neuropathologist specialising in neurodegeneration. She has shed light on Parkinson's disease, alcohol toxicity, dementia with Lewy bodies, and many, many more things. She has shaped international standards for the neuropathological diagnosis of neurodegenerative diseases and changed the way that clinicians diagnose these disorders, as well as her incredible fundamental discoveries that have enlightened us on the underlying mechanisms of these diseases. Professor Julian Gale is a chemist practising in computational science who constructs models of how atoms interact with each other and then uses supercomputers to analyse them to design new materials that can help with things such as clean energy solutions. Julian shared with me that as a child he used to play with leftover chemistry set and would mix chemicals together, then set them alight to see what would happen. I feel your family's insurance premiums might have been particularly high, Julian. But on that note, I hand over to Glenda to introduce our next speakers. Thank you.
Thank you, Anna Marie. I'd like to welcome Professor Leslie Weston to the stage. Leslie Weston is a pioneer in the study and application of plant interactions with other plants, microbes and herbivores. Her work has identified the role of various metabolites and biosynthetic pathways involved in plant defence systems, the regulation of plant-soil rhizosphere interactions, and the achievement of sustainable and effective weed management in diverse agro-ecosystems. Really looking forward to it, and please welcome Leslie Weston.
Thank you for the introduction today. It's a real honor to be here. I'd like to thank Professor Rana Munz and my colleagues at CSIRO who nominated me and also supported my original sabbatic, which resulted in the eventual application for a research fellowship that brought me here from Cornell University way back in 2007 to 2008. As a result of their support and Charles Sturt, I ended up coming and establishing my research program here, so I'm very grateful for that. Over the years, I've worked in bioherbicidal discovery of natural products with CSIRO and my colleagues Dr. Gerald Meinwald and John Clarty at Cornell University, and today I'm very grateful to them, along with my advisors at Michigan State University, who told me that it was always very important to know your enemy if you wanted to be successful in crop and landscape protection. This is kudzu vine, which is now a southern tropical weed in the US. It's also present in the top end of Queensland, and this has now been moving very rapidly into New York State. It's also present in the 5 boroughs of New York City, and this is what a derelict dairy farm in the southern New York State now looks like as a result of its invasion. And today I'm going to spend a little time not talking so much about bioherbicide discovery, but about invasive plants and their movement in Australia. So invasive plant numbers have increased dramatically, especially in the last 50 years. And this is really because of the emphasis on global transport, trade, and movement around the world. They've been introduced both accidentally and purposely, and many of them have escaped and for various reasons have established. This could be due to the changing climate, population movement and expansion, which results in land use management changes, and also the evolution of some very complex and competitive traits, including stress tolerance, water use efficiency, as Tim mentioned, nitrogen fixation, hybridization, and the development of novel weapons, which increase their ability to defend against competitors. And natural selection also plays an important role. Over the years, I've been studying here Australian invaders, and I've been recently working with my team and Paul Downey at Canberra to study how Australia's evolution of these invaders has occurred. And really, this is a relatively recent incursion. In the last 230 years, the beginning of this mass invasion of alien plants and animals occurred with the first colonists who came here and continued through the 18 and 1900s. But these invasions have had really strong impacts on the Australian landscape. And this is true because we have a very unique and isolated flora and fauna due to continental divide, which has evolved over millions of years. And while these invaders have occurred, not that many are invasive, but those that are have a really significant effect on landscape and agroecology here in Australia. And some of the biocontrol organisms that we've released to try to control these invaders have been quite successful, while others have completely failed. But what the resulting change is the significant loss of Australian native species. And right now we have greater than 60 species which have gone extinct and over 1,200 which are now listed at risk. So the invasives, the changes in land management, changing climate, and a variety of other factors have all seriously impacted our natives. And for that, we are increasingly concerned. I've developed a number of molecular approaches along with my team to study invaders here in Australia and to use these to actually identify these invaders, because many times they're improperly or taxonomically incorrect. So we've used molecular approaches to study identification, population genetics. We've also isolated and studied biosynthetic pathways and the genes that regulate them in terms of their defense metabolites. And we've noted that this gives us, these omics technologies, the opportunity for more work in functional genomics. And today, due to time, I'm just going to present a few examples of the plants and weeds that I've studied of interest over the years. These are two melon species that I've encountered while riding my horse on Charles Sturt property. And I first came, the first year I was here, to understand that these are called paddy melons. And while I didn't recognize them, I looked them up taxonomically, and I found that Citrullus lanatus was closely related to the cultivated watermelon, while Cucumis myriocarpus was a weed prevalent in the Middle East, but not native to Australia. Their introduction to Australia, historically speaking, we did some delving in the literature, and we found that these two plants, which had almost nothing published on them, came into Australia with the camel root. And they were introduced from the Middle East, India, Pakistan, Afghanistan area, with the development of camels for transportation in the 1800s. It was thought originally that camels ate these melons, but we found that that is not the case. Generally, the saddles that these camels carried were packed with these dormant seeds, which eventually germinated and expanded throughout Australia. Today, these two melons are amongst the most prevalent species which are impacting summer fallow ag production areas across southern Australia. And they're extremely prevalent and do very well under drought conditions. And they're also quite difficult to control. So today, their distribution mirrors that of some of those camel roots. We discovered that prickly paddy melon and camel melon had very complex seed dormancy attributes. They have very hard seed coats, which are difficult to degrade. Because of that, they last for years in dry soil. And we saw considerable morphological variation amongst their biotypes we studied, but we determined that their taxonomy was quite unclear. So we worked with the Australian National Herbarium and Brendan Lepshy, and we evaluated their taxonomy using DNA sequence analysis, and we soon discovered that the camel melon reported as the watermelon, cultivated variety lanatus, was actually incorrectly identified, and we've renamed it now as the citron melon, which has come to us from the Middle East. All of these species are drought tolerant. They're exceptionally good at competing weeds, particularly annuals. They grow in irrigated cropping areas, but they've now spread into very droughty soils as well. And they're quite difficult to control and manage because of this complex seed dormancy trait. We've also studied one weed that you might be familiar with if you live around Wagga, Canberra, or northern Victoria. And our colleagues call these tumbleweed species, but we know that they're a complex of panic grass species that produce inflorescences that tumble or move up to 100 kilometers a year. And they've spread northward from the Adelaide area in southern Australia and have now become exceptionally prevalent. And you'll see many news reports of these bioaccumulating around people's homes, across highways. They can cause fires due to sparking of agricultural vehicles and automobiles. They also are really problematic for agricultural producers because the toxicity of panic grasses is quite severe. And for grazing sheep in particular, we see severe liver damage due to their production of steroidal saponins and photosensitization, which results as bioaccumulation of these saponins actually occurs. They're an interesting species, and that we've worked together with a group of veterinarians and livestock specialists to identify the signs of disease clinically. We've studied the photosensitization, and we've noticed the actual problems associated with photosensitization. The diasgenin and protodiasgenin are really problematic. They're very invasive because they move very readily, they're drought tolerant, and they've been introduced and moved over the years as the wind and water propel them across the landscape. Most recently, we've discovered that this species, panic grass, is actually Hillman's panic grass, and it was introduced as a fodder species from Texas. And this is the primary species of interest and the one that's most toxic. And so we are now working on identification of these steroidal saponins, and we've noted at least 10 of them are prevalent, particularly in Hillman's. The last weed I'd like to briefly mention of importance is that of Patterson's curse. This is Echium plantageneum. It's a native of Andalusia. It's lovely. It was thought to be introduced as an ornamental, but we found that it's been introduced primarily with the grazing livestock industry, particularly the merino sheep, which have been brought over from Spain and Portugal as we established our colonies here in Australia. And we've discovered a novel group of chemicals present in the roots of Patterson's curse, which protect it almost like a glove along the root system. These are chiconins, and we've discovered at least 22 of them, which are exuded directly from living root exudates, but also bioaccumulate in the root periderm, protecting the plant against invaders, including bacteria, fungi, and other plants. The leaves of this plant are also extremely interesting in that they produce a complex group of pyrrolizidine alkaloids, at least 20 different alkaloids which accumulate primarily in the basal rosette, but they also accumulate in the leaves, in the flowers, and the stems. And these are also toxic to grazing livestock. The main prevalent ones are eshimidine and ethiamine, which are very, very active at low concentrations, causing hepatotoxicity as well. So these plants, together with their ability to produce large volumes of seed, to be able to live and undergo drought conditions, and to develop these interesting bioactive metabolites, have all established to have a real impact on the Australian landscape. What we'd like to suggest is that we can use plant genomics to study the genomes of these species. And interestingly, with respect to invasive weeds, only less than 30 plant genomes of invaders have actually been sequenced to this point, and the complete genomes represent, again, only about 30 at this time. So given that we have over 3,000 invaders in the world which are globally important, it's very important that we think about supporting research to use these as reference genomes and to study them to be able to report the importance of weedy traits and also consider using these weedy traits for the development of more competitive crops. So this emerging technology should help us in being able to study the reasons why these plants are so environmentally important and have adapted to our climatic conditions across the globe. I'd like to thank the people who've supported my research, especially here in Australia, including my CSIRO colleagues, my colleagues at Charles Sturt University, the 25+ students, staff, and postdocs who've worked with me over the years, and also my funding partners at a variety of institutions ranging from the ARC to various RDCs which have supported this work. And I'd also like to especially thank the producers who've been very grateful and helpful in supporting our research to try to find some solutions for these invasive pest problems. Thank you.
Thanks very much, Leslie. Unbelievable, so many alien species, and hopefully the 1,200 at risk, you can potentially help that. Open for questions.
There's one up in the top.
And there's one at the top, is there?
Yes.
We'll start the one below and get the one at the top.
Very nice talk, Leslie. Thank you. And just wondering, most of these weeds seem to produce toxins against animals. Is that an important feature of their spread, or is that simply for survival?
I think originally many of these would have been developed for plant defense characteristics, protecting the root or the shoot, or being able to support themselves against other pests. But grazing herbivores are an important factor in their ability to survive. So many of these same defense compounds have highly potent effects against herbivores, particularly younger animals like young sheep, cattle, or even horses in particular.
And up the back.
Hi, I'm Bernal Lidas. I'm a research scientist at CSIRO. So I probably have a silly question. So I'm just wondering, why do some plants seem to have much better defense mechanism compared to others in terms of, does evolution has something in that aspect?
I think natural selection and adaptive evolutionary processes are the reason for that. Sometimes these occur over millions of years, but in other studies we've done, we can see real differences that have occurred probably in a matter of 100 years or more. So for example, Patterson's curse in its native range in both field and common garden experiments produces much less chikunins than those Patterson's curse that have spread throughout Australia. And they produce higher levels of pyrrolizidine alkaloids in the native range. So competition above ground in the native range is much greater, while competition below ground for resources in Australia is much greater. And so depending on those forces of natural selection, sometimes these things can adapt quite quickly, whereas other things in terms of biosynthetic pathway development would have occurred over millions of years. So it depends on the time and the pressure of selection.
Thank you.
Okay, thank you very much.
Thank you.
I'll now hand over to Julian.
Thank you, Glenda. Our next presenter is Professor Xinghuo Yu. Xinghuo Yu has led pioneering advances in discontinuous control theory and its applications, terminal sliding mode control, and complex network systems. These developments benefit both industry and society more broadly, and have been directly applied to address industrial problems in areas such as smart grids, robotics, and cyber security. Please welcome Professor Xinghuo Yu.
Thank you very much. Firstly, I really would like to thank the Academy for the recognition, and I would like to pay special thanks to Professor Brian, my nominator. Another person I really want to thank is, which actually brought me here, is the late Professor Ren Potts. Many of you probably know him. He's an applied mathematician in the University of Adelaide. And he took me to Australia at the time. I was a struggling sort of PhD graduate. I was actually working at the electrical company selling lift technology. We call it variable voltage, variable frequency equipment for the lift, control the lift speed and art. I was a little bit frustrated. I was asked to go to Shenzhen to open up a company. Right? And I got an offer from Professor Ren Potts, and I said, hey, there's something in me, right? I had to do some bit of research. So then I took that offer, went to Adelaide. So to my surprise, and well, to my sort of shock, that's the first time I work in a mathematical department. So you still remember I worked with Professor Ren, a piece of paper and pen and chalks and whiteboard, a blackboard, doing the fundamental mathematics. So that opened my eyes. So eventually I think he's quite proud of me, called me an engineer mathematician. So today's talk is actually that kind of journey where I start from there, eventually leading up, I'm here. So thank you. Well, one of the things, so you probably wonder why, as an electrical engineer, I'm standing here in the Academy of Sciences, the electrical engineer. You probably realized that that one of the things, electric shock is the one that can kill you, right? I mean, everybody's afraid of you. So, but as I said before, this kind of thing, I love when I was a child to play around with electricity, but just remember, a right dose is actually give you a comfortable sensation, which is, I'll keep going doing that. So I'm glad I'll continue to do that work. So, but if you look at this, the industry where we started as a human being, as a civilization, we've been here for, the last couple of thousand years. It's only in the last 200 years we suddenly progress so fast. You can see this is the term you will read in the media, in the newspaper, in the articles about industrial revolutions. We start from the end of 18th century about mechanization, to the mass production and automobile in the beginning of 20th century, to the 70s to 80s, computer automation, robotics, and today, we usually call cyber-physical system is because everything is connected through the internet. And of course, the future is going to be. So the essential role of that is the control, right? But control, we all understand what control means. Control means you have a goal, and you design steps to achieve the goal. That's all we always do. So the control here, when we talk from electrical engineering point of view, is a mechanism, an algorithm, an approach to our steps to design the machinery, follow exactly the pathway we want to reach. So or the machine wants to reach. So that's what we usually call the control strategy. So one of the fundamental, I'm not going to do a sort of introduction of control theory, but one of the fundamental tools of control theory is the ordinary differential equation we all encounter in our sort of first year calculus. That's actually underpinning all the physical process, well, most of physical process. And the question is, how do you design this strategy, follow the physical law to reach your goal? So that's basically what we do. So the thing is, the majority of that is based on what we usually call asymptotic stability. You probably heard about eigenvalue, eigenvector, basically talking about how the system trajectory will converge to a solution asymptotically, right? So this is actually the fundamental. I'll show one of the things. Here we are, okay. So one of the things have been doing for many years is this continuous control. This continuous control is basically you switch things on and off, very easy to understand. But this is actually human intuition as well. This is the fastest way. If you don't find the tool works well, change it. In the physical sense, you can do the same, right? Usually you can get pretty good response or even optimal solution. So that's the theory will tell you. But mathematically, it introduced the challenge in the differential equations because most of the theory are not built based on the discontinuity. In particular, this is the general discontinuous property. So that's causing quite a bit of problem in mathematics. So, but the question is, of course, you have to use it, right? So you can see that if you look at the right-hand side, so this is probably give you intuition. When you drive a car, if you say, I want to control 60 kilometers exactly per hour, then you realize how your foot would operate so fast. It got to be operated very fast, very, very fast. That's actually reflect how the control system should be. But there's an issue of doing that, right? The issue of doing that. What about if you reflect not fast enough? So what is going to happen? You just can't. So the car will be shaking, right? Right. If you, if you switch in a wrong time, you go to back, you hit someone, right? So this is, so that's actually mathematically is one of the issues. As an engineer mathematician, this is something we study. We look upon the mathematics, look at the solutions. If there's nothing there, we create the solutions, and then we see how much that would impact in the mathematical discipline. So that's, we sort of come forward doing those things. So here is one of the things, I just pick up one of the key things we're doing. So sort of maybe being known for is we call this terminal sliding mode term. So on the, if you can hear here is usually this is first the differential equation. You can easily see this is first order differential equation. Solve it, then you got asymptotic stability. And then you probably also encountered this, the second one as well, have a little bit of fraction. Then we know that you can integrate to get a solution. But there's a beauty in it. Beauty in it is in the first one, the time, when you exactly reach the target, it's infinite time, right? You never get there, right? So that's the problem of that. But the second one is you actually arrive at that time exactly. But the reason of that is this is nonlinearity, give the little kick up, to speed up the whole thing. So that's actually fundamental, very basics. But the question is, for that very simple equation have so much engineering properties, what are you going to do with it? So we start with, this is the idea. So so we thought about, what about the Russian doll, right? You just build one after another. And then you channel together. But then the problem is there's mathematics in it. You cannot do that because there's what is singularity, all these issues. So what we found is the pairing conditions, how the parameters should be arranged, how the relationship, so that we don't have these kind of problems. So that seems to be a very basic idea. So there we start the whole thing theory again from all different aspects and apply. But if you're on the right hand side, you can see that we start in 1920. There's only a couple of thousand citations. Now it's just grown so many times. So people started picking up this kind of idea and then just using it for, here's for control, of course. This is we usually call finite control. For the signal processing, even for the neural networks, deep learning is one of the things that can be used. Deep learning is, if you look at differently, it is a control problem. The only thing is you stretch the time axis, right? You don't change action very quickly. You stretch the times. So from that point of view, deep learning is basically kind of a control problem. So we use that algorithm, and other people use that algorithm to demonstrate how sort of effective this kind of algorithms can improve the whole speed. We're also using many applications. You can see that they can be applied for many areas. Robotics certainly is one of the things. AIMO National Control System is one we just finished a work with CSRO and AIMO about the control room of the future. Is looking at alarm management, is looking at signal processing. How, they have 200,000 alarms, every hour. And how do you do it? And all this kind of on and off, the whole network is on and off a switching network. How do you do it? So there's the algorithm can do that. We also, my student, actually PhD student, finishing the degree with the theory, and one Caterpillar seems to be doing very well there. Basically control the Caterpillar, 2 trucks, for example, how to improve efficiency, so the accuracy, so that 2 trucks can move without human intervention, move within, let's say, 200 centimeters. So, and of course, media control. You can use the media control is one of the another examples. Okay, just to pick up a few. As an engineer, without impact, of course, we don't think the job has done, right? So we continue to pursue the work. We have done this work for the Victoria utilities using signal processing, trying to capture the signature of this current leakage, which caused actually 2009 Victoria bushfire. So we found the reason how that happens, and we found there's as a byproduct, we found that the current wooden pole maintenance is not satisfactory. Every 30 years they change it, but that's just not good enough. So we say you got to change it 20 years because that's start deteriorating. So that's actually they adopted. Another thing is we look at the network, AEMO's management over the network operations, and then sometimes they manage the network more thinking about economy rather than actually physical systems. So we show that you can't do that anymore because the whole system will collapse when the frequency changes. Your equipment will be died very quickly, deteriorate. So I think eventually they took the advice, they changed the rule. The last one is, I just want to show you, is we apply this, if you think about this money laundering network, it's just like social media. People on and off or sending money around. So we were asked to develop this algorithm without knowing anything how to search it. So this is also mathematics. We look at some Fourier transformation, some fractal mathematics, try to capture a picture. And they are doing very well. Eventually, it's reported in news, scientists, all our PhDs were took by them. But the problem is when you ask the impact to these people, because they are the financial information intelligence agency, then they talk about weathers. They never talk exactly what happened, how effective. But the only thing I knew is that PhD student become the director of science in Australia. So it means we are doing very well. So overall, I think I might just stop here. So I would like to thank many people who support my research, and the colleagues, the students, and my families for that. Without that, I wouldn't have been here today. So thank you very much.
Thank you, Xinghuo. It's great to see we're continuing yesterday's theme of having more mathematics in the talk and showing the value of mathematics, and as well as the play element. So the fact you came to engineering by playing with electricity. So
thank you.
We now have time for questions from the floor and online. Francis.
I have a couple, but I'll start with one. So I wanted to ask what you meant by the concept of stretching time.
Well, the thing is that stretching time means there's different way. How do you, like humans, right? How do you watch feedback that, for example, if you want to approach something, you look at the position, and then you feedback how far you are, and then you make your next move. And the time, how long you wait for that take. So what I see, that's my theory, right? If I require you change your position immediately, that's control theory, because you just need to constantly change it. But if I want you to learn, you don't have to change that as often. So you could be, depending on the time you require to do, or depending on event, what happened, the learning will take place. That's what we usually call optimization, right? So it's just a different time. That's what I see.
I was hoping you would use it as an anti-agent. But if there's not another question, I just quickly want to ask about bank card fraud. Is a similar thing used for, like you do for detecting money laundering?
It is. What happened is that what we were told that they have usually any $10,000 transaction will be recorded in this agency. So they have millions of them every day. The question is, if you know some criminal, you can easily find out the network. So you detect it. But the problem is there's so many of them. How do you find it? So it's just like finding a needle in a hay. So what we do is we narrow down this impossible search task to just a few thousand so the human expert can focus on that. I think that's basically is our work, yeah. To find the pattern, to find the signature and pick it up and let them investigate. Yeah.
I don't see any other questions, but I think to keep on time, we should.
Yeah. Okay.
Please thank Xinghuo.
Thank you.
For the wonderful job. Thank you. Our next presenter is Professor Gareth McKinley, who's a corresponding member who is visiting us from the United States. Gareth McKinley has made outstanding contributions to viscoelastic fluid mechanics, understanding flow instabilities and stretching flows. He's led the development of novel instrumentation and customized rheological analysis techniques that have driven the field of rheometry for complex and soft fluids. Please welcome Gareth McKinley.
Wonderful. Well, thank you very much to the Academy for inviting me. It's a great pleasure to be here. I'd like to particularly thank my nominator, Tam Sridhar, who's in the audience somewhere, as well as a co-nominator, David Boger, who unfortunately, due to health reasons, can't be here. But I really want to dedicate this not just to my students, but also to my family and my wife, who's here, who were willing to pick up everything 22 years ago and come on sabbatical here. So we brought 3 children under 8 who came to the school system, spent 7 wonderful months in Australia, and it was the beginning of a lifelong love of coming to Australia. So it was really fantastic. They're watching at home. They all had Australian accents for a while, went back home and, of course, immediately became American again. But they really enjoyed their time here. What I'm going to tell you about is a class of materials that I'll call complex fluids. And they're ones that probably everyone here is familiar with. In fact, if you put deodorant on this morning, or if you brushed your teeth, which hopefully most of you did, then you interacted with a complex fluid. Complex fluids are really everywhere in the world around us. So particularly in Australia, the mining and resource extraction industry makes huge amounts of complex materials, usually as waste products or byproducts, but clays and minerals and drilling muds and materials like that are all examples of fluids that don't flow like a Newtonian fluid like water or air, which are the kind of things that we learn about when we're students. Whatever you eat is largely a complex fluid, so particularly the mouthfeel of many materials is extremely important. When we say, is something creamy or is something chewy, what you're doing is really acting as a rheologist, and your mouth is telling you something about whether this is satisfying, pleasing to eat. Ketchup is a wonderful example of a material that flows, but it really doesn't flow like you expect it. You often have to hammer on the bottle to actually make the material come out. I think in the pandemic, probably the most important complex fluid that we've all interacted with is hand sanitizer. And anytime you've put one of that, a sample of that on your skin, you'll be very familiar with the idea that it has to sit there on your skin. You, as a rheologist, will actually then spread it over the surface of your skin to hopefully cover a large and uniform area. So these materials are everywhere in the world around us, many biological materials. I'll show a snail here. We've worked a lot on the thin lubricating film that's secreted by a snail. It's a mucus film. I'll come back to mucus later on. Spider silk is another great example. In fact, if you have a 3D printer in your lab, or anyone you know has a 3D printer, then it really relies on the rheology of complex materials to be able to produce the object. We just heard about money as being important. Australia is an expert in this. Anyone in your pocket who still has dollar bills, these are processed as complex fluids. This is a plastic, and the ink that's printed on it is a colloidal ink as well. And so you're actually dealing with 2 different classes of complex fluids in Australian currency, and Australia's really led the world a lot in that. You're probably all familiar with the phrase blood, sweat, and tears. Those are all non-Newtonian fluids, and they're all complex fluids, and they're all ones that actually I've worked on a little bit over the time. We heard 2 talks ago when Rich talked about clotting. The formation of a clot is something that takes just a simple viscous Newtonian fluid and turns it ultimately into a solid, and nature is extremely good at controlling that process. As we've learned, there are different ways of either slowing it down or speeding it up. So the formation of clots is a critical example of a non-Newtonian fluid. So the science, the study of this is called rheology, and there's a society of rheology. It's not theology, but it's next to theology, okay? There's also a very active Australian society of rheology, which I've had a great pleasure to interact with over the years, but it really is the study of the science and deformation of flow. And we all have a great experience of this. Anytime you put a knife or a fork or a spoon into a material, if you ever ask yourself, is something slimy? Then what you're really doing is a rheology test. You put a material between your thumb and forefinger, You might call that a rheometer, and you generate a sliding flow or a shear flow. And many materials, the viscosity, which I've called here eta, decreases as you try and rub harder and harder. That's what makes something slippery or slimy. However, there's another adjective that we often use, and this is a much harder one to measure, which is when someone says, is something sticky? And sticky is a measure of viscoelasticity, and it's what you see when you see the formation of long, thin threads, whether it be a silkworm thread or a fine thread from a material. And the idea is we actually want to stretch materials, and so that's one of the things that's mentioned in my citation is the ability to stretch materials that are liquids and form thin threads of material. So you can do that with your thumb and forefinger rheometer. In fact, in many ways, it's the instantaneous thing that you think of doing when you see something is sticky. And this shows up everywhere. Almost every part of processing industry has a different adjective that means the same thing. So in cheese, it's called stringiness. In adhesives, it's called tackiness. In yogurt formation, it's called ropiness, and I'll just say you don't want to eat a ropey yogurt, okay? In shaving, it's called pituity. In ink printing, it's called texture. There's many different adjectives for something that we all interact with, but really the time that I spent in Australia in TAMS Lab was trying to figure out a way of actually measuring this in a systematic way, and so that's what we've tried to do. These effects are difficult to measure because they're very frequently transient in nature. This middle image here is a good example of a small droplet of a pesticide being atomized as it's ejected out of a plane. And the formation of a bag and the formation of droplets is critical in getting good coverage of an atomized pesticide over, say, a crop. Foods, I'll call this okra because this is what it's called in the United States. Many of you here will call this a ladyfinger. Those of you who have been to Japan, a wonderful example of a stringy, sticky material is natto. The formation of these fermented bean curd threads is a high polymer that produces this idea of viscoelasticity. Adhesives, of course, are full of sticky and tacky materials. So the challenge is these things happen briefly, we all see them, but how can you measure them systematically? And that's one of the things that I set out to do when we were here. So I spent time 20 years ago, this is a very young version of me, 20 years ago with the filament stretching group that we built a very big exercise in control theory, in fact, it turns out, to build something, because the challenge is that we wanted to build an instrument in the lab, and the challenge is that you need to move your thumb and forefinger apart, not simply and linearly, but exponentially fast. And so the challenge is to move things apart exponentially. That allows you to stretch material elements. And so here's some experiments that were done in TAMS Lab. The filament stretching rheometer is taller than we are, and you'll take a small sample, pull it apart, and then these are simulations that we did that showed that we could actually exactly match what was done experimentally. Nature does this all the time. Here's some pictures of a simulation of what happens in a Newtonian fluid. The filament will thin down and break in the middle. In a material like silk, however, the stretching of the polymer molecules that produced there will actually stabilize the spin line, and so that it can evaporate and then form a stable thread. So next time you watch a spider spinning a thread, you'll notice that what it does is form a filament. The elongation of the molecules stabilizes the thread until heat can dry it out, and then you produce a material that can no longer actually be dissolved in water again. So nature's extremely good at taking water-soluble products and making non-soluble materials from it. So this was work that was done here in Monash. David Boger's name is associated with the class of fluids that we use to validate them, and this was an instrument that then became used widely around the world. But there's an easier way of doing something very similar, which you can all do afterwards, which is to go fishing under your tongue for a sample of saliva. So sublingual saliva is high in mucins, and mucins are very viscoelastic materials. And if you try this, then what you'll find is that you can see that the droplet doesn't break up the way you would normally think a thread of water would. So if you tried this with just water, you'll find that it just breaks into droplets. But instead, you'll see that you'll form a structure that we call beads on a string structure. And surface tension here is actually a very strong force that takes a mucin network, there's both a micrograph and a sketch here of mucin network, and elongates it and forms this long, stable, thin filament. And so beads on a string structure show up many places when you're trying to produce long, thin filaments. We figured out that actually if you do just a moderate strain and you do this very carefully, then you can actually build an instrument that really is nothing but a quantitative version of the thumb and forefinger test. We actually started a company, spun it out, and then there's a number of these instruments in labs around the world, including a number of labs in Australia, that really lets you ask quantitatively, how stringy is this material? How sticky is this material? What can I do to modify that by changing, for example, pH or temperature or materials like that? So I want to end in this by saying, well, now that you're all rheologists, you all understand what extensional rheology is. It shows up on fine scales, on short times. You might ask, why do I care about this? What's important about this? Well, next time someone sneezes behind you, I want you to have a mental image of exactly this. A sneeze is a great example of what is called euphemistically a violent expiratory event. That really doesn't do it justice, but here's an example. This is Barry Schaffman, a graduate student at MIT, and here's a great example of seeing extensional rheology in action. Okay, so this is why you put your hand over your mouth when you sneeze, right? This is my colleague Lydia Buriba's lab at MIT, and she's very interested in how rheology is connected to the ideas of disease transmission. If you take a Newtonian fluid, you could ask, what is that spray size distribution? And the amazing thing is it looks like an incredibly complicated process. It actually turns out there's a mathematical formulation that allows you to think about creation and destruction of droplets and leads to something called a gamma distribution. It's a one-parameter family of distributions that describes all of those droplet sizes incredibly well, if it's a Newtonian fluid. Okay. This was very clearly not a Newtonian fluid, as you can see, and you all know why now. You're extensional rheologists. You can see the thin threads that form where molecular elongation plays a role. And in fact, the surprise is that when you measure this property and you understand it, is that actually all polymeric materials, all mucus and all saliva, still obey a gamma distribution, but it's got a different parameter value set associated with it. And you get more big droplets and more small droplets. How can that be? Well, because it's a distribution, that means you get fewer average-sized droplets. The big droplets carry large amounts of disease. They're the ones that land on the neck of the person in front of you. The small droplets are the ones that are atomized and turned into aerosols. And you might like to know how many of those there are if you're going to decide whether 6 feet or 2 meters is the right distance for social distancing. So with that, I'll end with this. Rather than thanking all my students in person, I want to thank a small group of people particularly, because although I came here for sabbatical in '92, it's not my first trip to the Shine Dome. I came here in 1985 as a student at something called the National Summer Science School. That was a sister project of something called the London International Youth Science Forum that I went as a participant on myself as a student and then went back for many years to work at. And so just like for the class of us that we stood out there yesterday, I stood outside the Shine Dome in 1985, 39 and a half years ago. Really didn't know that I would ever be back, but this speaks to the power of international exchange. For all of the fellows here, many of us came from other countries originally, came to Australia. I want to particularly thank a guy called Rod Jury, who actually has a physics degree from ANU, who was instrumental in inviting me, and that was my first trip to the Shine Dome almost 40 years ago. Thank you all very much indeed.
Thank you, Gareth, for a wonderful talk and making us all honorary rheologists, even if I can see people edging towards the back of the room so that they have no one behind them. I'm sure you'll be happy to take some questions.
Absolutely.
Yeah. Lynn?
Thank you so much for an absolutely marvellous talk. What are rheologists going to do when we're on Mars or on the Moon? How is that going to affect how far sneezes go and all the other things that we'll be taking into space?
That's a fantastic question, in fact, because taking things into space is extremely expensive. Taking materials to Mars is extremely expensive. There's a large body of work, actually, at NASA at the moment and composites laboratories thinking about taking one extruder, for example, instead, and then 3D printing materials when you're actually there. So what's called Martian regolith. is basically silica that's never been eroded. It's razor sharp. It's extremely damaging to materials like spacesuits, but it can be compounded in with a plastic. You can put 40% Martian regolith in with plastic, double the volume of material you actually have to take there. So you will probably see that habitats will be 3D printed on Mars. Elaine?
Thank you. Fascinating talk. Are there different classes of chemicals that contribute to the non-Newtonian properties of these fluids? And do bacteria ever contribute to these, for example, in the sneeze case?
That's a wonderful question. We can nominally break it down into really 3 classes of materials. Polymers, and essentially all biological materials produce high molecular weight materials. Colloids, another example. Inks and pigments and things like that are usually colloidal in nature. And then really surfactants. And for example, the cytoplasm is really a complicated mixture of all 3 components. So you'll find that actually all 3 of these kinds of main classes of materials are definitely present in biological materials.
Francis? We have to let Francis
You have to ask at least one question of everyone, right? Yes.
I was wondering about medical applications. I wanted you to expand a little bit. Like, for example, people who have radiotherapy can no longer produce saliva.
Yes.
Or some of us lose material in our joints, Yeah.
Yeah, a wonderful question. Probably we'll have to talk more about it at the break, but it turns out that viscosupplementation is a great example. You may have had an injection of hyaluronic acid into your joint, for example. We've actually worked a lot on artificial joints as well. Polyethylene, high molecular weight polyethylene. We figured out how to infuse it with vitamin E, so actually your knee replacement will last much longer, for example. Dry eye, if anyone's had dry eye, we still struggle to make a synthetic tear that has anywhere near the properties of a natural tear film. It's what we call lubricious or slippery, and it's extremely difficult to produce a synthetic material that's anywhere like that. That's a great kind of topic for current research, actually.
Well, I apologise we have to leave it there, but we really thank you for an amazing talk and for your continuing connection with Australia. So please thank Gareth again.
Thank you.
I'd now like to invite Glenda back to the stage to introduce our next presenter.
Our final presentation from fellows elected in 2023 is Professor Pankaj. Pankaj has made seminal contributions to understanding the function and neural connections of the amygdala, a region in the brain that has a central role in processing emotions. His work has resulted in fundamental new insights into anxiety-related disorders, pathophysiology, and therapeutic targets. Please welcome Pankaj.
Well, thanks, Glenda, and look, I'd like to thank the Academy for this great honour, and my nominator, Perry Bartlett, who was the founding member of the Queensland Brain Institute and recruited me actually from Canberra up to Brisbane. You have to pardon my throat a bit, it's sort of moving around today. So learning, memory, and the amygdala. Learning and memory. Learning is the process of acquiring a skill or knowledge, and it leads to a change in behavior. And everyone here, of course, has learned things in their past. And memory, of course, is the ability to retain and recover information from a previous experience. And without learning and memory formation, I would suspect none of us would be here today. And certainly wouldn't be remembering things that we did 10 or 15 or 40 years ago. And so why is it you remember some things and not others? The time that you misbehaved at the age of 5 and your mum gave you a slap. I mean, people don't do that anymore, but, you might have had, many of us would have had a slap. Why is it you remember that so well in terms of all the other things that were going on at the same time? Well, that's your amygdala in action, assigning emotional valence to a certain event in your life. So learnings in memory leads to all your thoughts, feelings, and emotions. cognition and computation, and it makes you the person you are. And really the importance of this is really highlighted. If you meet somebody who's got Alzheimer's disease, one of the first things that goes is the ability to form memories. And of course, often the things you see is they stop being the people they used to be, and incredibly devastating disorder. So how is it that the brain learns and it stores information, and what is it we're doing? It really all started with these two guys, Golgi and Ramon y Cajal. Golgi was an Italian, and Ramon y Cajal was a Spanish anatomist. Golgi developed the technique of staining neurons, and Ramon y Cajal did an extensive study sectioning bits of brains of all sorts of animals. And they came to the idea that the brain was an organ like any other and contains cells which have connections between them. And they both got the Nobel Prize. In fact, they had a fight about what was actually going on in the brain during their Nobel lectures. So we now know for certain that the neurons, there's lots and lots of neurons in the brain. The human brain, there's about 10 to the 12 neurons. And they have connections between them. And on average, a cell has about 10,000 connections. So there's about 10 to the 16 synapses. These are what the connections in our brains are. This is the enormous number of connections. So why is this thing set up like this? So where do cellular theories for learning, and people started thinking about this a long time ago. In fact, Plato had theories about how we learned and did things. Cajal, in his lecture, Nobel lecture, actually he's made a suggestion. He was very prescient. Cajal was a very interesting character. If any of you are ever in Barcelona, you should go to the Cajal Museum. He was an amazing artist. It's the most fantastic drawings I've ever seen. So he has suggested, just wildly, that perhaps memory and learning formation happens through the changes in the connections in the brain. And Donald Hebb, who was a Canadian psychologist, wrote a book called The Organization of Behavior in 1949. This is a ginormous book, it's about 500 pages, and to this day there's only really one sentence that anybody ever quotes from this book. And what he suggested was, when an axon of cell A is near enough to excite cell B and repeatedly or persistently to its part in firing it, some growth process or metabolic change takes place, so that this changes, basically. And this is what's called the Hebbian synapse. Okay? And this is what underpins all our theories of learning and memory after all these years. So Hebbian synapses, so there's two cells, A and B. If you look at an event, it's a flower, and these two cells fire together. The idea of the connection between these cells represented here changes. So this is the dogma, the cells that fire together, wire together. And this is what drives the whole theories about learning. Now, this was just a theory. Nobody had any idea what kind of change might be taking place. And Tim Bliss and Terry Lomo discovered a phenomenon called long-term potentiation. They're working in the hippocampus, an area of the brain which we know is involved in learning and memory formation. Is the first areas which actually disintegrates during Alzheimer's disease as well. And what they found was, it doesn't really want they're plotting the strength of a connection. And what they showed was this connection is very stable normally, but if you repeatedly activate it, that's what those little arrows are up here, the strength of this connection changes dramatically, and this change is immediate and it's stable. And this is what they called long-term potentiation. So this is a rapidly acquired change which lasts pretty much, they looked over weeks in rabbits, and they found that works as long as you like. And this is essentially the principle of cells that fire together, wire together. So this is what the underpinning side of memory formation right now is. After all these years, we still believe that when you're learning something, what's happening is this phenomenon called long-term potentiation at some sets of connections in your brain. So how do you study learning and memory? So if I was to test one of you people in this room, and I was trying to understand how you learn something, it's pretty straightforward. I'll tell you a few facts. So let's meet tomorrow, we'll talk about it, and off you go, right? And then maybe I'll cut your brain out and have a look what happened, okay? Something that's not really feasible. So animal models are what you need. So how do you do this in animal? You can't actually ask them questions. Okay, very difficult. And because LTP was first discovered in the hippocampus, people started looking for things that involved the hippocampus. And there's lots and lots of different things. Now, what became immediately obvious, there's 3 phases to memory consolidation. There's the learning process, the time somebody teaches you something. There's a consolidation phase, which this thing improves and gets set in stone. And then there's retrieval, the time I ask you questions and you tell me, Oh yes, my mum's name was Barbara. Okay, this is your retrieval. Now, the hippocampus had a problem. The hippocampus is connected to many, many types of brain. It's involved in almost any process you can think of. And finding a behavior that works with specific connections to the hippocampus. So what are we trying to do here? We're trying to find out what are the connections that do this behavior, and when it changes, what are the changes that took place? That's essentially the object of the exercise. So, we hit upon this process called fear conditioning. And fear conditioning is a Pavlovian learning process, and you've all know about Pavlov. He was ringing a bell and feeding dogs food. And fear conditioning, what you do is you present animals with a neutral stimulus, a tone or a light, or sometimes a bit of food, and you pair it consistently with an unconditioned stimulus. This is generally an aversive stimulus, but it can be a reward as well. And when you do this, the animal learns that this unconditioned stimulus is going to predict reward, pretty much what Pavlov did, right? The bell predicted food. And typically what we do now is present a tone and give the animal a brief foot shock. This is like a little brief current to their feet, which doesn't feel nice. And what's plotted here is exactly what happens in fear conditioning. If you play this tone over and over again, you get a conditioned response, which is some response you can measure. Okay, during the acquisition phase, these two are paired. And the CS plus is a stimulus which is paired, and a CS minus is a second stimulus, which is different but is not paired. And you can see the conditioned response changes, it grows. And you come back sometime later at retrieval and you find it has a bigger impact, okay? So this can be day 3 or so this thing in between the line, that's our consolidation phase, okay? The amazing thing about fear conditioning is it turns out that it involves the amygdala. This is in blue you have the basolateral amygdala in the center. The amygdala is a very complex structure. It's in the mid-temporal pole in us down here. And the connections with the amygdala, barring small differences, are pretty much the same in any species you look at. And fear conditioning works with everyone. You could do it with you tomorrow, and it works like gangbusters, okay? It's very, very robust. And what was found was that if you lesion the amygdala, fear conditioning is abolished. So it's very clear that these circuits are what happens during fear conditioning. This is a very simple behavior. This is not like asking you your mum's name or, the answer to a differential equation. This is a very simple learned behavior, learning to associate one stimulus with another. And the circuits for this, turns out, are very similar in rats, dogs, cats, and humans. Okay, so if you use a tone, for instance, we know it goes via the thalamus and cortex into the basolateral amygdala. And the foot shock, which provides the aversive stimulus, comes via the thalamus and also goes to the amygdala. And we know these inputs converge onto single cells. So we know the circuits that are involved, and we know what the outputs are. This is the conditioned response, and what this is a behavioral outcome for you trying to learn to avoid that tone. You don't want to get a shock, okay? So when I got into the field, I was very interested in learning and memory formation and hit upon the amygdala as a place to work. And what we did for a number of years was try to understand the system of synapses that these connections make. And we understood that receptors, and within the BLA, we described the types of neurons in the BLA, the connections, and the nature of the inputs, and showed the inputs to interneurons were plastic. And now what we're doing is using this to produce a computational model for this learning. Okay, how is memory stored? So this is what during acquisition. Well, this is a set of connections that's normally available, and a memory is thought to be encoded in a pattern of connections. And this is called the engram. So this is when you remember something, this is thought that as a bunch of connections activate a bunch of very specific cells, and when you try to remember this, the whole engram gets activated. So what we're doing now is, it turns out, modern genetic techniques, you can label the engram. And we're trying to understand how do the engrams store information. And what you can do using genetic manipulations, and this is just one example from an experiment that was done literally 6 months ago. And this is measuring freezing in an animal. This is trying to recall this memory of this aversive event. And what we're doing, we're using light-activated ion channels to drive the cells of the engram. And this animal, for instance, when the light came on, it freezes. So this is what's called a phantom retrieval of the memory. The animal is not actually remembering that event, but we're making it remembered. Very interesting idea, and we're hoping this is what's going to lead to our answers about what kind of information is stored there. So just before I go on, let me show you. So this is what's been occupying my life for the last 30 years. Science and learning. I just mentioned this in passing because, learning, like we talked about learning, education, of course, is a form of learning. And skills, knowledge is transferred from a generation to another. It's what you do with your children and what happened to us when we go to university. We started the Science and Learning Research Center. This was a classroom, and we created these classrooms and we started making electrical measurements in kids who were being taught. And this has led to a whole new endeavor where we're trying to teach how neuroscience can affect learning, and the whole idea really is to improve the learning experience and to have an objective basis to education. And we can talk about this at lunch, and education is driven by pedagogy, and this is trying to get the scientists into the education business. So I'd like to thank all the students and postdoctoral fellows in my lab. That's the Queensland Brain Institute, as it now is. The funding from the NHMRC and the ARC, and of course in the current climate, philanthropic funding has been absolutely critical, particularly for the QBI. And thank you for your attention.
Thanks, Pankaj. I know everyone's probably has that Pavlovian taste of lunch already, but let's have a few questions. There's one here and one here. Oh, and one here, sorry, 3 questions. Okay.
Thank you so much, and congratulations for the wonderful work you're doing with school kids. It's now right across the whole of Queensland, you were telling me, and it's a wonderful model to extend further. Congratulations on that. I wanted to ask you about the role of sleep in all of this, because, people will say, well, let's sleep on it, and by the next morning you've remembered when you couldn't think what it was the night before. How does all that tie in, please?
Well, remember the consolidation phase. It's very clear. It needs about 12 hours. And it's pretty critical. Sleep really helps the consolidation phase. There's oscillations that happen both in the amygdala and the hippocampus that drive the consolidation phase. That's actually a complete change in the way that changes that take place, they become embedded in some genetic changes, and that's happening during sleep. If you don't sleep, you won't remember things nearly as well.
Really enjoyed the talk. What changes social media made? So when I went to university, we used to sit for an hour and listen to a lecture. Now it's just sort of flash, bits of disjointed information in much shorter time packets. What does that do to the learning process?
Well, if you think about the way social media operates, a lot of it is designed to have very strong emotional content to it. And that's why you tend to remember those things. Kids particularly tend to remember those things nearly so well. It's actually disrupting the learning process in many ways. So this Science Learning Center, among other things, we've created a framework for education. This is actually in university students, and you can download this from many sites. It's called Health, Higher Education Learning Framework, and that talks about how social media interacts with what's happening in the classroom at university level. So, we've taught a lot about how the two interact.
So the idea of a Hebbian response with, repetitive firing leading to long-term potentiation makes a lot of sense, but many of the events we remember are very acute, often life-threatening, lasts for maybe a millisecond or two. Doesn't seem a lot of time for repetitive firing. So how do you explain embedding of memory in that case?
Fear conditioning, it can be a one-stop process. You can fear condition them with a single presentation. And what these cells do is they discharge at high firing rates for brief periods of time. So despite the fact that the stimulus is very short, it sets up a pattern of activity which lasts long enough. It's a one-stop shop. Yeah, fear conditioning is very strong.
One last one. Sorry.
Wonderful talk. It's well known that the neurons in the cerebrum terminally differentiated soon after birth at age 2 to 5, carbon dating studies have shown. And you wouldn't want to have much turnover if you have memory, otherwise you'd lose your memory. Is the amygdala cells, can they still replicate later on in life?
Yeah, indeed, you do have newborn cells later on in life in the amygdala as well. Interestingly, the interneurons compared to in the hippocampus and cortex, where they're always excitatory cells. The amygdala is a very strongly inhibited structure, and it's very stable.
Okay, well, thank you very much, Pankaj. Great talk. And can I just ask that we actually thank all the 2023 fellows for all the fantastic talks that we've had. I'll now hand back to Anna-Marie and she'll close the session.
Thank you, Glenda and Julian, and thank you all for your attention and your thought-provoking questions, as well as to our audience online. We're going to break for lunch, but I do need you to take your things with you, because the next session after lunch in the theatre will be the admission ceremony for the 2024 fellows, and we need to set up the room for that ceremony. For fellows who are newly elected, if you haven't already had your portrait taken, and I know I've reminded you a few times, but we really want to photograph you, please make your way to the Becker Room today or tomorrow to have that done. Also, a quick reminder to our early and mid-career researchers, if you're wearing a purple lanyard, I am speaking to you. We have a competition that is running with our Gelato Cart partners. They don't only deliver gelato. Grant Ed Group, who have sponsored the gelato, have also enabled us to provide a prize, which is their Sprint Research Grant Development Program. It's a program that is of a value of $8,500, and it allows those people who are applying for grants, and they are ARC DECRAs, ARC Future Fellowships and NHMRC Investigator grants, with assistance in developing applications. So if you feel you could use assistance in developing a more competitive NHMRC or ARC Fellowship grant application, head over to the Gelato Cart and scan the QR code and enter the competition. It's very straightforward. We have one opportunity for someone, and you have to be in it to win it. Lunch is being served in the Jaeger room. The Dorothy Hill room contains meals for those people who have listed dietary requirements. Of course, make use of all of the venues and outdoor area, and we'll see you back here for the 2024 New Fellows admission ceremony at 1:30. Thank you.
For many animals, their sex is determined by their genetics. But researchers have found that the sex of some species at birth is determined by their environment.
They do it by temperature, and we've always wondered, how does that work?
Distinguished Professor Jenny Graves and collaborators at the University of Canberra have spent the last 2 decades studying this phenomenon in Australian dragon lizards. The lizard's sex is determined by sex chromosomes. When the eggs are incubated between 22 degrees Celsius and 32 degrees Celsius, the usual temperature range in the wild, ZZ eggs hatch as males and ZW eggs hatch as females. But when the temperatures rose above 34 degrees Celsius, the team noticed something strange.
Amazingly, what we found is at the higher temperature, the ZZ eggs, which should have turned into males, they turned into females. So they hatch as females, and amazingly, they're fertile.
Because these sex-reversed females can successfully mate with males, it has wiped out the female W chromosome.
So we've changed the whole sex-determining system. We've shifted it from genetic sex to environmental sex in 1 generation.
Temperature also drives sex determination in other reptiles such as alligators and sea turtles. It's a system that could have huge implications as the Earth's climate warms.
If global climate change keeps on getting hotter, we're going to have all-female lizards. We have all-female turtles and all-male alligators. Not going to work very well.
Scientists are now looking at whether different species could adapt to rising temperatures and avoid the disappearance of one sex.
Professor Pankaj has one of the toughest jobs in science. But he says no matter your job, everyone needs a hobby.
I've got notes on every bread I've made for the last 3 years. I like mucking around with the starter, mixing the dough, kneading. It's kind of fun, it's very relaxing. And then watching it rise, and then cutting it the first time and seeing what it tastes like, that's just great.
When he's not in the kitchen, Professor is studying something far more complex. As director of the Queensland Brain Institute at the University of Queensland, he leads some of the country's top neuroscientists on a mission to unlock the brain's many mysteries. One puzzle Professor Sar's own research has helped solve is around the amygdala. This region of the brain plays a key role in processing emotion, learning, and memory formation.
At the time we entered the field, there was virtually nothing known about it, as to what kind of cells there were, what their electrical properties were, what the local connections were like, and that's what we were initially doing. But then we moved on to thinking about connections to the amygdala with the rest of the brain, and then what it does behaviourally.
These days, Professor Sar's research group is interested in questions around how memories are formed at the cellular level and what happens once those memories are consolidated.
The world is coming into your brain, and then your amygdala, what it does is assigns what's called emotional valence to your sensory world, saying, oh, that was good, that was bad, that was insignificant, or whatever you like. And things which are significant to you, for whatever reason, are more likely to be remembered, basically.
That's why memories like your first kiss or a breakup tend to stick. It also helps to explain anxiety disorders such as PTSD, where amygdala dysfunction occurs because the memories have a detrimental impact on the person's quality of life. Professor Sarr says fundamental research into brain biology is crucial to developing better therapies for mental health conditions, now one of the leading causes of disability worldwide.
And a lot of these drugs that we have right now, they're not targeted drugs, all right? They just work across the board, and they do a lot of things along with treat the disorder that they're given. Things like Valium for anxiety, sure, they treat those disorders, they sort of help along that way, but they do lots of other things as well. And that's because we don't really understand why is it that you're getting a particular sort of symptom.
Those knowledge gaps were the reason he decided as a medical intern in the 80s to pursue neuroscience.
We used to walk around, as students and as interns, and trying to treat these people during the psychiatry term and then also the neurology term, really. And really, it was just hit and miss is what it felt like to me.
While scientists now know a lot more about the brain, Professor Sarr says there's still work to be done.
We're not at the level of understanding as we have, say, with the liver or the kidney or something like that. But we do have tools now which allow us to interrogate the brain in try to understand its complexity.
As a fellow of the Australian Academy of Science, he hopes to advocate for more funding into this fundamental research. In other words, the bread and butter of science.
The discoveries are going to be unbelievable. Things you're going to find in neuroscience you haven't even thought about today.
Cancer is a leading cause of death in Australia and disproportionately affects our Indigenous communities.
Aboriginal people are about 14% more likely to be diagnosed with cancer compared to non-Aboriginal people, and also about 20% less likely to survive for 5 years after diagnosis. So there's a massive gap there.
It's a gap that Dr. Justine Clark is on a mission to tackle as an Indigenous genomics researcher and recipient of the 2024 Aboriginal and Torres Strait Islander Award from the Australian Academy of Science. Indigenous Australians still face many barriers in accessing standard and precision cancer care, but Dr. Clark hopes her research will help change that. At Telethon Kids Institute in Adelaide, she's begun work to build up a knowledge base of Indigenous Australian genomics to drive advancements in clinical care for Indigenous Australians living with cancer.
The exciting thing about it is that we can learn about our DNA and find new treatments for cancer that are targeted to specific genes. And we can also find better uses for our current treatments, better diagnostics, better screening, and that's what we call precision medicine.
Dr. Clark plans to work with members of the South Australian Aboriginal community to better understand the cultural implications of this research and ensure it will ultimately benefit Indigenous Australians. She will also travel to New Zealand to meet with Maori colleagues with hopes of establishing collaborations there.
Doing it together is the best way forward to create a global agenda where we can improve cancer outcomes for every Indigenous person.
It's work that Dr. Clark feels incredibly proud to be a part of.
I always think of my ancestors, and they were the first scientists in this country. And I think of the legacy of people that have come before me that have fought for a better future for our communities. And I'm just honoured that I get to do that now through my research.
This is no ordinary scientific lab. These field sites in the Riverina, New South Wales city of Wagga Wagga help Professor Leslie Weston improve our fundamental understanding of crop protection.
My entire career has been developed around looking at mechanisms, how things work, why things are working, and commercially, what does that mean for a nursery industry or horticulture, or in this case, broadacre agriculture or livestock producers.
Professor Weston is a plant biologist. She studies how plants interact with everything around them.
So specifically, plant interactions with pathogens, plant interactions with soil inoculants, plant interactions with weeds, and plant interactions with insects, as well as biocontrol agents.
Charles Sturt University is the perfect research base for Professor Weston's team. The campus is home to not only a working farm, but also state-of-the-art labs.
We can now assess, using mass spectrometry and metabolomics, the presence of the whole complement of chemistry that's present at any point in time, either in a plant or in the recipient organism, the animal or the pest.
These techniques have offered new insights into how plant roots release various molecules into surrounding soil and the resulting impact. This knowledge is then shared with industry and other scientists to create what she calls big picture solutions for farming.
Together we work on a systems-based approach to understanding how competitive crops work or how we can protect a plant or livestock from interactions that are detrimental.
It's work that comes naturally to Professor Weston. As a child, she spent a lot of time in glasshouses, helping out at her family's commercial nursery and garden centre.
My family's enterprise in terms of the nursery, garden centre, and production has probably always made me recognize that the industry is really what drives a lot of these discoveries too.
She sees her election as a Fellow of the Australian Academy of Science as a recognition of the work from her entire research team.
I'm also pleased that we're from regional Australia. We're hoping that this will bring an awareness of the types of research and the impacts that we can have on the community. So, very pleased that this happened.
Billabongs are bodies of water left behind when a river alters course or after floodwaters recede. These are some of the last remaining in Melbourne and were once an important gathering place for the local Wurundjeri people and neighbouring tribes.
Slow, I've got to get through.
When Dr. Joe Greet first started working here alongside the Wurundjeri Narrap team 5 years ago, things were looking grim.
And most of the sites were filled with weeds, there were few frogs, no water birds.
That's because the billabongs weren't getting the water they need to remain healthy.
The water coming down the Birrarung is only 2/3 of what it was pre-colonisation, so the Birrarung doesn't connect with its landscape, with its billabongs as frequently.
A collaborative effort involving the Narrap team and Melbourne Water has begun turning things around.
We are starting to introduce solutions, hopefully to increase the resilience of these sites.
Solutions such as creating a drainage swale to enable water to flow more freely between the river and Billabong. Dr. Greet and Narrap Rangers are leading a monitoring program to better understand how native plants and animals respond both to these managed flooding events as well as natural flooding from the Birrarung.
We're seeing a drowning out or suppression of the terrestrial weeds, a flourishing of the native wetland vegetation, and the increased condition of these beautiful old river red gums. Just previously, the water birds seem to know when these billabongs are full. We've seen eel numbers increase.
Who's getting the gloves on?
And we see through our eDNA sampling that those sites that are more frequently engaged with the river have a greater representation of native fish and frogs.
But Dr. Greet says there's more to be done, not only to improve the health of our billabongs, but also to increase the role of traditional owners in water and land management. As a recipient of the 2024 Aboriginal and Torres Strait Islander Scientist Award from the Australian Academy of Science, he hopes to continue efforts to do just that.
We want to get together and hold a series of workshops and understand better from a Wurundjeri perspective how we can better assess the health of these billabongs, the health of country, and better manage them into the future.
Indigenous-led research is also crucial in the field of cancer genomics, where fellow award recipient Dr. Justine Clark works. In Australia, cancer disproportionately affects Indigenous communities.
Aboriginal people are about 14% more likely to be diagnosed with cancer compared to non-Aboriginal people, and also about 20% less likely to survive for 5 years after diagnosis. So there's a massive gap there.
Indigenous Australians still face many barriers in accessing standard and precision cancer care, but Dr. Clark hopes her research will help change that. At Telethon Kids Institute in Adelaide, she's begun work to build up a knowledge base of Indigenous Australian genomics to drive advancements in clinical care for Indigenous Australians living with cancer.
The exciting thing about it is that we can learn about our DNA and find new treatments for cancer that are targeted to specific genes, and we can also find better uses for our current treatments, better diagnostics, better screening, and that's what we call precision medicine.
Dr. Clark plans to work with members of the South Australian Aboriginal community to better understand the cultural implications of this research and ensure it will ultimately benefit Indigenous Australians. She will also travel to New Zealand to meet with Maori colleagues with hopes of establishing collaborations there.
Doing it together is the best way forward to create a global agenda where we can improve cancer outcomes for every Indigenous person.
It's work that Dr. Clark feels incredibly proud to be a part of.
I always think of my ancestors, and they were the first scientists in this country. And I think of the legacy of people that have come before me that have fought for a better future for our communities. And I'm just honoured that I get to do that now through my research.
Shrine Dome is an iconic heritage-listed building in Canberra. It's also a symbol of scientific achievement and endeavour in Australia.
It's the place where Australia's most distinguished scientists, the Fellows of the Australian Academy of Science, come together to deliberate, to share their knowledge. It's where collaborations are born, and it's iconic in Australia.
The hailstorm in Canberra in 2020 has made a huge impact on the Shrine Dome in terms of damaging the copper tiles and also leading to damage to some of the key science documents of various scientists. We are very pleased the roof has now been repaired, and this event reminded us of the importance of the Shrine Dome as a symbol of scientific excellence in Australia.
We used that opportunity to create a campaign called the Celebrate Science campaign, and to cloak the Shrine Dome metaphorically in great Australian scientists, and also to recognise teachers of scientists.
Any individual can dedicate a particular tile to a scientist of their choice, including science teachers. Without their help in my childhood, I would not be here today. So I've dedicated tiles to my teachers as well.
Donating to the Celebrate Science campaign is a unique opportunity. There are only a limited number of tiles, 1,888 of them, and when you make a donation, it exists in perpetuity. Funds
provided by the donors provides the independence for the Academy to be able to do work which is important for the nation.
A key mission of the Australian Academy of Science is to ensure science is heard wherever decisions are made. That's in our parliament, in our courtrooms, in our boardrooms, in our classrooms, and in the public square.
We want to say thank you. The donation which you are making is going to make a huge impact. We are touched by your kindness and generosity. You are making the big difference to Australian science.
People think that reconciliation is about Aboriginal and Torres Strait Islander people reconciling with the rest of the population, but it's actually not. Reconciliation is about how non-Indigenous Australians recognise and celebrate that Aboriginal and Torres Strait Islander people were here 65,000 years ago. We've had a continual relationship with the country. We are the longest continuing cultures in the world. How do we reconcile in the last 230 years? We've had a dramatic impact since colonisation on Aboriginal and Torres Strait Islander people. There are people that say, oh, you know, why should Aboriginal and Torres Strait Islander people have a say in these matters? And I just point out that whenever we have a natural disaster, you know, be it a flood or a fire, the government might think that they can go in and make changes and redevelop those communities, but they cannot do it without the engagement of those communities. So, you know, if we can see that as being good practice for the rest of the population, we have to see it as good practice for Aboriginal and Torres Strait Islander people. Why should we differentiate?
The war in Ukraine has had a devastating impact on the country's people.
We have 9 million displaced people within the country. We have 5 million displaced who are outside of the country.
That includes many scientists working on everything from medical research to the future of space exploration.
So the impact is huge. Imagine you out there, you know, working in your lab, and all of a sudden you have to give up everything and move out.
Thanks to an $800,000 donation from the not-for-profit Breakthrough Prize Foundation, the Australian Academy of Science is now extending a helping hand to Ukrainian scientists.
This opportunity allows Ukrainians who are impacted to collaborate with Australia so that their research effort can continue.
The initiative will sponsor visits to Australia by Ukrainian researchers and make it easy to send data and samples to Australian research facilities with the results returned to Ukraine.
That is really important so that the research capability that's required to help rebuild their nation in the fullness of time is there and is not depleted over the course of the war.
to rebuild Ukraine will need support of the world. And I think these connections which will be built as a result of the program will help us better help Ukraine in the future.
For more information, head to science.org.au/Ukraine research. Ticks don't have the greatest reputation. They are, after all, parasites that pose a danger to people and animals. But the very thing we despise them for is also what makes them useful to Professor Richard Payne.
What ticks do amazingly well is feed on blood for long periods of time.
That's because their saliva has powerful blood-thinning molecules, molecules that could help treat conditions such as strokes. That's where Professor Payne comes in. He's a chemical biologist who uncovers the functions of proteins and peptides made by plants and animals and then improves them for our use.
Nature has been evolving their molecular arsenal probably over billions of years. So they've had a lot of time to, hone the bioactivity and the selectivity of these molecules. And so that's a great starting point because we don't have to do all that legwork. We can take that and then use some of the chemical tools that we now have to sort of put on aftermarket modifications to make those molecules even better than what nature makes in the first place.
In the case of ticks, it's not just their blood-thinning prowess that interests Professor Payne. His team has also been able to synthetically produce the anti-inflammatory proteins found in their saliva.
So a tick can be sitting on your back and you won't necessarily know it's there. And the reason is it's producing really potent immune modulatory molecules that can suppress your immune system to stop you from basically itching and scratching the tick off.
So, can those molecules enhance treatments for symptoms caused by lung inflammation in respiratory illnesses like COVID-19? That's an example of the questions being asked within the University of Sydney node of the ARC Centre of Excellence for Innovations in Peptide and Protein Science, where Professor Payne is Deputy Director. The team studies everything from venomous species to native plants and Australian marsupials.
And we're particularly interested in molecules that are produced in the pouch of these animals that can have either anti-infective activity, so antibacterial activity to prevent the young from getting sick, but also immune modulatory activity.
Professor Payne's election as a Fellow of the Australian Academy of Science is recognition of his pioneering work in developing powerful technologies to modify peptides and proteins for applications in biology and medicine. He says he's particularly proud of his role in mentoring the Fellows of tomorrow.
Mentoring is actually my favourite part of the job. I now have a large number of alumni that are working both in industry, in the pharmaceutical industry, but also running their own labs in universities all around the world. And I think that's just massively rewarding.
I think it's the idea that you can't be what you can't see. So if we don't have women, people of colour, all sorts of diverse representation in science, the next generation is going to look at that and say, well, it's not for me.
My first position as a bioinformatician was in the computer science department, and you could really count the number of females that were in the building, let alone in leadership roles. When you have people who you can relate to, it certainly allows you to at least imagine that that could be possible for you. So I think that representation, that diversity does matter when you're trying to inspire the next generation.
There's been studies that show people with diverse experience bring new ideas, a fresh perspective, and just think of all the scientific breakthroughs that we will miss out on if people don't see this as a career where they are valued and their experiences are valued.
From mobile phones to electric cars, rechargeable batteries can now power just about all things electronic. But they're far from perfect.
Battery performance worsens over time due to a combination of factors, including the battery material degradation, electrode degradation, electrolyte breakdown, and also environment issue as well.
Here at the University of Adelaide, Professor Xinghuo is focused on developing the next generation of rechargeable batteries.
So if the battery has perfect performance, let's say high energy density, can be fast charged with long lifespan, very safe, and also very cost effective, then we are happy. That's our target, to develop rechargeable battery with excellent performance.
Her research group investigates novel electrode materials and electrolytes for lithium-ion batteries to increase the amount of energy they can store and extend their cycle life. They're also exploring more affordable and sustainable alternatives to the widely used lithium-ion battery. This includes potassium-ion batteries, which Professor Guo herself pioneered.
Potassium is in the same group in the periodic element table. So in terms of chemical properties, quite similar to lithium. Potassium is more abundant, it's cheaper. That's the key thing.
While these battery systems are still in very early stages of development, Professor Guo believes they have great real-world potential. In fact, some of her innovations have already attracted investors. For instance, her team successfully synthesized silicon-carbon composites to use as anode materials instead of graphite in lithium-ion batteries. And they're now collaborating with a startup company to scale up this technology.
Graphite has a relatively low capacity. Therefore, the energy density of current lithium battery is limited, normally below 250 watt-hour per kilogram. But with this silicon carbon composite to replace the current graphite anode, we can boost the energy density.
Professor Guo came to Australia more than 20 years ago to do her PhD in materials engineering at the University of Wollongong. She says being elected a Fellow of the Australian Academy of Science is a great honour and hopes it can inspire other young women in science to follow in her footsteps.
Being a woman in science is quite challenging but rewarding as well. Work-life balance is not an easy thing to manage, but I'm proud today. I'm able to mentor the next generation, and I have very fantastic female scientists working in my team. I'm very proud of this.
The average Australian spends at least 90% of their time indoors. But how often do you think about the air you're breathing in? Indoor air pollution is considered a serious risk to public health in societies around the world. Despite that, most countries don't have any indoor air quality standards.
Every drop of water we drink from the tap, its quality is highly regulated. Every piece of food we take to our mouth is highly regulated in quality. Indoor air, which we take into our lungs 12 times a minute, is not regulated at all.
Professor Lydia Moravska from Queensland University of Technology was named one of Time magazine's 100 most influential people in 2021 for pressuring the World Health Organization into acknowledging that COVID-19 could be spread through the air, particularly indoors. Now she's leading a new push for international action. Professor Moravska and a group of global experts have used the prestigious journal Science to publish a blueprint for mandating indoor air quality in public buildings.
Our paper, which is a consensus of over 40 scientists and experts from many disciplines from around the world, is the best practice, better than anything else which is happening anywhere in the world.
The group proposes standards for 3 key indoor pollutants: carbon dioxide, carbon monoxide, and PM2.5, which are particles so small they can lodge deep in the lungs and enter the bloodstream.
This is my favorite little monitor. It measures carbon dioxide. Together with carbon dioxide, we emit other things, in particular pathogens, bacteria or viruses if we are infected. So if concentration of carbon dioxide increases, potentially concentration of these pathogens increases as well. And it fits into my handbag, so it goes with me everywhere.
The scientists also want to ensure mechanical ventilation systems remove and dilute pollutants at a higher rate than their production. She points to schools as an obvious place for action, where many classrooms are now being fitted with air conditioning.
One issue with this is that while it provides a good thermal comfort, it does not provide fresh air. It just recirculates the air, so it doesn't do anything to improve air quality.
Outdoor air quality is already regulated in many places in line with the World Health Organization's global air guidelines. Professor Moravska says countries such as Australia can now lead by example for indoor air as well.
We know how to do it. We know how important it is. Why won't we start now?
Distinguished Professor Brajesh Singh says there's a very simple reason why we should all care about soil.
All terrestrial life is dependent upon the soils to sustain it.
A single teaspoon of topsoil can contain up to 6 billion microorganisms. That makes soil one of the most biologically diverse materials on the planet and one that is critical for plants, including most of the world's food crops.
They are entirely dependent on the soil microbes to provide them the nitrogen, the potassiums, the phosphorus and other chemicals the other elements that it needs to grow.
Professor Singh is a world-leading soil ecologist. His work has helped shed light on the highly complex nature of the ecosystem beneath our feet and how that ecosystem is adapting to climate change.
All the data we have today suggests that soil biodiversity is vulnerable to the climate change. If you change the soil biodiversity, if you reduce the soil biodiversity, the service and the functions they provide will be reduced.
Professor Singh helped edit the Global Soil Biodiversity Atlas that brought together the findings of more than 150 researchers across 26 countries. It was a major piece of work, but scientists think they've still only isolated around 1% of soil microorganism species. Professor Singh believes countries, including Australia, should keep track of soil biodiversity as part of their global reporting obligations.
You cannot deliver a solution for a global challenge without high-quality science.
His own high-quality science is helping farmers to implement sustainable approaches and minimise the impacts of climate change. That includes the development of patented technologies to detect a soil problem that becomes more common as the weather warms, food and environmental pathogens.
We need to understand where they are distributed so that our own human health is managed.
Professor Singh believes all soil ecosystems are now facing similar threats.
In the natural ecosystems, you're looking at how naturally planted microbes interact. But the pressures, now we're putting all sort of pressure there as well. We're putting climate change, we're putting pollutions. Not many parts in the world where there is no chemical pollutions.
He says becoming a Fellow of the Australian Academy of Science is an unexpected honour and one he hopes will help make a genuine difference.
We need to come together, not just as a researcher or the scientist, but as a society, to tackle these issues more vigorously than we are doing at this moment. Because unpredictability of its consequences means we can't plan. And if we can't plan, the repercussions can be more severe.
Should a good bottle of wine be sealed with a cork or a cap? There was a time when the high-end wine world scoffed at the mere mention of screw caps.
It might seem a simple thing, but that required an enormous amount of research.
It was in fact industry leaders in Australia who led the push, eager to increase consumer choice. Research has shown that screw caps offer a better seal, meaning oxygen can't sneak into the bottle. The result is a crisper and more well-preserved wine, suited to most varieties of white. Some winemakers insist that fuller reds benefit from a little oxygen in the bottle and therefore age better with corks. But screw caps have another benefit. They prevent the wine from ever being corked, a term used to describe the presence of undesirable chemical compounds almost always transferred from poor cork batches. Corked wine has a characteristic odour that some compare to a wet dog.
We understand so much about the underlying science in Australia that we virtually get it right every time.
Professor Peter Hoy is Vice-Chancellor of the University of Adelaide, but he was also Australia's first Professor of Viticultural Science and then Enology. In other words, an expert in the science of grapes and wine.
This is where science always comes out on top. It enables things because people trust it.
Australia is the world's biggest exporter of iron ore. China is our biggest customer.
Roughly 50% of iron the world consumes is produced in China, and the iron industry contributes 7% of the global carbon dioxide or greenhouse gas emission.
That's because the process of converting iron ore into iron and steel requires a massive amount of heat.
And that takes a lot of energy, and usually coal is burnt to provide that energy.
So the challenge is to replace coal with something much more friendly to the environment.
If it was easy, it would have been done.
Professor Eric May leads the Future Energy Exports Cooperative Research Centre, or CRC. The centre is tasked with working out how to make Australia's energy exports greener and more sustainable.
The problem we're trying to solve is not a national problem, it's a global problem.
And that's why collaboration with China is critical. So Professor May turned to his longtime colleague from the University of Western Australia, Professor Dongke Zhang. Professor Zhang moved from China to Australia in 1988 to complete his PhD in the coal capital of Australia and steel-making city of Newcastle. Now listed as one of the most influential engineers in Australia, Professor Zhang has maintained close ties with industry and researchers in China. He convinced one of China's oldest steel producers and the major consumer of Western Australia's premium iron ore to take part in the Australian research.
Their specific interest in participating in the CRC is our work in using hydrogen and ammonia to reduce iron ore.
Both hydrogen and ammonia can be used as carbon-free reductants and fuels produced from renewable sources. The CRC is exploring the best way to produce, store and transport these fuels. Professor Zhang has experimented with renewable fuels in the lab, but the CRC will enable his ideas to be tested systematically and at a much larger scale. If all goes to plan, it will create a major new export for Australia while helping to reduce emissions overseas.
People in this field can make a real difference and help secure humanity's longer-term future, because we're going to need to have cheap, sustainable, clean energy indefinitely. How we get to that point is the journey we're on now.
Scientia Professor Li Meng Dai had never thought about moving to Australia. But while learning English in his hometown in China, his Australian teacher asked him to fill out a PhD scholarship application form for the Australian National University, purely as a language learning exercise.
To my pleasant surprise, the teacher sent the form that I fill in to ANU, and he also acted as my referee.
The application was accepted, and Professor Dai was soon living in a city he'd never heard of, undertaking a PhD in chemistry. In the more than 30 years since, he has risen to become a global authority on using carbon-based materials in renewable energy and clean chemical production.
So we need to develop cheaper and as abundant materials as a catalyst for decarbonization of the world.
Professor Dai and his team developed carbon-based metal-free electrocatalysts. As the name suggests, they are catalysts that can be made from carbon dioxide, biomass and other carbon-rich wastes without any precious metals.
Precious metals, including, say, platinum, palladium, uranium, and gold, are typically good catalysts, but they are expensive and also unsustainable.
Carbon nanomaterials have similar properties to that of precious metals, such as high strength and good electrical conductivity, but they're also abundant and much more environmentally sustainable. Professor Dai believes that makes them ideal for fuel cells, advanced batteries and water splitting systems. His team has found carbon catalysts to be 3 times more efficient than platinum in the lab for alkaline fuel cells.
It should be at the stage for industry support to commercialize the carbon catalyst for clean energy production and also clean chemical production.
That will require industry partners to undertake broader scale pilots. Helping to meet zero-emission targets through revolutionary carbon science is the central focus of the ARC Centre for Excellence for Carbon Science and Innovation, which Professor Dai is leading here at UNSW Sydney.
We should be able to commercialize some of the carbon catalysts for practical applications. We are leading the field.
Now proudly a Fellow of the Australian Academy of Science, he says he never regretted making the bold and unexpected move to Canberra. And although his career has since taken him around the world, Professor Dai and his family are very proud to call Australia home.
We love this country and we love people here, and also, of course, life quality and particularly the climate.
You're watching time-lapse footage of dung beetles at work. Their remarkable ability to remove dung makes them the perfect biocontrol agents. Research has shown their presence significantly reduces the number of flies and other pests, while also improving pasture growth and soil retention. So scientists like Professor Leslie Weston have been searching the world for the best species to bring home. But wait, doesn't Australia already have dung beetles?
We have 500 native dung beetles, but those dung beetles are geared to sourcing marsupial dung or drier, small, hard pelleted dung, whereas when cattle, sheep, horses and other livestock were introduced to Australia, they generate a larger, wet, moist paddy.
CSIRO began importing dung beetles to Australia in the 1960s. The program has been overwhelmingly successful. However, the introduced species haven't adapted to all regions of Australia, and some will simply stop working during certain periods of the year. Researchers are working to address these gaps with a joint venture called the Dung Beetles Ecosystems Engineers Project, supported by Meat and Livestock Australia, with federal funding from the Department of Agriculture, Fisheries and Forestry.
So we'd like to have a full complement of dung beetles that are active all year round, because obviously cattle or sheep are producing dung all year round that needs to be processed.
3 new species have been introduced from the Mediterranean region, but they don't all deal with dung in identical ways.
We have rollers that create these rolled balls and bring them below. We have the tunnelers that bring the dung down and create the balls. And then we have those that remain on the surface and dwell above. The dung beetles actually subsist by sucking out the moisture on the dung. It's like a liquid milkshake. That's their nutritional source.
Researchers at Charles Sturt University in Wagga Wagga are helping to rear the new arrivals after their strict quarantine regime is over. The offspring will then be distributed to field sites across southern Australia.
Professor Shizhang Qiao is firmly fixed on our future energy needs. Here at the University of Adelaide, he's designing new technologies to help power our world in a more sustainable way. This includes novel catalysts for producing green hydrogen.
In traditional, the electrocatalytic process, they use fresh water produce green hydrogen. But in Australia and some countries, fresh water is a shortage. If we develop a new catalyst, we can use seawater directly.
Before seawater can be used for hydrogen production, it typically needs to be desalinised and purified. But his team found a way to bypass that expensive process. They designed catalysts to suppress the formation of chloride byproducts. These chlorides can corrode the electrolyser quickly during direct seawater splitting. Their catalysts also prevent precipitates from forming at the electrode and impacting the electrolyser's lifespan. The findings, published in the prestigious Nature Energy Journal, have caught the eye of many prospective investors.
We talked with several companies already, so I think in next 2 or 3 years we can commercialize our technology and use technology in a real application.
His lab is also exploring aqueous metal batteries as a cheaper and safer alternative to lithium-ion batteries for the storage of electricity in households and power grids.
For lithium battery, a lot of problem. First problem is cost. It's very expensive. Second one, is easily cross fire. That's the reason. So we need to develop new battery technology.
Professor Qiao is a leading expert on energy storage and conversion. He says that's a result of a genuine passion for learning and discovery.
Even now, if we find a new research phenomena or a new research results, I'm very, very exciting and very happy.
As a fellow of the Australian Academy of Science, he's eager to continue doing fundamental research to tackle the world's energy problems and hopes to inspire young researchers to do the same. Sydney's Royal National Park is home away from home for Professor David Keith.
I've spent a lot of time in this park doing research to better understand how biodiversity works and how to help it persist. There are 1,100 plant species that occur in Royal National Park, so it's an absolutely massive convergence of species represented there by plants in that example, but the plants also have an enormous web of dependent invertebrates that live with them.
These communities of species are at the heart of Professor Keith's work. He's considered one of Australia's foremost experts in plant ecology and vegetation dynamics. It's work that is fuelled by a genuine love for nature.
We're on a mission from God, and that mission is to conserve, nature, to give something back for the way that it supports us people. Public good is a really important contribution for scientists to make.
But conserving nature is becoming increasingly difficult. He lists many threats, including humans' excessive use of land and water resources, a growing number of invasive species, and the rising frequency and severity of natural disturbances such as floods and fire. The impacts are obvious, even here in a protected national park, where some long-lived plants are undergoing sustained declines associated with changing fire regimes and novel diseases.
We've been able to show that there's been quite substantial declines over the last few decades in species that normally live for hundreds of years. About a third of plants that we tagged 30 years ago are no longer with us, and they're not being replaced at anything like the rate that they need to be by new seedlings to maintain a stable population.
But Professor Keith's research extends far beyond Sydney. He led an international team classifying the world's ecosystems into 110 functional types. He was also the driving force behind the so-called Red List of Ecosystems, a global scientific tool to assess the risk of ecosystem degradation and collapse.
There's no species that can exist without an ecosystem, a place to live in nature. What we're seeing, in the last few hundred years in particular, is a simplification of ecosystems where we're losing species that contribute important functions that keep other species afloat, because there are an incredible array of dependencies amongst different species.
But despite the mounting threats, Professor Keith remains hopeful. As a fellow of the Australian Academy of Science, he wants to share his knowledge to ignite social change.
Ultimately, it's about what people value and what they want to do. If they value nature, then they will look after it. I'm utterly convinced about that. So sharing, what we learn about this might actually help a few people appreciate, nature a little more because of, its incredible diversity and intricacy and complexity.
I think it's the idea that you can't be what you can't see. So if we don't have women, people of colour, all sorts of diverse representation in science, the next generation is going to look at that and say, well, it's not for me.
My first position as a bioinformatician was in the computer science department, and you could really count the number of females that were in the building, let alone in leadership roles. When you have people who you can relate to, it certainly allows you to at least imagine that could be possible for you. So I think that representation, that diversity does matter when you're trying to inspire the next generation.
There's been studies that show people with diverse experience bring new ideas, a fresh perspective, and just think of all the scientific breakthroughs that we will miss out on if people don't see this as a career where they are valued and their experiences are valued.
The average Australian spends at least 90% of their time indoors. But how often do you think about the air you're breathing in? Indoor air pollution is considered a serious risk to public health in societies around the world. Despite that, most countries don't have any indoor air quality standards.
Every drop of water we drink from the tap, its quality is highly regulated. Every piece of food we take to our mouth is highly regulated in quality. Indoor air, which we take into our lungs 12 times a minute, is not regulated at all.
Professor Lydia Moravska from Queensland University of Technology was named one of Time magazine's 100 most influential people in 2021 for pressuring the World Health Organization into acknowledging that COVID-19 could be spread through the air, particularly indoors. Now she's leading a new push for international action. Professor Moravska and a group of global experts have used the prestigious journal Science to publish a blueprint for mandating indoor air quality in public buildings.
Our paper, which is a consensus of over 40 scientists and experts from many disciplines from around the world, is the best practice, better than anything else which is happening anywhere in the world.
The group proposes standards for 3 key indoor pollutants: carbon dioxide, carbon monoxide, and PM2.5, which are particles so small they can lodge deep in the lungs and enter the bloodstream.
This is my favorite little monitor. It measures carbon dioxide. Together with carbon dioxide, we emit other things, in particular pathogens, bacteria or viruses if we are infected. So if concentration of carbon dioxide increases, potentially concentration of these pathogens increases as well. And it fits into my handbag, so it goes with me everywhere.
The scientists also want to ensure mechanical ventilation systems remove and dilute pollutants at a higher rate than their production. She points to schools as an obvious place for action, where many classrooms are now being fitted with air conditioning.
One issue with this is that while it provides a good thermal comfort, it does not provide fresh air. It just recirculates the air, so it doesn't do anything to improve air quality.
Outdoor air quality.
You're going to be standing for a long time. I don't know how you do it.
Thank you. So we are waiting for Anna Maria to join us.
Yeah.
Maybe she might have thought that I will start it, but of course I can start that. Yeah. You're all set with the names?
Yep.
I think Ravi is the only one. Ravi? How are you?
heavily culture.
I realised when I was doing those stage notes that you would know how to say it.
Well,
but then I figured that the person who's introducing
Jagadish, you want us to start for you and introduce you instead of Anna Maria?
No, that's fine, but let her come because she's just coming.
Okay. Talking to Kate, I wonder whether it's not a good idea if Jagadish could say something about philanthropy and if the new fellows can help with that.
So it's in his speaking notes. It's not put as you've just put it, but it talks about them.
Yeah. We've got to use every opportunity we can. What are we still waiting for? There's no... Okay, Francis is not there. And Lynn's not there. She's so enthusiastic. She's supposed to be out. She's not supposed to be.
Lynn's sitting up there.
Yeah, she should be at the, she should be down there.
It's at the back. She's gone and sat at the back.
And Francis
Got it. That'd be right. That'd be right.
And where's Francis? She's not there.
Francis is eating ice cream.
Oh, my God.
Francis is with Linda.
Yeah.
Okay. Good afternoon, everybody. I know most people in this room, but as we are about to introduce the 2024 Fellows, some people have just arrived, so you may not know everybody here. My name is Anna Maria Arabia. I'm the Chief Executive here at the Australian Academy of Science, and it's my great pleasure to welcome you all here to the Shine Dome for this very important occasion, and that's the admission of our 2024 Fellows. Before I hand over to our President, I'd like to ask you to place all of your phones and mobile devices on silent, if you haven't already done so. If you'd like to share this wonderful occasion with the world, in addition to the world that's watching through our online streaming, you may do so using the hashtag ShineDome24. I'd also like to thank our online audience who have joined us. We've had a strong response from some people who aren't able to be here but are very keen to follow what's happening and to learn about your science. I have reminded people over the course of the day on many occasions, but again, there are some people who have just arrived. We would like to take photographs, portraits of each of our new fellows. Those photos are being taken in the Becker Room, just outside of this theatre, in a circular building. Just keep walking and you will find the Becker Room. Please make your way there at some stage to have that captured. There will also be a group photo of the 2024 Fellows at the end of the ceremony. To commence this wonderful ceremony, may I now please invite to the podium the 20th President of the Australian Academy of Science, Professor Jagadish. Thank you.
Thank you, Anna Maria. I also would like to pay my respects to the traditional custodians of the lands on which we meet, Ngunnawal people, I pay my respects to their elders, past, present, and emerging. Colleagues, it's an honor to welcome you to the Australian Academy of Science this afternoon and to celebrating scientific excellence with you here in the home of science, the Shrine Dome. Adding to the significance of this occasion, this year we are celebrating the Academy's 70th anniversary. The Academy is proud to bring science to the service of the nation and to have made substantial contributions to shaping Australian and global science over a period of 70 years. Our country has a deep reservoir of talent within the sciences, including some of the world's most eminent researchers and professionals. 614 of which are currently elected to the Australian Academy of Science for their outstanding contributions to science. Each fellow is nominated by their peers and chosen through a rigorous election process. You are no exception, and today you join our fellowship who are collectively the brains trust of the nation, a true national treasure. 2024 fellows, I personally and warmly welcome you and your families and guests. This afternoon's proceedings are to celebrate you and your achievements. We are delighted you are here with us to formally be admitted to the Academy. The admission of new fellows is a time-honored tradition established by the founding fellows of the Academy and the fellows of the Royal Society of London before them. Upon admission to the Academy and by signing the Academy's charter book, fellows subscribe to the following obligation. I hereby solemnly promise to promote, declare, disseminate scientific knowledge, and to serve the Academy according to its charter and bylaws. The charter book was a gift from the Royal Society and has been signed by every fellow of the Academy. Your signatures today will sit alongside those of some of the world's most esteemed scientists, and you are welcome to look through, which is a significant piece of Australian history at the conclusion of the ceremony to see how deep the traditions and knowledge of this learned academy run. To officiate the formal admissions, I will be assisted by the Academy's Vice President, Professor Bob Graham, and immediate past Vice President, Professor Malcolm Sambridge. Bob and Malcolm will announce the names of each new fellow and read your citation. At the conclusion of the citation, I invite each new fellow to come to the stage to sign the charter book and receive your certificate, and lapel pins are already been given to you. A photo will be taken on stage before we enjoy a video summary of your achievements. Fellows will be called in alphabetical order to come forward and join me at the charter book. Before I start the process, I also would like to introduce the other executive committee members of the Academy, Professor Jim Williams, spokesperson for integrity, Professor Lynn Beasley, Secretary for Education, and Professor Ivan Morisic, Secretary for Physical Sciences and the Vice President, and Professor Ian Chubb, Secretary for Science Policy, and Professor Frances Aparovich will come and join us, and she's engaging with the Lindau Fellows at the moment in the Ian Potter House. She's our Foreign Secretary as well. Our Treasurer, Professor Marilyn Anderson, could not join us, and then she is in Melbourne, and then hopefully you'll be able to meet with her later as well. With that, the first fellow we'll call upon is from the physical sciences, so I'll now hand over to Malcolm. Malcolm, please.
Thank you, Jagadish. Bob and I are delighted to be here today to officiate the admission ceremony for the fellows elected in 2024. The first fellow is Professor Nerali Abram. Nerali Abram is one of Australia's top paleoclimate scientists who has made outstanding contributions to our understanding of how Earth's climate system behaved over the last millennium at both regional and global scales. Her multidisciplinary approaches have brought critical perspectives to modern-day human-induced climate change, including bushfire, drought, the onset of anthropogenic warming, Abraham has had an impressive record of scientific leadership and has an international standing for her service, outreach and communication. Neroli.
Congratulations, Neroli. Congratulations, Neroli, and I welcome you into the Australian Academy of Science Fellowship.
I think every time I've been to Antarctica, that moment where I step out of the plane and onto the ice sheet, I kind of look around me and look at myself and go, I can't do this. And then you get on with it and you get the job done.
The job is to understand Earth's climate history. As a paleoclimate scientist, Professor Neroli Abram uses the planet's natural archives to reconstruct climate variability and change over the last 1,000 years.
So when we stand on the top of the ice sheet and we drill down through those ice layers, we're drilling back through time.
Each layer provides a snapshot of the weather systems that formed it, which can be used to build a picture of how these weather patterns have changed over time.
The past helps us to understand how the climate system works, so that then we can look at that through the lens of what's happening now to the climate to understand the processes that we expect to play out in the future.
Their findings couldn't be clearer.
The data everywhere that we look shows us just how unusual human-caused climate change is. We're pushing the Earth system far outside of the natural envelope that it's operated in and that our human society has been built around.
That's a point Professor Abram has repeatedly made, including as a coordinating lead author of the 2019 IPCC Special Report on the Ocean and Cryosphere in a Changing Climate.
I do get very frustrated at how slow we've been to act, because the evidence is clear. It's been clear for a very long time. And we hear a lot of talk about emission reductions and what we're doing, but when we look at the data, we actually haven't gotten past peak emissions yet. So at the moment, we're still making this problem worse every year by the greatest amount that we ever have. So this is a coral from Christmas Island in the eastern Indian Ocean. A core that's this long maybe has 50 years of climate information.
Professor Abram uses coral records in a similar way to ice cores. Her team used them to reconstruct a pattern of climate variability called the Indian Ocean Dipole, and demonstrated these cycles are becoming more frequent and intense due to human-caused climate change. That's a big deal because of the major impact that cycle has on Australia's drought and fire risk. In 2019, a positive Indian Ocean dipole drove the extreme hot and dry conditions that led to the disastrous Black Summer bushfires.
And when we look at that evidence that we have from the past, we look at what we see in our observational record, and then we combine that with projections from climate models for the future, that tells us a consistent story that because of human-caused climate change, we expect this variability to become stronger and have more of an influence on Australia's climate in the future.
Professor Abram hopes becoming a Fellow of the Australian Academy of Science will help amplify the findings of climate change science.
The planet, I think, is worth fighting for. That sort of empowers me to make sure that we're doing everything that we can to get the scientific evidence out there so that it can be used to make good decisions about how we address climate change.
Professor Andrew Blakers. Andrew Blakers has made major contributions to the advancement of solar energy as the key to decarbonising the global economy. Blakers is globally prominent in the analysis of 100% renewable energy systems and developed the global atlas of pumped hydro energy storage. He is co-winner of numerous awards, including the 2023 Queen Elizabeth Prize for Engineering and the 2018 Eureka Prize for Environmental Research.
Congratulations, Andrew. Congratulations, Andrew. I welcome you to the Australian Academy of Science Fellowship.
Solar farms like this one are now commonplace. But when Professor Andrew Blakers first started working on solar cells in the 1980s, few saw their true potential.
It was laughable to those in charge of large-scale energy production. Solar energy was widely perceived as perfect for satellites or the middle of the Simpson Desert. But as for serious power, no.
Alongside Professor Martin Green at UNSW, Professor Blakers set about changing that, breaking a series of world records for solar efficiency. In just a few years, they took silicon solar cell efficiency from the previous record of 16.5% up to 20%.
I think it was a combination of new equipment, new ideas, and simply determination.
That determination led them to develop the PERC cell and increase efficiency again, this time to 22.8%, by introducing an additional layer on the back of solar cells. The layer served to reflect unabsorbed light and electrons back into the cell, allowing more electrons to be collected and ultimately converting more sunlight into electricity. Their PERC technology now dominates the global solar panel market.
It was also very clear to me that silicon solar cells in particular had an unlimited future because there was never going to be a short supply of materials or availability of land and resources. And the growth of this industry would eventually lead to very dramatic reductions in price. And this is exactly what has happened. Now solar energy is the king of energy production.
An achievement that caught the attention of a king of a different kind. Professors Blakers and Green were among the recipients of the 2023 Queen Elizabeth Prize for Engineering, awarded to them at Buckingham Palace by King Charles.
And that was really something. I've been to Buckingham Palace, but never from inside.
Since UNSW, Professor Blakers has founded the Solar Cell Research Group at the Australian National University, and now leads a team researching 100% renewable energy futures and pumped hydro energy storage.
Pumped hydro is 97% of all energy storage for the electricity industry, but almost all of the pumped hydro systems are on rivers. But rivers are only 1% of the global landscape, so you throw away 99% of the potential pumped hydro sites if you confine yourself to rivers.
His team has developed a global atlas of potential off-river sites for pumped hydro.
You can pan and zoom across about 1 million sites around the world, including 5,000 or 6,000 in Australia, and click on tunnel routes or reservoirs to get 26 pieces of information for all 1 million sites.
For this new fellow of the Australian Academy of Science, one thing has become certain.
So 80% of new generation capacity around the world in 2023 was solar and wind, and 99% in Australia. And this is increasing every year. The game is over. Solar and wind have hands down won the energy race.
So our next new fellow is Professor Rachel Buchbinder. Rachel Buchbinder is a rheumatologist and clinical epidemiologist, internationally recognised for her work on musculoskeletal health. Her work includes evaluation of a world-first mass media campaign that successfully shifted misconceptions about back pain. She has led over 20 musculoskeletal trials that have challenged the value of accepted treatments, been influential in changing paradigms about the value of placebo surgical trials, and tested and implemented strategies to improve care.
Congratulations, Rachel. Congratulations, Rachel, and I welcome you to the Australian Academy of Science Fellowship.
Professor Rochelle Bookbinder has two jobs. The first is running her small rheumatology practice in Melbourne.
You put your chin on your chest, put your head back.
And the second is as a distinguished researcher in the School of Public Health and Preventive Medicine at Monash University.
I get great satisfaction from doing both of them, and the patients just give me ideas for what needs to be studied.
But her career as a researcher got off to a shaky start when, as part of her PhD, she was tasked with giving lab rats arthritis.
When my supervisors were surprised that none of the rats had developed arthritis, I actually had to come clean and say I couldn't actually bring myself to inject the rats.
She went on to do a master's and later PhD in clinical epidemiology, the basic science of clinical medicine.
There's a lot of good things about modern medicine. We've had a lot of wonderful discoveries that have cured disease and increased people's longevity. But the current play is also that there is too much medicine, and we know from studies that about 30% of medical care provides no benefit to the patient, and about another 10% is actually harmful.
Professor Bookbinder is co-author of the book Hypocrisy, which argues the benefits of treatment are often overstated and harms understated, while overtreatment and overdiagnosis are rife. She says the result of medicine being driven by business models is a not fit for purpose medical system that prioritises health care over health.
Well, if there was one thing that I could change, I would completely get rid of fee-for-service, and we would all be salaried. We'd all get the same income irrespective of how many patients we saw.
In 2009, she published the findings of a randomised placebo-controlled trial of vertebroplasty, a common treatment for painful spinal fractures that involves injecting the damaged vertebrae with a cement.
Even when we were doing the trial, I was already receiving negative feedback. People thought I was being unethical by studying a treatment that was well accepted. And my answer to that was, well, we don't know that it actually is better than not doing it, and we need to know the evidence, and I think it's unethical not to study the procedure.
Her conclusion that the treatment was no more effective than a placebo led to a flood of abusive emails and calls.
So when you think about the reasons that they're attacking me, often it's because they've got vested interests. They believe with their own eyes, so they think that what they see is the truth, and they don't understand that you need a control group, and that just because someone gets better with your care, it doesn't mean that it's because of your care.
The backlash only made Professor Bookbinder more determined. She's now published more than 20 musculoskeletal trials that have challenged the value of accepted treatments. But she's also committed to finding things that work, and was highly cited for evaluating a world-first media campaign in Victoria that successfully shifted misconceptions about back pain in the late 1990s.
That exercise is a safer and surer bet than an operation.
It was really at a time where you could use TV to get messages out to the general public. Unfortunately, I don't know that you can do it in the same way now.
That's why Professor Bookbinder says it's critical to keep exploring new ways of promoting evidence-based health messaging, something she hopes to do as a Fellow of the Australian Academy of Science.
And if you don't think about things in a scientific way, then you can end up with the wrong answers. You can be easily led by people that want you to believe something different to the truth.
Professor Kylie Catchpole. Kylie Catchpole is a leader in the research field of solar energy. Her pioneering work on the mechanisms of nanoscale interaction between light and semiconductors established new approaches to improve light absorption in solar cells. She has also led the creation of solar hydrogen technology that has been internationally recognized for the achievement of high-efficiency hydrogen generation using low-cost materials.
Congratulations, Kylie. Congratulations, Kylie. I welcome you to the Australian Academy of Science Fellowship.
Helping the environment was something Professor Kylie Catchpole always wanted to do.
I really didn't see myself as an environmental activist, though, and I didn't really know what to do with it. So I studied physics. And I got to the end of my degree and I thought, well, what shall I do now? It wasn't really clear.
A question that ultimately set her down the path of solar energy research. Today, she's a world expert on all things solar and jointly leads a group here at the Australian National University on advancing perovskite solar cells. While silicon has long been the dominant material used in solar cells, perovskite has emerged more recently as a low-cost alternative with potential to be as efficient as silicon. A potential proven with world records set by Professor Catchpole's team. In 2022, they took perovskite solar cell efficiencies to 22.6%, breaking their previous record of 21.6%.
Perovskite cells now have efficiencies that are similar to silicon solar cells, so just a single perovskite cell, they have efficiencies, the top efficiencies around 25%, whereas the top silicon cells are around 26%.
Professor Catchpole has also led the development of more efficient tandem solar cells. These systems involve stacking a perovskite cell on top of a silicon one, an advancement that could push the solar efficiency ceiling even higher.
The perovskite cell will absorb the blue light really well and give you a higher voltage, and then the red light will actually pass through the perovskite solar cell and be absorbed by the silicon solar cell, and then that will give you additional voltage. So together, that can give you a higher efficiency.
In theory, the efficiency limit of a two-cell tandem system sits as high as 44%. But more efficient and affordable solar power isn't just the end game for Professor Catchpole. She also sees it as a means to help produce another type of renewable energy, green hydrogen.
We'll need hydrogen to produce various materials to refine metals, for example. And if you can produce those using solar energy, then you can reduce the cost of hydrogen.
In another project, her team connected their solar cells with a catalyst to directly generate hydrogen at high efficiency. Efforts that Professor Catchpole hopes will accelerate our path to decarbonisation, both globally and here in Australia.
There's an extremely bright future for renewable energy in Australia. There's just so much opportunity. We're really blessed with the amount of renewable energy resources we have, and we should make use of them.
A message she now shares as a new Fellow of the Australian Academy of Science.
Professor Mary Garson. Mary Garson is a world-leading organic chemist. She's an internationally recognised expert in chemistry, biosynthesis and bioactivity of secondary metabolites from both marine and terrestrial environments. Best known for her pioneering use of stable radioactive isotopes to define unusual biosynthetic pathways, she probes complex stereochemistry, ecological interactions and environmental aspects of marine and plant metabolites.
Congratulations, Mary. Congratulations, Mary. I welcome you to the Australian Academy of Science Fellowship.
When Emerita Professor Mary Garson came to Australia, the first thing she set out to do was learn how to snorkel and scuba dive.
I needed to go underwater and collect the specimens myself. And this was a bit of a voyage of discovery for me, a voyage of learning, because I could not swim very confidently.
She was researching marine sponges and the defences they use in their natural environment. Professor Garson was fascinated by their chemistry.
Looking up the chemistry of marine sponges is a little bit like trying to decipher a molecular jigsaw puzzle. We use a technique called nuclear magnetic resonance spectroscopy to investigate the different types of atoms, the hydrogens and the carbons in the molecule, and also to understand how those atoms are connected to each other.
Life underwater is a competition for space and food. Marine sponges use a range of bioactive chemicals to defend themselves against predators such as fish and crabs. Professor Garson's pioneering work has been on investigating the function of these chemicals and defining the unusual pathways through which they're manufactured.
And I was able to establish that, amazingly, the marine sponge is taking a precursor, cyanide, which is a poison, and converting it productively into a bioactive molecule, diisocyanoidosine, which it can use in its chemical defense.
Sponge metabolites like diisocyanoidosine often provide protection in other ways, such as by inhibiting the growth of pathogens and other marine biota. Some of these molecules also have pharmacological properties that could be effective against cancers and infectious diseases. Professor Garson says she's had an enriching career not only because of the scientific discoveries. One of her personal highlights was having a marine flatworm, Maritigrella merrigarsoni, named after her.
When I was a third-year student back at university in England, if you had said to me that I would end up going scuba diving on the Great Barrier Reef, undertaking science on marine bioactive chemicals, and ending up with a little marine animal named after me, I would have said, oh, you must be joking.
Professor Garson has shifted her focus to leading and inspiring the next generation of scientists. Since 2019, she and her colleagues at the International Union of Pure and Applied Chemistry, or IUPAC, have run an initiative called the Global Women's Breakfast.
What happens at one of these breakfast events is that there is informal conversation about breaking barriers in science, catalysing diversity, and the conversation includes men as well as women.
Professor Garson is now Vice President of IUPAC and in 2026 will become its Global President, the third woman and second Australian ever in its 104-year history to hold this title. She sees this as an opportunity to help put Australia firmly at the centre of the global science community, a mission she also hopes to propel forward as a Fellow of the Australian Academy of Science.
Professor Arthur Georges. Arthur Georges is an ecologist and herpetologist who is an internationally recognised expert on the evolution and ecology of reptile sex determination. His proposal that genetic and environmental sex determination represents ends of a continuum has revolutionised understanding of how the complex thermal environment of nests dictates sex in reptiles. His work redirected thinking on how reptiles survive past climate changes and how human activities constrain their ability to respond to future challenges.
Congratulations. Congratulations, sir. I welcome you to the Australian Academy of Science.
Distinguished Professor Arthur George's journey to becoming one of Australia's top scientists began when he was 15. He clearly recalls reading the popular science books of Isaac Asimov.
Those books were outstanding, and he switched me on to the delights of science. I decided to be a scientist from that point on.
He says the key to being a good scientist is having a genuine passion for tackling problems.
You have to have fire in your belly about the process of discovery and also about the area in which you're working. It's not a 9-to-5 job.
Professor Georges is an ecologist and herpetologist who studies the evolution and ecology of reptile sex determination via both traditional ecological observations and high-powered genomics.
Little fellow just behind me on the right-hand side is a central bearded dragon. They're found in the semi-arid regions of Australia, and he's the model species that we use for investigating the research questions that we're interested in.
Many of those questions involve epigenetics, or the set of switches that regulate genes without changing the underlying DNA.
Whether an animal is a male or a female is determined by its development, and that development can be modified by environment to change the trajectory from, in the case of the dragon, from a male lizard to a female lizard.
It's a form of developmental reprogramming that Professor Georges has demonstrated in the lab by changing the incubation temperature of the reptile's eggs. But to his surprise, in 2015, he, along with collaborators Claire Hollerley and Academy Fellow Distinguished Professor Jenny Graves, identified the first instance of bearded dragons experiencing temperature-driven sex reversal in the wild.
And of course, that means that as the climate warms, there's a real possibility that there'll be an increase in the frequency of sex reversal in the wild, males being converted to females, an oversupply of females, and then evolutionary processes come in to drive the heteromorphic sex chromosome to extinction. In the last several 100,000 years, the climate's gone up and down by about 8 degrees, and these animals have persisted. So the question is not so much, are they going to be in trouble because they've experienced these sorts of things in the past. The question is, what have we done to constrain their ability to respond in the way in which they've done in the past? And of course, simplification of the habitat through grazing and habitat modification has constrained their abilities to respond in the way in which they have in the past. And are we going to accelerate their extinction as a result?
The applications of his research aren't limited to dragon lizards. Professor Georges says studying animals often leads to breakthroughs for humans.
We're not operating in a vacuum. We're not particularly interested in dragons. We're interested in the dragon as a model for probing how epigenetic processes can program or reprogram developmental outcomes.
Professor Georges says becoming a Fellow of the Australian Academy of Science will give his research greater prominence, allowing for even greater collaboration.
It used to be publish or perish. Now it's partner or perish. You need to link up with the best possible people that are willing to address the questions that are of interest to you. It's really important.
Professor Rosalind Gleedow. Rosalind Gleedow is a world leader in the field of cyanogenesis, the process of cyanide release from stable bioactive compounds in plants in response to herbivory. She has integrated opposing theories of plant defense to demonstrate that these bioactive compounds are intricately involved in nitrogen management, which is the evolutionary driver, not herbivory resistance. Her work explains why so many crops are cyanogenic and helps to predict changes in nutritional value of plants in response to environmental challenges. She actively translates her work to impact food security, human and environmental health, and crop adaptation.
Congratulations, Weston. Congratulations, Weston. I welcome you into the Australian Academy of Science Fellowship.
Professor Rosalind Gleadow studies plants that kill. She's a world leader in the research field of cyanogenesis, the process by which plants release cyanide.
About 10% of all plants make cyanogenic glucosides, but when you go to crop plants, it's at least 30%, if not 50% of plants make these compounds, which make them really interesting because somehow humans have had this love affair with plants that make toxic cyanide.
A love affair that Professor Gleadow and her team began exploring with two highly toxic but widely eaten crop plants, cassava and sorghum. While the sorghum grain is safe to eat, its leaves contain dangerous levels of cyanide, and cassava roots can cause death and paralysis if not properly processed.
What was the big surprise, both with sorghum and to some extent with cassava, is the plants that are less toxic don't actually grow as fast when they're young as the plants that are toxic.
At the time, it was thought that toxicity was a defense mechanism against herbivores, but their findings suggested something more nuanced was at play. They discovered that more toxic varieties, such as the bitter cassava, tend to be better at tolerating environmental stresses such as drought. And importantly, when under stress, these plants seem to store the nitrogen that's usually used for growth and photosynthesis inside their cyanogenic compounds.
There's no point in putting any resources they have into photosynthesis because they're not going to be able to grow anyway. Why not store it as something toxic so the animals won't eat you? And then as soon as there's some rain, they can remobilise that and then grow.
This efficient management of nitrogen means these plants tend to grow better. But climate change has thrown a spanner in the works. Professor Gleadow says the effects of global warming could make the crops of the future more toxic and less nutritious. That has huge implications for food security.
You've got decreased protein in the grain, which means that people who rely on grain for their protein are going to need to eat more grain. The cattle that rely on grass for eating are going to need to eat more grass to satisfy their protein level. So that's from the direct effect of carbon dioxide. Then if we have drought and heat effects, we're going to have reduced plant production. It's not a happy story.
Professor Gledow's own academic story has been a rather unconventional one. After doing her master's, she worked in various roles, including as an educator, before starting her PhD at 39.
I thought, well, if I'm going to stick around a university, I absolutely have to have a PhD, right? It's just a ticket. What I didn't realise is that I absolutely adored research, totally adored it, and just loved what doing a PhD did to my brain in sharpening up my thinking.
She now joins her husband, Professor Andy Gleadow, as a Fellow of the Australian Academy of Science and hopes her journey can inspire others.
The fact that I can have this amazing honour and yet have come through a rather unorthodox pathway is, I hope, an encouragement to people who, for various reasons, no matter what their background, have a different route that they've taken.
Professor Dmitry Goldberg. Dmitry Goldberg is a pioneer in the fabrication and analysis of diverse inorganic nanostructures with an emphasis on boron nitride nanotubes, nanoparticles, and graphene-like nanosheets, as well as design, development of sophisticated methods in their in situ operando transmission electron microscopy for the physical properties exploration. These methods allow for real-time investigation and provide the deepest insight into a holy grail problem in material science, establishing a relationship between the nanomaterial structure and its functional properties for implementation into modern and future technologies.
Congratulations, Dimitri. Congratulations, Dimitri. I welcome you into the Australian Academy of Science Fellowship.
Most of what Professor Dimitri Goldberg studies can't be seen without an electron microscope. His research involves studying nanomaterials at the atomic level.
You see everything like in real life, but everything downsized to 10 million times. It's about 10,000 times thinner than a human hair.
In the lab here at the QUT Centre for Material Sciences, Professor Goldberg synthesises his own nanomaterials. Then, using a technique known as in situ transmission electron microscopy, he manipulates these structures and observes how they change under different conditions such as heat, stress and light. Through tensile strength tests, his team discovered carbon nanotubes are about 100 times stronger than steel.
In theory, it was predicted that all these nanostructures are much stronger, but before these tests were initiated in the microscope, people actually didn't know what's the real properties of these nanomaterials.
Professor Goldberg began his research career in Moscow before joining the National Institute for Materials Science in Japan in 1995. There, he became one of the first researchers in the world to study the properties of boron nitride nanotubes.
By serendipity, we found that these tubes exist in nature, because at that time only carbon nanotubes were known to the society.
It turned out boron nitride nanotubes are not only as mechanically strong as carbon, but they also have some other impressive qualities.
Compared to carbon, which can be easily oxidized, boron nitride is very stable to any type of oxidation. So it means for any type of high temperature applications, boron nitride tubes are preferable opposed to carbon.
Today, boron nitride nanotubes may be used to reinforce aluminium, a popular material for aircraft. It's research with a wealth of real-world applications. Professor Goldberg has more than 130 patents, primarily in polymers.
Once you put these nanomaterials into the polymer matrix, first of all, you can increase the strength, and second, you can dramatically increase the thermal conductivity.
Beyond the applications, Professor Goldberg also has a deep appreciation for the images he sees under the microscope.
When I was a boy, I was very much interested in painting, so I have a choice where to go to become a painter or scientist, and I found that this electron microscopy is a good match between art and science.
In 2016, Professor Goldberg received an Australian Laureate Fellowship, prompting him to move to Queensland, where his children were living. He says he's honoured to now also be a Fellow of the Australian Academy of Science.
In Australia, I think people of different nationalities, they can feel Australia home. And I'm still very much happy that at some point of my career we came to Australia.
Professor Michael Kearney. Michael Kearney is an ecologist. His transformative work has forecast the future and fate of diversity through the development of mechanistic species distribution model. In contrast to other approaches, its mechanistic nature enables projections for the novel environments that changing climates will bring. Kearney's work has transformed biodiversity, forecasting globally, delivering significant benefits to fundamental and applied biology and the well-being of people and the ecosystems we rely on.
Congratulations, Michael. Congratulations, Michael. I welcome you to the Australian Academy of Science Fellowship.
Professor Michael Carney always knew he wanted to be an ecologist.
My interest in biology started from a really young age, in my backyard, finding interesting animals and plants like blue-tongued lizards and cicadas.
So it's been a dream come true for him to now travel to observe the diversity of animals, plants and environments across Australia. What he didn't expect was that physics would also become a crucial part of his research.
So how hot can a species get? Has it got enough water? Has it got enough food? How fast is its life cycle going? When are the offspring emerging? Are they emerging into a time when there's lots of resources, when it's stressful? These kind of constraints we can actually work out with physics.
Professor Carney has developed models that help predict the distribution of species in novel environments. His models link organisms' biological processes with environmental physics to understand the impacts of changing climates and human activity.
So if we're going to make good decisions about particular kinds of organisms that are important to us, we really need to understand the processes by which they're linked to the climate. And so these models, by going into the details of those processes, can help us make better management decisions.
Professor Carney's process-based models rely on an understanding of the conditions in an organism's immediate surroundings, known as their microclimate. The models are developed not only from being in the field, but also from taking measurements of animals and their characteristics back in the lab.
A little grasshopper sitting out on a rock in the sun is not experiencing the air temperature that a weather station would record, which is about 1.5 meters off the ground.
Professor Carney's work has made it possible to accurately calculate the little grasshopper's experienced conditions within its microclimate.
We can calculate the temperature of an animal out in the middle of nowhere in Australia within a couple of degrees and work out, whether it's going to get too hot, whether it's going to run out of water, those sort of things.
That's useful not only in conservation efforts, such as determining where to translocate an endangered species, but also in controlling the spread of pests, such as cane toads.
And so we were able to apply this approach by understanding the physics of where the cane toad can and can't live to really identify the no-go zones where cane toads will definitely not be able to go because of physical constraints.
Professor Carney has since worked to make these tools accessible to ecologists through web-based applications.
And then the measurements themselves that we make in the field allow us to test the models. So there's a beautiful feedback between theory and observation that we get from having these models.
A transformative approach to ecology that has now seen Professor Carney elected as a new Fellow of the Australian Academy of Science.
Professor Matthew Kiernan. Matthew Kiernan is an expert in neurodegenerative diseases, particularly motor neuron disease and frontotemporal dementia. He has devised and introduced neurophysiological techniques that enable ion channel function to be studied in vivo in humans. This has enabled the delineation of normal function, mechanism of disease, and therapeutic targets. He has defined diagnostic criteria for disease and established consensus guidelines adopted globally by the World Federation of Neurology. Kiernan is implementing novel therapies based on these discoveries and using his techniques to measure responses to treatment. These are now being trialled in centres worldwide.
Congratulations. Congratulations, Matthew. I welcome you to the Australian Academy of Science Fellowship.
Professor Matthew Kiernan was a young doctor and researcher in the 1990s when his mum was diagnosed with motor neuron disease.
So when my mother, couldn't walk, I wasn't actually sure what it was. And then when it's diagnosed as motor neurone disease, there's no treatment. You think, well, that's hopeless.
That experience motivated him to help in the clinical trials for what remains the only approved medication for the condition in Australia, known as Riluzole.
To have been part of that journey and to improve an outcome for a condition means the world. Is that good enough? No, it's not good enough because people still die from the conditions. There's still a lot more work to be done.
A lot of that work is being done here at Neuroscience Research Australia, known as Neura. As Chief Executive of Neura, Professor Kiernan oversees more than 400 researchers in studying the brain and nervous system.
Just turn the hands over and just close your eyes and keep them there.
But to this day, he still finds time to see patients.
Just doing pure, very fundamental research, isolated, without thinking, where is this going, is not good research.
His own research helped change the way neurological disorders are diagnosed. While working in London at Queen's Square, Professor Kiernan devised a non-invasive way to study the behaviour of ion channels in patients. A major advance, given those channels control the flow of electrical signals in nerves. Soon after returning to Australia, he was able to prove the value of the technique when asked to consult on a group of patients who'd eaten a soup full of the poisonous pufferfish.
And then over the next hour or two, they started to notice tingling around their lips, tingling in their hands and feet, gradual paralysis. By the time they came to Prince of Wales, one of them needed to go to ICU, was ventilated. Then we were able to, with our techniques, do mathematical modelling, and we were able to show that, in fact, all of the abnormalities we had recorded was due to blockade of their sodium channel. So it was a proof of concept.
Across his career, Professor Kiernan has remained at the forefront of innovation. That includes better understanding the genetic underpinnings of brain disorders, and particularly a specific gene mutation called C9orf72, traced to a person in Finland from about 1,000 years ago.
And a colleague here, Emma Devaney's interviewed now 1,400 family members of patients with this genetic mutation and found very high incidence of schizophrenia, mania, bipolar disease, Parkinson's disease, multiple sclerosis. So these are conditions of the brain and mind that are all linked.
Knowing that opens up the possibility for new genetic therapies.
But then also understanding when you get a gene, it doesn't mean that you're going to develop the condition. And it turns out that 30 to 40% of people with that gene will manifest the disease, so 60% don't. So there's presumably things in our environment, epigenetic approaches, and that really is taking this forward.
Professor Kiernan says becoming a Fellow of the Australian Academy of Science is recognition of the value of working not just in pure science, but also in its eventual application.
Being engaged with the community, seeing progress, coming up with better therapies, that's the joy of an applied scientist.
Professor Glenn King. Glenn King is the leading figure worldwide in the study of arthropod venoms and the development of venom peptides from these animals as pharmacological tools, human therapeutics, and bioinsecticides. He has made seminal contributions with respect to our fundamental understanding of the evolution, ecology, production, and functional and structural diversity of arthropod venoms. His work has resulted in safer ways to control disease, spreading pests, and to protect crops, which have been recognised by the 2023 Prime Minister's Prize for Innovation.
Congratulations. Congratulations, Glenn. I welcome you to the Australian Academy of Science Fellowship.
If you aren't a fan of spiders, a visit to Professor Glenn King's laboratory may change your mind. His lab at the University of Queensland houses one of the largest collection of venoms in the world. And within these venoms are a treasure trove of molecules known as peptides that could be harnessed for good.
And we use venomous animals to develop eco-friendly insecticides and human therapeutics.
It's research Professor King first became interested in the 90s, when people started realising some insecticides are very harmful not just to pests, but also other animals and humans. So Professor King set out to find an alternative.
And we went and looked at the best insect killers on the planet, which are spiders. They'd been working on this problem for up to 400 million years. They'd probably worked out the best way to kill insects.
Not everyone was convinced, as spider venom peptides were thought to be too unstable and not orally ingestible by pests such as caterpillars.
Turns out that many spider venom peptides are orally active, and for that to happen, they have to have two properties. One, they have to survive the insect gut, which like our gut is really, really good at chewing up proteins and peptides. Then they have to cross the insect gut to get into the hemolymph, the blood system, if you like, of the insect. It turns out many of them can do that as well.
He found that several peptides found in venom of the Blue Mountains funnel web spider selectively kill insects, including key agricultural pests. Importantly, these peptides don't hurt pollinators such as bees. Realising the commercial potential of this discovery, Professor King founded the agricultural biotechnology company Weston in 2005.
This year, they released the first product for use on broadacre crops, things like corn, cotton, soybean, and that's where it's going to have a really major impact around the world.
He's also enlisted the help of spiders to treat human conditions such as stroke and heart attack.
So your brain becomes acidic during a stroke, and it turns out that activates a receptor that causes the nerve cells to commit suicide. And our simple idea was, what if we could find an inhibitor of that receptor?
They discovered the venom of the Gari funnel web spider has a very potent inhibitor for this specific receptor. In experiments with rodents, delivery of the inhibitor within the first few hours of stroke reduced brain damage by 80%. And in a heart attack, that same inhibitor reduced damage to the heart by about 40%.
So stroke and heart attack together are responsible for almost 30% of all deaths around the world every year. And yet, remarkably, we don't have a single drug to prevent the damage caused to the tissue in either of those cases. If we could change that, it would be profound in terms of the treatment of people that have a stroke or heart attack.
Professor King's groundbreaking work saw him awarded a Prime Minister's Prize for Innovation in 2023. And it's something that this new Fellow of the Australian Academy of Science says also speaks to the power of doing basic research.
Professor Mark Krumholtz. Mark Krumholtz is a world leader in the study of star formation and the interstellar medium. He has contributed fundamentally to our understanding of physical mechanisms that control the rates at which galaxies form stars, the mass distribution of those stars, and the way stars cluster in space and time. These advances have been enabled by new techniques that Krumholtz pioneered in computational astrophysics.
Congratulations, Mark. Congratulations, Mark. I welcome you into the Australian Academy of Science Fellowship.
Professor Mark Krumholtz uses computers to help solve the mysteries of our universe.
My particular focus is on how stars form, where do they come from, when they die, where does their material go, how are galaxies assembled.
He models the gas between stars that form part of the interstellar medium. These interstellar gases collapse together under the pull of gravity, forming new stars. But when those stars inevitably die and explode as supernovae, they drive the gases back out into the galaxy.
So you have this constant tug of war between gravity trying to pull everything in and the newborn stars that try and push everything back out.
These processes take place over millions of years, making them hard to observe and measure. That's where Professor Krumholz's work as a computational astrophysicist comes in.
We can't watch this happen in real time, but we think we understand the basic physical principles that govern, for example, the flows of gas, the way stars work. And you can build those physical principles into a computer simulation.
Simulations that could help inform new observations or paint a picture of what galaxies millions of years into the future might look like. It's research that requires immense computational power, more than the level offered by current infrastructure here in Australia. In 2022, Professor Krumholz was awarded an Australian Laureate Fellowship to develop new software to drive the nation's supercomputers to peak performance. But he says Australia still lags behind in terms of hardware.
If you want to be at the cutting edge, you actually have to replace the computer every 5 years or so. So it looks like that.
As a kid, Professor Krumholz wasn't particularly interested in astronomy, but loved maths and physics.
I grew up with a view of the New York City skyline, so there were no stars. It was that I, sort of at an early age, found it fascinating that you could use math to represent reality. It seemed like magic.
In 2016, he moved across the globe to his current role as professor at the ANU Research School of Astronomy and Astrophysics. He's proud to now also be a Fellow of the Australian Academy of Science.
It feels like it's a sign of acceptance that, I'm not viewed anymore as an outsider, that I'm actually now a member of this community, and that's nice.
Professor Xinghuo Yu. Xinghuo Yu is a renowned geoscientist whose work has led to paradigm shifts in our understanding of how the Earth's engine works. This is important because it catalyzes the formation of deep Earth resources as well as climate and environmental changes. He played prominent roles in the discovery of the periodical occurrences of two pre-Pangea supercontinents. He also co-developed the dynamical supercontinent superplume geodynamic model, addressing the driving forces of plate tectonics.
Congratulations, Xinghuo. Congratulations, Xinghuo. I welcome you into the Australian Academy of Science Fellowship.
Emeritus Professor Zhangxiang Li grew up during China's Cultural Revolution. It was an era when high school graduates were commonly sent to work on farms or in factories, as all the country's colleges and universities were shut down.
All of a sudden, the Cultural Revolution finished. The university reopened. Then my teacher told me, you should think about going to university. I think, what?
Professor Li calls this the first turning point in his life.
At the age of 16, and from a small village, I knew very little anything, really. So suddenly I meet all those people who know everything. So to me, I think I grew up in the university.
His studies ultimately brought him to Australia for his PhD and led to a decades-long career in geoscience. He's interested in what drives plate tectonics and how the movements of these plates have shaped our planet over time. Professor Li and his colleagues often go into the field to test their hypotheses. That involves finding the critical information they seek in ancient rock samples.
So we can date them with analyzing the magnetic needle recorded in the rock. So that's called paleomagnetism. You're lucky, we found the original signal kept in the rock. So we can use that to analyzing the history of the Earth, like DNA, how the Earth moved, how they moved, and also we can compare the DNA of one rock from one continent with that of the other. If they match, we know, those two continents were together in certain way. Perhaps the first supercontinent.
A world authority on the assembly and breakup of supercontinents, Professor Lee played a pivotal role in reconstructing the history of pre-Pangea supercontinents Rodinia and Nuna.
So Nuna formed at 1.6 billion years, survived for 300 million years. Then 600 million years later, Rodinia formed 900 million years and broke up at 700 million years, survived 200 million years. Then another 600 million years later, Pangea assembled. Now you start to see a pattern, a cycle.
Professor Lee and his colleagues established that Earth's evolution for the last 2 billion years has involved this cyclic assembly and breakup of supercontinents. He says we're right in the middle of another cycle, with his team's models predicting the next supercontinent could form in about 280 million years through the closing of the Pacific Ocean.
2.6 billion years old.
Professor Lee says it's only by understanding the past that we're able to recognise the processes that drive the present and predict what will happen in the future.
Everything from Earth, from the magnetic field which protects us from the solar flare to the water we drink, the food we eat, only by understanding this evolution and the process, so we can understand the planet which really we call home.
His work has now seen Professor Lee elected as a Fellow of the Australian Academy of Science.
I've never dreamed of this career going this far, and now become a Fellow of the Australian Academy. It's something beyond way beyond my dream. It's a lucky man, and born in a lucky time, came to a lucky country.
Professor Georgina Long. Georgina Long is an oncologist and scientist specialising in melanoma, who has led an extensive program of clinical trials revolutionising the way melanoma is treated worldwide and leading to Australian government funding of 10 new melanoma drugs which have significantly increased survival. She has introduced the first new checkpoint inhibitor drug into clinical oncology in over a decade. Her recent work is now revolutionising how operable cancers are treated. In recognition of the contribution of her outstanding research and work, Professor Long was awarded the 2024 Australian of the Year, jointly with her colleague, Professor Richard Scolyer.
Thank you. Congratulations. Congratulations, Georgina. I welcome you to the Australian Academy of Science Fellowship.
Professor Georgina Long grew up believing all of us can have an impact.
In my family, it was what you were passionate about, what you were good at, and how you could make a difference.
How are you? Come on in.
That mission to make a difference has seen her become a medical oncologist.
So all of that's normal.
And one of the world's leading researchers of melanoma.
Melanoma is Australia's cancer. We have the highest incidence of melanoma in the world. When I went into it, there were no effective drug therapies. It was pretty depressing.
Professor Long would go on to lead an extensive program of clinical trials,
I'm just selecting my antibody.
resulting in 10 new melanoma drugs, including immune checkpoint inhibitors that help T cells recognize cancer cells as an enemy.
So I lived through this incredible journey of all of a sudden, patients were doing a Lazarus. They were dying and then they were living. High-dose ipilimumab.
Her latest trials have centered on combination immunotherapy targeting multiple checkpoints. The results show the approach categorically improves survival rates.
And as time goes by, we now know that about 50% of people with melanoma that's spread, so advanced melanoma, are having long-term durable control of their cancer. Just short of a cure. We're not brave enough yet to say cure, but I'll say it, it's a cure.
Her Her research saw her become Joint Australian of the Year with pathologist and fellow melanoma researcher Professor Richard Scolyer in 2024. When Professor Scolyer was diagnosed with an aggressive form of brain cancer known as glioblastoma in May 2023, Professor Long was determined to intervene.
This problem was too big, and the grief was so great, and I wanted hope for Richard.
That hope would be found in Professor Long's game-changing research into treating secondary melanoma tumours in the brain. Specifically, the work that she led with her colleague in the Netherlands showed that immunotherapy could be used to shrink a tumour before surgery and improve outcomes. So could the same also be true for glioblastoma? Professor Scolyer consented to the treatment, which involved a pre-surgery immunotherapy regimen devised by Professor Long and supported by experts worldwide.
The ideas made sense. They were grounded in experience. Of course, there were many unknowns, and they were grounded in science, and that gave hope.
A year on from his diagnosis, Professor Scolyer is cancer-free, and Professor Long is working to publish their results and establish clinical trials based on the treatment. Professor Long says that's the beauty of science.
Whatever problem we're faced with that really, really matters to humanity and to our planet and to the universe, it's going to be in science, the answer.
A message she can now share as a Fellow of the Australian Academy of Science.
Professor Budiman Minasni. Budiman Minasni is a soil scientist who has used breakthrough soil mapping and modelling techniques to unlock fundamental new knowledge urgently needed to protect and enhance soil carbon stocks and to sustain agricultural and ecosystems worldwide. His development of cost-effective digital techniques enables soil, organic carbon and other properties to be rapidly and precisely measured and mapped. Minasni's research underpins numerous critical real-world applications from soil carbon benchmarking to a global movement to increase soil carbon.
Congratulations, Budiman. Congratulations, Budiman. I welcome you to the Australian Academy of Science Fellowship.
If you think soil is boring, you haven't met Professor Budiman Minasni.
I find soil amazing. It's an amazing topic to study. Hello. This one is from Canberra, so this is from our university farm.
Soil plays a vital role in sustaining agriculture and ecosystems. Around 97% of our food comes from soil. It also purifies our water, helps prevent flooding, stores nutrients, and contains extraordinary levels of biodiversity.
So that's why I think that we should take care of our soil to ensure that we have a sustainable future.
Professor Minasni is recognised as a global leader in digital soil mapping. He's developed detailed maps of soil properties using underlying soil processes and machine learning supported by satellite and field data.
So all of these sensors are just like taking a fingerprint of the soil. From the fingerprint, then we can try to identify what are the constituents of the soil.
His methods are used by soil scientists around the world, from the paddock level to the global scale. The ability to rapidly and precisely predict and map soil carbon levels is shaping up to be a vital tool in the fight against climate change.
Soil is a carbon sink. Soil is the largest carbon storage on the land.
He wants to safeguard and enhance this carbon storage potential by improving soil quality. Here in Australia, his maps are helping to assess and improve land management practices.
with the help of remote sensing images and statistical tools, we are able to extrapolate this data, letting us know how much, for example, carbon is stored in every 30 meter in Australia.
Most Australian farmers now practice a technique called minimum tillage, where they don't remove the residues of previous harvests such as stubble and roots before planting new crops. That prevents a substantial amount of carbon from being released into the atmosphere, while also making the soil itself more robust.
So now we need the next step to increase further carbon in the soil. So there are trials now being conducted by our researchers trying to put cover crops in between the season and different types of varieties of cover crops to increase carbon back in the soil.
Professor Manasni grew up in Indonesia before moving to Australia to undertake his Master of Agriculture at the University of Sydney. 30 years later, he's still at the university and can now also proudly call himself a Fellow of the Australian Academy of Science.
It is an honor, and also it is a recognition towards the discipline that we know Soil Sciences Australia is changing the world.
Professor Jose Polo. Jose Maria Polo is a biochemist and stem cell biologist who has made seminal contributions to our understanding of the mechanisms of cellular reprogramming. By developing and combining advanced cellular models and genomic techniques with computational approaches, he has made major contributions to the reprogramming, embryo modeling, and stem cell fields. His work has driven a translation and expansion of reprogramming advances with additional research areas and industry.
Congratulations, Jose. Congratulations, Jose. I welcome you into the Australian Academy of Science Fellowship.
What makes a skin cell different from a brain cell? And can one type of cell be changed into another? As a biochemist and stem cell biologist, Professor Jose Polo is working to better understand these questions.
So that's why my lab works in so many aspects, but actually we are always working in the same thing, how cell A becomes cell B.
While every cell in the human body has the exact same genes, the epigenome controls what genes are expressed, which determines what the cell becomes. By harnessing these epigenetic processes, Professor Polo and his team are able to reprogram one cell into another cell type.
And we found a way to control how those genes are open or closed. We use these proteins that are called transcription factors that are like, I call it the librarians in that library that controls which books are open or closed, which genes are active or not.
Cellular reprogramming can use viruses or RNA to introduce these transcription factors into the cell. Professor Polo's lab has used these techniques to grow everything from lung cells to placental cells and even models of the early human embryo.
This is the pregnancy test of the cells?
Yes.
So it's already positive?
It's already positive.
During the pandemic, we were able to check if, for example, if the SARS-CoV-2 virus, the virus that caused COVID-19, was able to infect placenta cells. But we didn't need to collect placenta cells. We were able to make the placenta cells directly.
In 2012, Sir John Gurdon and Professor Shinya Yamanaka received a Nobel Prize for their foundational work on induced pluripotent stem cells, or iPS cells, which have transformed the field of regenerative medicine.
Induced pluripotent stem cells, or iPS cells, are super important because they are able to differentiate into almost every cell type of the body, which it means that you can take skin, reprogram them into iPS cells, and then from those iPS cells, we can convert them into heart cells, brain cells, blood cells, etc., etc.
But as a postdoc researcher, Professor Polo found that those cells carried an epigenetic memory of what they used to be, a discovery that was simultaneously being made by another postdoc researcher at the time, Academy Fellow Professor Ryan Lister. Years later, when they both came to Australia, they worked together, firstly to figure out why that memory was retained, and then to develop a way to wipe it.
And with that, we created a method that gave rise to corrected iPS cells, which we showed that they differentiate more homogeneously into all the cell types. They don't carry these epigenetic aberrations, and in general, they are easier to work with.
It's research with significant implications for disease modelling and drug screening.
And you can apply this to everything: cardiac problems, muscle problems, any problem where the problem is a cell that is not having the function correct. You could model it in vitro now, directly from a patient, and then test drugs.
Groundbreaking work that has now seen Professor Polo elected as a Fellow of the Australian Academy of Science.
Professor Xinghuo Yu is a geophysicist who has made fundamental contributions to the study of the Earth's interior, notably the inner core, using an innovative approach to detect shear wave propagation and so demonstrate the solidity of the innermost part of the Earth. His breakthrough work in the understanding of the seismic correlation wave field opened the way for global and planetary seismology to illuminate the interiors of the Earth and other planets. He has also made significant contributions to seismic source studies and analysis techniques of seismograms.
Congratulations, Troy. Congratulations, Florian. I welcome you into the Australian Academy of Science Fellowship. Thank you.
Professor Hrvet Kalčić is a modern terranaut. Instead of exploring space, he explores the Earth's inner core.
The inner core is a planet within our own planet with its own complexities.
Complexities he's worked to understand.
Here you can see the inner core.
Through studying seismic waves generated by large earthquakes. These waves traverse through the Earth's interior as vibrations. And by analysing information such as their speed, amplitude and shape, Professor Kalčić can make inferences about the Earth's internal structure and dynamics.
We use seismometers, these sensitive instruments that sense the ground motion during the passage of waves. We use it in a mode of an inverted telescope. So instead of the sky, we point it to the center of the Earth.
In 2018, Professor Kalčić and his PhD student published groundbreaking research that found the first unambiguous proof that the Earth's inner core is solid.
What we try to do...
They came up with an innovative approach to detecting shear waves in the inner core, a type of wave that can only travel through solid objects. The challenge with detecting these waves is they have such small amplitudes they can't be observed directly. But Professor Kalčić's team overcame this by developing the so-called correlation wave field method.
The best way to explain it intuitively is to think about the similarity of the tiny signals that we observe many hours after large earthquakes in the seismic wave field. The similarity between two tiny signals becomes our tool.
A tool that also allowed them to measure the speed of the inner core shear waves.
We found that it was lower than anticipated from the previous models of the Earth. And that also meant that the inner core is a bit softer than we anticipated.
Earthquakes aren't the only seismic activity Professor Kalchich is interested in. He's also studied Marsquakes. Using just one seismograph, he and another PhD student were able to scan Mars' core. They used mathematical trickery to turn the 20 or so Marsquakes they had detected into virtual receivers and their single receiver into a virtual source.
And so by applying this mathematical trickery on Mars, we were able to detect the presence of Mars' core and actually measure its radius.
As a university student, Professor Kalchich was initially interested in pursuing astronomy, but he soon found his calling in the earth sciences instead.
I realized that a knowledge about other worlds and other planets of the solar system is predicated on our knowledge about the Earth.
His own contributions to that body of knowledge has now seen Professor Kalchich elected as a Fellow of the Australian Academy of Science.
Conducting research in this area really, fulfills our role as pioneers in the grand journey of discovery.
Professor Jean Tyson. Jean Tyson is an internationally recognised microbial ecologist and bioinformatician who has developed and applied novel approaches to characterising microbial communities. He has pioneered new methods for studying microorganisms from diverse clinical and environmental settings, allowing us to characterise many of them for the first time. This has paved the way for many important discoveries, enabling a more holistic understanding of microbial ecosystems and how they influence human and environmental health.
Congratulations, Jean. Congratulations, Jean. I welcome you into the Australian Academy of Science Fellowship.
Microbes may be small, but they play a significant role in our lives and the environment around us.
Microorganisms control all of the major biogeochemical cycles on Earth. If microbes stopped doing their function, those major geochemical cycles would stop and life as we know it would cease.
But studying them hasn't been easy, as most of these microorganisms can't be cultivated in a lab. As a microbial ecologist and a bioinformatician, Professor Jean Tyson took a different approach. As a PhD student, he published a seminal paper on metagenomics, proving for the first time that it was possible to sequence multiple microbial genomes at once. Since then, he's become renowned for his development and application of novel approaches to understanding microbial communities.
We're basically able to take any environmental sample, we're able to shear up the DNA from those microorganisms and then reassemble them into their genomes to understand what they're doing. We can overlay transcriptomics on that tells us about what genes are turned on under different conditions, and then we can see what proteins are being produced.
His team has also developed new bioinformatic tools for exploring these complex data sets. These advances have led to many discoveries, including completely new lineages of microbial life and their activities on Earth. In one project, Professor Tyson studied the microbial communities associated with thawing permafrost. Permafrost contains a huge amount of carbon, an estimated 1,500 gigaton globally. As this frozen layer thaws, bacteria can break down the organic matter stored inside, releasing that carbon into the atmosphere as carbon dioxide and methane. Professor Tyson's team uncovered what they believe to be the most dominant methane-producing microorganism in these systems.
We've been able to characterise this organism in detail, understand its role, and actually show that it is directly linked to the isotopic ratio of methane production globally.
More recently, Professor Tyson has also turned his attention to human health, studying microbial communities inside the human body and their link to disease.
When we look at a healthy microbiome versus a dysbiotic microbiome, what we're able to see is very clear differences in the microbial species that are there. Every month, there's about 500 papers that are published linking the microbiome to various disease states.
He co-founded the company Microba, which has created diagnostic tests to help people better understand what's happening in their microbiome, and is also developing new therapies for diseases such as inflammatory bowel disease.
One of the motivations for me is always to have that impact on developing new solutions that improve people's quality of life.
Impactful contributions that have now seen him elected as a new Fellow of the Australian Academy of Science.
Professor Madeleine Van Oppen. Madeleine Van Oppen is an ecological geneticist and expert on microbial symbiosis and climate change adaptation of reef corals. Her research has revealed mechanisms by which corals adapt or acclimatise to environmental changes, including those mediated by their microbial symbionts. She has built a platform of innovative assisted evolution technologies that harness the natural adaptive mechanisms to enhance coral climate resistance, technologies critical to coral reef restoration in Australia and overseas.
Congratulations, madam. Congratulations, Madeline. I welcome you into the Australian Academy of Science Fellowship.
You're looking at what brought Professor Madeleine Van Oppen to Australia.
Coral reefs have such beauty, they're just amazing, and they're really important for the marine environment because coral reefs are home to about 30% of marine species, so they're incredibly important in that sense.
In 1997, she took up a postdoctoral position at James Cook University. The following year, the world witnessed its first global mass coral bleaching event, another stark reminder of the consequences of climate change.
When you see the damage, that is very frightening.
Professor Van Oppen started to investigate the relationship between corals and the tiny algae and bacteria that live within them, known as microbial symbionts. A relationship that we now know is vital to the coral's health and survival.
We also now understand that the microalgae that live in the coral tissues play an important role in the level of heat that the coral can cope with.
Coral bleaching occurs when intense heat waves cause corals to expel the algae living in their tissues.
The corals cannot live without these microalgae, so we often see a high rates of mortality following these intense summer heat waves that cause coral bleaching.
But Professor Van Oppen's research has shown it's possible to also harness those microalgae to help the corals cope better with heat.
So certain species of microalgae give the coral better protection against heat than other species. We're sort of playing with that. Can we make them more tolerant to heat by manipulating that relationship?
A question that has since paved the way to a whole new approach in coral restoration known as assisted evolution. The concept was first introduced in a paper Professor Van Oppen published in 2015 with her colleague and renowned coral biologist, the late Ruth Gates.
We are accelerating adaptive processes, and those are natural processes that happen in the field. There's quite a bit of evidence that this might not happen fast enough if we don't help these processes a little bit. So we're basically speeding up adaptation.
In the labs at both the University of Melbourne and Australian Institute of Marine Science, where Professor Van Oppen now works, she's seen promising results. In one experiment, her team subjected microalgae to increasing temperatures to improve their heat tolerance before reintroducing them back into the corals.
We showed that they increased coral heat tolerance, but without compromising coral growth. So we're very excited about that.
They've started small-scale field trials to see if those outcomes will also hold true in nature.
Restoring reefs with enhanced corals is going to be really important because if we restore with just the stock that is there, that has just bleached, then maybe the next heat wave we might lose it all.
But she says restoration efforts are only part of the solution.
There's a limit to what we can achieve, so the most important thing is that we deal with the emission of greenhouse gases and that we stabilise the climate.
A message she now carries forward as a fellow of the Australian Academy of Science.
Professor Xinghuo Wang. Xinghuo Wang is a world leader in materials science and chemical engineering. He has made unique and enduring contributions to the development of functional nanomaterials for solar fuel production, new generation solar cells and batteries. His seminal achievements include breakthroughs in interfacial engineering and defect passivation of perovskite quantum dots to realize stable solar cells with champion efficiency and redox intercalation mechanism discoveries to achieve scalable production of layered battery electrode materials.
Congratulations, Leandro. Congratulations, Leandro. I welcome you to the Australian Academy of Science Fellowship. Thank you.
A new understanding of materials has formed the cornerstone of every technological revolution.
From the Stone Age to the Bronze Age, Iron Age, Steel Age, now Silicon Age, maybe to the possible future Quantum Age.
As a materials scientist, Professor Xinghuo Wang studies the behaviour of materials at the molecular and atomic levels.
My research has been mainly focusing on how to design better functional material and for the renewable energy application.
That includes using nanoparticles called quantum dots to convert solar energy to electricity.
Yeah, oh, this looks beautiful.
The quantum dots developed here at the University of Queensland are more stable and can be used to generate electricity even in low light conditions.
We use the quantum dots to design the solar cell and which have the potential actually to reach very high efficiency. They need to communicate between the dots very efficiently, and in between there are a lot of chemistry.
Professor Wang's team set a new world record for efficiency in quantum dot solar cells.
The thermal evaporator on this side is already in good condition now, yeah?
Their new class of quantum dots are flexible and printable, meaning they could one day be used to power things such as cars and wearable technology.
You may say some exciting things about the science. I would say 90% of our efforts actually has, try to make the working better. That means you have a lot of failure. If you can make some good progress or find something that's different or unique, and you have the opportunity or you have the potential to make change.
But Professor Wang is not just interested in energy conversion. He knows it's just as important to create more efficient ways to store it.
The energy stored in this battery must be quite good.
His team has developed a super thin cathode coating that can significantly extend the lifespan of high-voltage lithium-ion batteries. That work has seen him awarded a prestigious 5-year Industry Laureate Fellowship by the Australian Research Council to help improve the next generation of lithium-ion batteries.
We really want to develop new energy conversion and storage solutions at a low cost and a large scale.
Professor Wang says becoming a Fellow of the Australian Academy of Science will help him advocate for greater investment in research.
The power of the science is massive, actually, to shape our future.
Professor Eric Warrant. Eric Warrant is a world leader in the field of neuroethology, comparative vision and visual ecology, as well as navigation in extremely dim light. He has elucidated the optical and neural principles that dramatically improve visual performance in nocturnal and deep-sea animals, and has also discovered the sensory mechanisms that enable nocturnal animals to navigate while homing or migrating, including the magnetic and stellar compasses of the iconic and endangered bogon moth.
Congratulations, Eric. Thank you. Congratulations, Eric. I welcome you into the Australian Academy of Science Fellowship as a corresponding member.
This was initially meant to be just a holiday home for Professor Eric Warrant.
I love the snowy mountains, ever since I was a kid.
But it turns out this would also be the perfect spot to study the 1,000-kilometre migration of Australian bogong moths.
Somehow these moths can migrate from all around southeastern Australia to a really specific destination, all heading to the same place. And this place they've never been to before.
That place is a few select caves in the Australian Alps.
Look at how they're covering the walls. It's incredible.
And up in there too, look.
Yeah.
Every year, about 4 billion moths from all over southeastern Australia arrive to summer in the cool alpine caves.
How do they know the direction to travel in? But a very other obvious second question is, how do they know when to stop?
To find out, Professor Warrant built his specialised Snowy Mountains lab.
And because we're dealing with the magnetic sense of the moth, we've made everything in the lab, including the entire lab building, out of non-magnetic materials so that there's no interference from magnetic materials.
Professor Warrant's team discovered that not only do the moths use the Earth's magnetic field as a compass, they're also able to navigate by the stars.
And that screen allows us to project the starry night sky above the moth. Then we can rotate the night sky and see if we can get a reaction from these visual neurons in the central complex of the brain as the sky rotates, and we do.
His work on bogong moths is an enduring connection to Australian science, but as a professor of zoology at Lund University in Sweden, he's renowned internationally for his research on dim light vision and navigation. He's studied the visual and navigational abilities of nocturnal and deep-sea animals.
Extraordinarily good vision can be found even in rather small animals like insects. So nocturnal moths, nocturnal beetles, we've found have phenomenal night vision, not only because of this very sensitive type of compound eye which they possess, but also because of the specialised types of neural tricks that they use in the brain.
Neural tricks that have caught the attention of companies.
Toyota approached us and was very keen in developing camera systems that they could mount in cars for use at night to allow drivers to detect animals on the road and even pedestrians on the road.
Professor Warrant did his PhD at the Australian National University before taking up a postdoctoral fellowship in Sweden, where he's lived since.
I usually say I have two homes, and that's very much because I could never permanently leave Australia. It would be impossible for me to do that.
He says being elected as a corresponding member of the Australian Academy of Science is a great honour.
It's especially heartwarming because it's the academy of the country where I was born and was educated. And I find that election to be something that is more special than being elected to any other academy anywhere else.
Professor Willy Zanupol. Willy Zanupol is a leader in experimental software systems. His groundbreaking work on distributed shared memory has made fundamental contributions to a variety of subjects, including multicast communication, fault tolerance, internet servers, graph processing, virtualization, and storage systems. The influence of his work is evidenced by its large number of citations and its translation into production systems.
Congratulations, Willy. Congratulations, Willy. I welcome you to the Australian Academy of Science Fellowship.
Professor Willy Zwanapul is a world leader in computer systems research. His pioneering work in operating systems and distributed computing underpins much of today's cloud computing, work that began with his PhD at Stanford University in the 1980s.
The PhD was based on the internet. It was to see if we could, this is going to sound terribly mundane now, but in those days it was a big leap forward. It was to see whether we could access files over the network.
At the time, local disk storage had a long list of reliability issues, and creating backups was cumbersome and time-consuming.
Most notably that they would crash quite often.
His thesis demonstrated the advantages of a networked storage system, which was adopted first by Sun Microsystems and later across the industry. Professor Zwanapul says witnessing the commercialization process greatly influenced his outlook on research.
It evolved quite rapidly, so Sun Microsystems, which was then still on campus, became a company. So I think there's sort of a very productive give and take between the industry and the academia, at least in my field.
Over the next few years, while an assistant professor at Rice, Professor Zwanapul turned his attention to the idea of sharing computer power over a network.
In the network, most of the time, most of the machines are idle. There's not much going on. And so there was this idea of how could we coalesce all this computing power to work on really big computations.
His work on distributed shared memory and the resulting trademarks system overcame the major obstacles faced by earlier systems, enabling multiple computers to work together on a problem in parallel. Professor Zwanapul spent more than 20 years in the United States before being enticed to Switzerland to lead the School of Computer and Communication Sciences at EPFL. During his tenure there, he led a paradigm shift that saw the university rise from relative obscurity to a world-renowned institution in the field.
The thing that gets me up in the morning is to work with young people and help them in, sort of guiding their career.
At the heart of his academic leadership has been a commitment to supporting the growth and success of the next generation of researchers. A commitment he brought with him to the University of Sydney in 2018 when he became Dean of Engineering, a position he held until 2024.
Their first role should be to produce the people that will make technology advance. That, to me, is number one, and perhaps number 1, 2, and 3 of what the university should do.
His outstanding dedication to the advancement and promotion of computer science has now seen Professor Zwanapul elected a new Fellow of the Australian Academy of Science.
We'd now like to invite to the stage some fellows elected in previous years who have not yet signed the charter book. And firstly, I'd like to call on Tim Brodribb. Tim Brodribb is an evolutionary biologist. His work explains why leaves die during drought, why some plant species are more vulnerable to water stress than others, and how flowering plants evolve the capacity for rapid photosynthesis and growth. Many of these discoveries have been accomplished through technical innovation, yielding new tools that are currently used worldwide to assess plant vulnerability to drought. Tim?
Congratulations, Tim. Congratulations, Tim. I welcome you to the Australian Academy of Science Fellowship.
Plants fight a daily battle for survival.
The water in the soil is not just liquid water, it's liquid water that's pulling away from the plant. So the plant has to exert this huge amount of tension on that water column to drag that water up out of the soil and up to the leaves. And the plant again sort of has to fight against the atmosphere which is dragging water out of the plant.
Professor Tim Brodribb has spent much of his career probing those interactions. He studies the vascular system, a series of pipes responsible for distributing water through the plant. This system involves a constant tug-of-war between the soil, which holds water, and the plant, which tries to pull that water away. But it's a delicate balance. Too much tension to the continuous threads of water inside the pipe, and they will snap. In the past decade, Professor Brodribb has worked on visualising those processes of tension and cavitation. His team has developed a way of looking into plants in real time using tiny cameras fitted on their leaves. They were the first scientists in the world to capture the moment a leaf dies, a lightning-like bolt zapping along its veins. They've now installed these cameras not just here in Tasmania, but also overseas, including on sensitive ferns and redwood trees in California.
I have sensors on trees that I watch with my mobile phone, and I spend half the day just watching my mobile phone because I'm curious to see what's going on. To be able to communicate with plants in that way is just really pretty thrilling.
It's also critical work to better understand how we can make our forests more resilient to climate change. Professor Brodribb says the ability to monitor the impact of worsening heat and drought on different tree species will play a key role in saving them.
It would be a bad look if 2 degrees of climate change equals half of the world's forest suddenly dying from acute evaporative stress. And if we don't have the science to tell us that that's a really, really critical limit that we shouldn't be reaching, then we're just floating around in the daydream. The way that we're putting limits on climate extremes at the moment are not based on plant behaviour. I mean, they should be uniquely based on plant behaviour because they're the things that sustain everything on Earth.
Professor Brodribb has always had a love for plants, and studying them delights him to this day.
I just have an incredible respect for plants, I think, and an incredible curiosity that in everything we do, we get this little step and an interesting insight into something, but it just opens up a whole lot of new questions, just really basic questions that we don't understand, and questions that we really must understand.
His work, shedding light on some of those fundamental questions, has now seen him elected a Fellow of the Australian Academy of Science.
I sort of feel the classic imposter syndrome perhaps, but I'm really excited to join the Academy and to be a part of this amazing community of people and many members who I know and have huge respect for.
Professor Emma Johnston. Emma Johnston is a leader in the field of marine ecology. She's also a highly influential leader, advocate and voice for Australian science. Combined with her extensive track record of science communication and outreach, Johnston is an exceptional role model and science champion.
Congratulations, Emma. Congratulations, Simon. I welcome you to the Australian Academy of Science Fellowship.
Professor Emma Johnston investigates how humans interact with marine ecosystems.
My work has primarily been in coastal marine ecosystems, which is where humans have the most impact because we tend to love living near the coast and we use coastal ecosystems for all of our transport and our recreation, and we get a lot of food from them.
Global shipping means that none of those ecosystems work in isolation.
We actually have this mechanism for transporting species from one country to the other on ships, and that could be either in the ballast water or connected to the hull of a ship.
Some of Professor Johnston's research included laboratory and field experiments, which identified new drivers of invasive species success and clearly demonstrated their ability to rapidly evolve in new environments.
In many of the ports and harbours that I work in, there are more than 300 different species that you can see of marine organisms that are introduced. They're non-native.
Understanding this paves the way for better management strategies, such as dredging up contaminated sediments to help improve outcomes for native species. Professor Johnston believes in translating research into action, particularly evidence-based public policy. She was chief author of the Australian State of the Environment report in 2021.
We are at the forefront of climate impacts, and without rapid, drastic, urgent, unimaginably fast carbon emissions reductions, we stand to lose a lot of those precious ecosystems.
Professor Johnston is Deputy Vice-Chancellor Research at the University of Sydney. She's become a highly influential voice for science, having served as President of Science and Technology Australia and Dean of Science at UNSW. She's determined to make the sector more open and inclusive, but says that requires a whole-of-society approach.
It's the schoolyard where they're encouraged to do particular activities. It's their role models that they see as they go through high school and the choices in subject that they're encouraged to make. And then in their actual undergraduate and their career, it's about building in the flexibility so that people can have a diverse array of different cultural backgrounds, but also lifestyle choices and come in and out of scientific careers.
Research funding is another key priority for Professor Johnston. She says government and industry investment is proportionately much lower in Australia than in many advanced nations.
We have incredible minds and incredible fundamental research being done, but sometimes we cannot follow through because the investment in the translational work, in the innovation, in the bringing the product to market, the money is just not there.
Professor Johnston sees becoming a Fellow of the Australian Academy of Science as not just an honour, but an opportunity. She wants to work with other Fellows in promoting the value of science and guaranteeing a vibrant sector.
Science is a method that has enormous potential for benefits for the whole of society.
Professor Anne Dell. Anne Dell was amongst the first investigators worldwide to successfully apply advanced mass spectrometric techniques to carbohydrate-containing biopolymers for solving structural problems in glycobiology and facilitating the development of new drugs and vaccines. Her research has been recognised by election to the Fellowship of the Royal Society in 2002 and the Academy of Medical Sciences in 2008. Anne?
Congratulations, Anne. Congratulations, Anne. I welcome you into the Australian Academy of Science Fellowship as a corresponding member.
What role do sugars play in human health and disease, and even the beginning of life? Professor Anne Dell has made significant headway on those questions, having spent her career characterizing the chain-like structures of sugar molecules known as glycans.
Glycans are important because they are part of molecules that sit on cell surfaces. They make what we call a glycan or sugar coat, and that sugar coat is really important when cells communicate with each other.
Scientists have long known that glycans are important. Even small changes to them can have a big impact on processes in the human body, such as metabolism and immune responses. But studying them has proven tricky, primarily because of a lack of tools. Professor Dell helped to overcome that by using an analytical device that measures molecular weight, called a mass spectrometer.
The mass spectrometry techniques that I pioneered were combining microchemical methods with the emerging mass spectrometry technology that was being designed to analyze biopolymers, which prior to my PhD could not be analyzed by the existing techniques.
The first biopolymer mass spectrometry lab in the world was set up by her mentor, Professor Howard Morris, here at London's Imperial College.
Very soon after that, scientists from around the world knocked on our doors at Imperial and asked us to help analyze the precious samples that they were carrying in their pockets, many of which they had spent months, if not years, isolating and purifying from biological materials.
During one such collaboration, Professor Dell and her colleagues discovered a novel glycan, silyl Lewis X. The glycan, consisting of 4 sugars linked together, has proven to be very active on cancer cells. That's made silyl Lewis X a key target in research in how to fight tumors. But it turns out this particular glycan has another big job.
Well, in more recent research, we made the very exciting discovery that silyl Lewis X is also the sperm receptor on the human egg.
When sperm arrives at the egg, it has to recognize it as an egg before it can fertilize it. This glycan is key to that happening.
So at the very beginning of life, at conception, there is a sugar or glycan-lectin interaction where the glycan is silyl Lewis X.
Research now underway at Imperial College is investigating what role silyl Lewis X plays in preterm birth. Professor Dell recently stepped down as head of the university's Department of Life Sciences. She says she's looking forward to spending more time traveling, including to Australia, where she was born. She says it's an enormous honour to have her life's work recognised as a corresponding member of the Australian Academy of Science.
Professor Peter Cork. Peter Cork has made significant contributions to the field of robotic vision. In particular, he has developed a solid foundation for the theory and practice of visual surveying. He is the founding director of the ARC Centre of Excellence for Robotic Vision. In addition, he has pioneered the practical use of his theory in aerial, marine and land robotic systems for applications as diverse as mining and environmental monitoring.
Congratulations, Peter. Congratulations, Peter. I welcome you into the Australian Academy of Science Fellowship.
Robots are often used to increase productivity and profit, as well as to save time and improve safety.
That might be underwater, surveying large parts of coral reefs. It's dangerous to have human beings under the water for long periods of time. Robots could do that job beautifully, and no one gets harmed or injured.
Professor Peter Cork is a renowned robotics expert who has pioneered aerial, marine and land robotic systems for mining and environmental monitoring.
The thing that I'm most proud of in the area of robotics is something that we take for granted, and that's hand-eye coordination. And it's particularly challenging if the object you're trying to reach is moving.
Professor Cork says the ultimate goal is to program robots to see.
Being able to see is something we take utterly for granted, but it's a capability that robots don't have, and it absolutely hinders them in being able to do the sorts of things that we expect robots to do.
He's also involved in researching deep learning.
So instead of writing a program that can say, this is what a human being looks like, we write a program that can learn, give it lots of examples of human beings, and say every time this is a human being, this is not a human being. And it will learn some generalisation, some representation of what a human being is.
Global research is underway to coordinate discoveries and design the robots of the future.
There are people working on vision, people working on fingertips, people working on legs, all of these different components of the robot system being worked on simultaneously, and it leads to a huge body of work which is going to give us in the future very, very capable and competent robots.
Professor Ben Andrews. Ben Andrews is a leading international researcher in differential geometry and related partial differential equations, and is particularly famous for his work on geometric evolutions. Among many notable achievements is his resolution of Fiery's conjecture in 1999 on the shape of rolling stones, and the recent resolution of one of the most celebrated open problems in mathematics, the fundamental gap conjecture for the eigenvalues of the Laplacian.
Congratulations, Ben. Congratulations, Ben. I welcome you into the Australian Academy of Science Fellowship. Obviously, you're wondering why Ben doesn't have a video. It turns out that the Ben was waiting for the perfect prime number to be inducted into the Academy. So he was elected in 2013. He's been inducted into the 2024, 11 years afterwards. So that's why at that time we were not creating videos for our new fellows. It's a new phenomenon which we have started. That's why, sorry, Ben, we don't have a video for you. Thank you, Bob and Malcolm, and congratulations and welcome to the fellows elected in 2024. As I stand here today, officially welcome you to the fellowship, I cast my mind back to 2005 when I was elected to the Australian Academy of Science and took my own solemn promise to promote, declare, and disseminate scientific knowledge and to serve the Academy according to its charter and bylaws. As academicians, we have a unique responsibility to uphold standards of scientific excellence, to be inclusive, to shepherd the next generation, to provide scientific leadership, and use this platform to lift others. Our collective challenge is to harness the knowledge you generate and utilize it for public good. The Academy's independence means we have no skin in the game, we are impartial, and are not beholden to any single institution. Our only interest is to ensure your expert knowledge can shape the actions and direction of our nation and its people so everyone can benefit. The current times demand that we bring to bear the full power of science and technology to meet our national ambitions. And I know that if we work together, we can rise to that challenge advance our nation and our globe. To encourage you to engage with the Academy to pursue this challenge. There are many ways in which you can become involved in the life of the Academy, some more time-consuming than others. In my address to all fellows this afternoon, I will elaborate on this, and you'll have the opportunity to familiarize yourself with our work and impact, as well as meet members of Council with whom I encourage you to liaise with. I acknowledge that many of you already made significant contributions in the past few months since your election, and some of you probably a long time back. And for that, I thank you, and I look forward to continuing to work with you for many years to come. I conclude by saying the Academy is its fellows. You are the beating heart of this great institution. I know I speak on behalf of my colleagues on the Academy's Council and the Academy staff when I say that our doors are always open. We want to work with you so that the Academy can contribute to advanced science. Again, my heartfelt congratulations on your election and welcome to our fellowship. It's wonderful you could be joined by family and friends. I now invite Anna Maria Arabia to close this afternoon's ceremony. Thank you.
Thank you, Fellows. May I also extend my congratulations on behalf of all of the staff of the Australian Academy of Science on your election to our fellowship. Ian Potter House is just across the car park, and they're available for use at any time, even when you are in Canberra on non-academy business. Our facilities, including a Fellows Lounge, meeting facilities, AV facilities, a recording studio, a place to rest your bag, a place to connect. The Academy staff are always available and ready to assist you. To draw this admission ceremony to a close, may I ask that all 2024 Fellows go to the main foyer for a group photo. There will be plenty of time after that to come back and photograph the wonderful charter book that you've just signed, adding your name to other great scientists, including Sir David Attenborough, Dorothy Hill, Suzanne Corey, Jagadish, many other scientists who have signed the book. I remind you that all newly admitted Fellows need to have their official portrait taken. There is also some afternoon tea being served in the Jaeger room and in the Dorothy Hill room, as well as the marquee outside. Please do take advantage of the lovely space you're in. We'll see you back in the theatre at 4:15 rather than 4 o'clock for the President's address and for the launch of the Academy's Innovate Reconciliation Action Plan. Thank you very much.
Taking medicine in the future could be as simple as eating a salad.
The example that we're actually doing at the moment is to have a multiple sclerosis drug in tomatoes so that people with chronic multiple sclerosis could maybe have their Greek salad and have some level of medical protection from the disease.
These drugs are based on molecules known as peptides. They are smaller than proteins and carry out important biological functions such as regulating other molecules, and some even act as hormones. Several peptides have pharmaceutical potential, including as potent painkillers. But because most break down if they're taken by mouth, Professor David Craik and his colleagues have engineered them into more stable circular peptides called cyclotides. Cyclotides are naturally found in some plants, so Professor Craik's team can embed them into edible plants such as tomatoes.
One of the issues with current painkillers is that often they don't work. And then we also know with opioids, there are horrible addiction problems in the so-called opioid crisis. This is a different class of molecule that doesn't have those addictive effects.
Professor Craik says the potential applications extend beyond chronic pain and disease to areas including agriculture.
We could be creating a new biofarming industry in Australia where farmers could be, rather than growing a crop that might be worth $100 a tonne, it could be worth $100,000 a tonne by producing a high-value medicine in those plants.
The innovation isn't far from becoming reality, with the first crops potentially commercialised in the next few years.
Obviously there's lots of regulatory and social and ethical issues that we would need to address in doing this, but I think it does open up a new world for combining agriculture and medicine and delivering medicines in a novel way.
So why does science matter?
Science is what drives our advances in medicine and what gives us new treatments, new vaccines, new techniques in surgery. All of those need to be evaluated, developed, many of which come from crazy ideas, from fundamental science. Other advances come from astute observations by clinicians. So science is so critical to advances in medicine.
And I think this was one of the most beautiful examples of science in action, where every biomedical researcher on this planet over the last 3 or 4 or 5 years has read a paper, and many have written papers, about a virus that they had never heard of. I find this mind-blowing to what degree we have, as a community of scientists, come and tackled this new threat.
I'm motivated to research antibiotic pharmacology because I see the human cost of deadly bacterial infections. I think science is fundamental to discover the mechanisms, and then we can use the mechanistic findings to develop novel therapeutics. This kind of translation is not come out from a simple thinking. It come up with a long-term research. So that's why science is so critical.
Science is just the way that we learn to think. You know, we learn to gather data, interpret data, make decisions based on data. And over the last few years, it's been really surprising and horrifying, actually, to see that not everybody thinks that way.
We live in a world where we need to make informed decisions about how we build our houses so that they use less energy, so that they're more comfortable, healthier, better places to be. Even just down to the simple decision of, you know, whether or not you put insulation into your walls or into your roof, science can tell you the answer, which is the better of the two things to do.
You know, if you think about thousands of years into the future, without science, we don't have a great hope of survival, I don't think. You know, science will underpin all of that. A better future for our kids, you know, that's going to come from science.
Kathleen Folbig has always insisted she never killed her 4 young children.
Did you kill Caleb?
No!
Did you kill...
No!
Did you kill Sarah?
No.
Did you kill Laura?
That's a position she maintained while exhausting all of her legal appeals and during 2 subsequent judicial inquiries. 20 years into a 25-year prison sentence, the justice system has now accepted there is reasonable doubt about her guilt.
I think the nation will look at the case of Kathleen Folbig as a major miscarriage of justice.
Ms. Folbig was convicted of smothering her babies Caleb, Patrick, Sarah and Laura. The 4 died on separate occasions between 1989 and 1999. But at the time of her conviction in 2003, there was no way of knowing if gene mutations played a role in any of the deaths.
In fact, it was only in 2003 that the first draft of the human genome sequence was completed.
Now, since then, it has become possible to sequence whole human genomes and exomes in a much faster and affordable way.
That's Professor Carola Vinuesa, who analysed sequences from DNA extracted from old heel prick samples of 2 of the Folbig children.
She was concerned that the analysis of that genetic information had not been fully heard during the first inquiry into the convictions of Kathleen Folbig. And it was those concerns and her resolute understanding of her science and her scientific integrity that in some ways didn't let her sleep at night.
Professor Vinuesa took those concerns to the Australian Academy of Science, where she is a fellow.
The more we looked at it, the more we too became concerned around the way science had not been heard in that first inquiry.
The science was complex. Research undertaken by Professor Vinuesa and complemented by independent experts around the world showed that Ms. Folbig and her 2 daughters possessed a rare gene mutation known as CALM2G114R,
which encodes for calmodulin. Calmodulin mutations typically are associated with cardiac arrhythmias that can cause sudden unexpected death in children and adults, both while awake and while asleep.
The second inquiry, which was instigated at the urging of prominent scientists, including 3 Nobel laureates, also heard medical evidence about epilepsy and respiratory problems, which could help explain the deaths of Ms. Folbig's 2 sons, Patrick and Caleb.
There is a reasonable doubt as to Ms. Folbig's guilt.
Retired Chief Justice Tom Bathurst KC accepted that the weight of the evidence cast reasonable doubt about Ms. Folbig's guilt, recommending her pardon. A key difference in the way the second inquiry considered medical evidence was the role played by the Australian Academy of Science.
I think now that with this new evidence, it's beyond reasonable doubt.
This is a clear indication that the independent scientific advice has an important role to play in the justice system.
The Australian Academy of Science was granted leave by the inquiry to act as an independent scientific advisor to it. I think unique to this case, and the role the Academy's been able to facilitate, is that the inquiry's been able to hear from a range of experts from the right subdisciplines to give a complete picture of the sort of science that is complex, and some of it's still emerging, that's been able to shed light on the possible causes of death of the 4 Folbig children.
The Academy believes the case serves as a dramatic example of the need for a more formal mechanism to re-examine cases after appeals have been exhausted when new science is revealed.
Science is always emerging. Nothing is going to stop the advancement of knowledge, nor would we want to do that. It would be an enormous missed opportunity if we couldn't bring about some of the law reform that's needed so that science can more effectively inform the justice system.
Taking medicine in the future could be as simple as eating a salad.
The example that we're actually doing at the moment is to have a multiple sclerosis drug in tomatoes so that people with chronic multiple sclerosis could maybe have their Greek salad and have some level of medical protection from the disease.
These drugs are based on molecules known as peptides. They are smaller than proteins and carry out important biological functions such as regulating other molecules, and some even act as hormones. Several peptides have pharmaceutical potential, including as potent painkillers. But because most break down if they're taken by mouth, Professor David Craik and his colleagues have engineered them into more stable circular peptides called cyclotides. Cyclotides are naturally found in some plants, so Professor Craik's team can embed them into edible plants such as tomatoes.
One of the issues with current painkillers is that often they don't work, and then we also know with opioids there are horrible addiction problems in the so-called opioid crisis. This is a different class of molecule that doesn't have those addictive effects.
Professor Craik says the potential applications extend beyond chronic pain and disease to areas including agriculture.
We could be creating a new biofarming industry in Australia where farmers could be rather than growing a crop that might be worth $100 a tonne, it could be worth $100,000 a tonne by producing a high-value medicine in those plants.
The innovation isn't far from becoming reality, with the first crops potentially commercialised in the next few years.
Obviously, there's lots of regulatory and social and ethical issues that we would need to address in doing this, but I think it does open up a new world for combining agriculture and medicine and delivering medicines in a novel way.
As the sun rises over Melbourne, you'll often find Professor David Commander on a kayak.
It's just such an amazing way to connect with nature, and you cannot beat the serenity and the beauty of sitting on your kayak in the morning. Sometimes you have dolphins coming up and you catch a fish.
The biochemist and cell biologist started kayak fishing during Melbourne's COVID-19 lockdown and soon realised it helps focus his mind.
It does help my work because it gives me that balance and in my mindset that I really need to, not completely get eaten up by the stress.
That stress comes from trying to unravel the mysteries of the ubiquitin system. Ubiquitin is a tiny protein that attaches itself to bigger proteins to modify the way they function or interact within our cells.
If you attach one ubiquitin to a protein, that's not so much the big deal. What is a big deal is the idea that ubiquitin can now be attaching to itself, and we build what's called ubiquitin chains.
The fate of the targeted protein can vary depending on the structure of these chains, and here's why that matters.
What we're describing here is a possibility to turn our basic research insights into potential for new drug discovery.
The ubiquitin system plays a critical role in a wide range of cellular processes, and problems with the system can cause a variety of diseases, including cancer. One function of ubiquitin ensures that broken proteins and damaged organelles such as mitochondria are removed from circulation by flagging them for destruction. When this process malfunctions, obsolete and damaged proteins or mitochondria accumulate, which can cause neurodegenerative diseases such as Parkinson's. Professor Commander has advanced our understanding of that relationship through fundamental research into proteins called Parkin and PINK1.
PINK1 is a protein called ubiquitin kinase. Kinases have a well-studied function in taking a tiny chemical tag and attaching a chemical phosphate group to a protein of interest. What was a shock to the entire field of ubiquitin researchers, including myself, was when we and others identified that PINK1 is a kinase that attaches the phosphate group not to a protein but actually to ubiquitin as a protein. So now we're talking about the modifications of a modifier.
PINK1 modifies the ubiquitin attached to damaged proteins on the outer membrane of mitochondria. Modified ubiquitin attracts Parkin, which attaches more ubiquitin to the surrounding proteins. A heavily coated surface initiates the disposal of the entire organelle to nearby proteins, creating a layer of modified ubiquitin that initiates their orderly removal. Importantly, failures in either Parkin or PINK1 cause certain cases of Parkinson's, particularly for people who develop the condition before the age of 50. The race is now on to work out how to activate PINK1 or Parkin to treat the condition. Professor Commander describes that as a perfect example of how fundamental and translational research can work hand in hand, and something his team does every day here at WEHI in Melbourne. A critical mission that's now seen him elected a Fellow of the Australian Academy of Science.
It means everything. I feel so privileged to be part of this learned society.
There's so many people working in science doing great work.
We use research innovations to solve the world's biggest problems.
The link between science and a happy and healthy society is as important as it's ever been.
That's why I do what I do, because I want to see my science make a difference.
And I think that this is a great opportunity for recognition of that work.
To now be awarded for my contribution, for me, that's just truly incredible.
I highly encourage anyone to apply for these honorific awards. It's a good way to get our science out there.
Receiving this medal is a real honour.
It means a tremendous amount to be acknowledged by the Australian scientific community, because it means other people have seen there's a reason I do what I do and they want me to keep doing it.
I would absolutely encourage anybody who is passionate about their science to apply for one of these Australian Academy of Science honorifics.
Our immune system plays a key role in protecting us from microbial invaders, but its overactivity can lead to complications.
The capacity to respond to a pathogen is just as critical as the capacity to turn off the response when it's no longer required. When an immune response is not turned off, then the dysregulated immunity and the ensuing inflammation can cause a lot more problems than those caused by the actual virus or the pathogen itself.
Professor Maria Pia degli Esposti studies that delicate balance. As an immunologist, she has made pioneering contributions to understanding natural killer cells and their role in the immune system. These white blood cells have long been recognized as a first line of immune defense, the early assassins that promptly kill infected cells.
We now know that NK cells have functions that are much more sophisticated than we previously thought. And our work has contributed to a body of knowledge that identify the regulatory function of NK cells. These functions are well beyond their capacity to attack and kill, and they extend to their ability to actively regulate immune responses and to adjust various facets of the immune system to ensure that the pathogen, the threats, is effectively handled while also turning off the initial response once the issue has been addressed.
Her team developed innovative preclinical disease models based on infection with a prevalent virus known as cytomegalovirus, or CMV, to better understand how the immune system functions. CMV is unusual in that once it infects a person, it is never completely eliminated. In most cases, the immune system seemingly manages the virus so that it doesn't cause symptoms. But when the immune system is compromised, for instance in transplant patients, the virus can reactivate and cause significant illness.
And it is in those clinical situations where we need to be able to intervene and take action.
Professor degli Esposti and her team found that using antibodies, specifically ones that matched the particular strain of infecting CMV virus, offered the best protection against CMV reactivation. That discovery won them the Eureka Prize for Scientific Research in 2019. It's impactful research that speaks to why Professor degli Esposti fell in love with science and immunology in the first place. Growing up in Italy, she had initially wanted to be a pediatrician, but she soon realized her passion lay elsewhere.
What I was much more excited about was the potential to understand the origins of diseases, as this would really enable us to alter their trajectory. And the best way to achieve this is by fundamental research, because that allows us to understand and ultimately target the processes that are at play.
Her critical work has now seen her elected a Fellow of the Australian Academy of Science. She says she accepts the honour on behalf of her entire team.
Now to growing concerns about the deadly coronavirus.
Can the spread of this highly infectious disease be contained?
As word spread of a potential global health emergency, Professor Sharon Lewin was up early every day. She'd jog while listening to COVID-19 updates from the New England Journal of Medicine.
I actually do really like physical exercise or ways to be completely distracted from all the other issues that cloud your mind in a busy work week.
And as director of the Peter Doherty Institute for Infection and Immunity, there was plenty on her mind. A joint venture of the University of Melbourne and the Royal Melbourne Hospital, the Doherty played a key role in formulating Australia's pandemic response. The Institute's scientists were the first outside of China to develop a lab-grown version of the virus.
And we made a decision very rapidly to share it globally, and the virus was available to over 70 different laboratories around the world.
The Institute also created the first COVID PCR test in Australia and diagnosed the first patient.
Professor Sharon Lewin is the Doherty Institute's director. Sharon Lewin.
Professor Lewin became a familiar face on Australian television, sharing findings from COVID modelling and updates on vaccine development.
I never thought to myself, we're under immense pressure here, we must deliver. I was very conscious that science, public health, it's a real group effort.
She says there are plenty of lessons from the pandemic, including the vital need to effectively communicate science to mainstream audiences.
Science plays an incredibly important role, but science is only useful if it's done in partnership with the community and also our political leadership.
And she believes the world's leaders must take steps to ensure low and middle income countries aren't left behind in the next global health crisis.
So we have to make sure we've got a system that can produce 7 billion vaccines, or vaccines for everyone, very quickly, and there's equitable distribution.
Fighting to improve health outcomes globally is a day-to-day job for Professor Lewin. She spent more than 2 decades unlocking the secrets of HIV and is now president of the International AIDS Society. Professor Lewin was at the epicenter of HIV science in the late 1990s, undertaking a postdoctoral fellowship alongside the famed AIDS researcher David Ho in New York. He had just become Time magazine's Man of the Year after pioneering the use of combination antiretroviral therapy, which transformed HIV from a terminal disease into a chronic disease. There was genuine hope it would pave the way for a cure.
And within 3 weeks of me arriving in New York in December 97, HIV latency was discovered.
HIV latency is when the virus goes into hiding within the body's cells, preventing its eradication. Her research has focused on not only finding where it hides, but coaxing it out so it can be destroyed.
About 1 in a million cells carries a virus that we actually care about. So you've got to sift through 1 million cells to find that infected cell. But we've now got ways to do it.
More a marathon than a sprint, but Professor Lewin, now proudly a Fellow of the Australian Academy of Science, remains determined to reach the finish line.
We now have widespread access to treatment. Treatment's relatively simple. The next big challenge is finding a cure.
This farm near the New South Wales city of Wagga Wagga is a long way from war-torn Ukraine.
These varieties here, they're all spring canola, and in Australia we mostly grow spring canola.
But it's familiar territory for Marina Tsu and Svetlana Omulchuk.
Big surprise for me that we have a lot of in common things in agriculture in Ukraine and in Australia. It's really was like, Australia was something very fine, different from us, and when we are here we look and wow, we have the same thing. So we are very interested in
Marina and Svetlana research canola at the Ukrainian Scientific Institute of Plant Breeding in Kyiv.
For both countries, it's an important export crop, and for the globe, countries like Australia and Ukraine that basically provide the world trade of that commodity, what happens in those countries, in both of our countries, is important to world trade.
And it's what's happening in Ukraine right now that has brought these young scientists to Australia. When war broke out, the US-based Breakthrough Prize Foundation teamed up with the Australian Academy of Science to ensure Ukrainian researchers could continue their work.
Ukraine is actually a powerhouse, particularly in genetics and in plant science, so it's important for us to keep those links open. But whatever helps Ukraine also helps Australian science and international science.
Like everything, the work of scientists has been seriously interrupted by the conflict.
It's impacted all spheres of our life, not only science, because you always are under stress. You're always worried about safety of yourself, of your family, your friends, and it's hard to plan your experiments and projects when you think just how to survive.
Але життя змінилося, але ми якось продовжуємо працювати. Дуже вдячні всім, хто нас підтримує, всім країнам, також Австралії, які надають допомогу.
One of their hosts in Australia was CSIRO agricultural scientist and fellow of the Australian Academy of Science, Dr. John Kierkegaard.
They told me sometimes they're travelling along roads which have mines. It takes them hours, 10 or more hours to travel safely to see some of their field experiments. Quite challenging, and I found them remarkable, but in their pragmatism and determination to sort of keep the work going.
Dr. Kierkegaard had hoped other Ukrainian scientists would join the Australian trip, but fighting-aged men have been barred from leaving Ukraine. Australia has made sure they still have support, including remote access to scientific infrastructure, including supercomputers, microscopes and telescopes.
Science and technology has clearly underpinned the growth of canola as a crop in both countries. The only way to sort of stay ahead of that game is that scientists around the world collaborate, share ideas, share information, and that's what conferences like this and exchanges like this can really promote.
Ukraine knows science will play a major role in rebuilding the country after the war.
And it's all about the human capital. We can rebuild buildings, right? It's not a problem, but we need to keep our human capital. It's our competitive advantage, the talent that we have, and we need to nurture the talent. We need to help the talent to grow, develop, and become better. And I think this will be of huge contribution to Ukraine's reconstruction eventually.
The support of Australia and Australian people are really important for us and for Ukraine now, because I know it's important to feel that we are not alone in this need. And we really appreciate all that you do for us.
For stargazers, this is the ultimate light show. Auroras are normally only seen in polar latitudes, which is why they're often labelled the northern and southern lights. But this photo was taken in the central New South Wales city of Dubbo. Auroras happen when charged particles from the sun interact with Earth's magnetic field.
Fellows, colleagues, honoured guests, good afternoon. Congratulations and a warm welcome to our newly admitted fellows. Our fellows are the brain trust of our nation. You are among the nation's most distinguished scientists. Since the Academy was established 70 years ago, 982 fellows have been elected to it. Our fellowship currently stands strong at 614. Aside from the scientific work we do, our fellows contribute their expertise to the Academy so science can be heard. And so your knowledge can inform the actions of our decision makers, whether they be in our parliaments, boardrooms, courtrooms, in our classrooms, and in our homes. The 19 years since I was elected to the Academy have flown by. But I still vividly recall the deep satisfaction at being recognized by my peers for my contributions to science. I imagine you feel the same today. I was one of 15 fellows elected in 2005. Among them was microbiologist Professor Ruth Hall. She was the only woman elected that year. How times have changed. for the better. Election to the Academy remains a fair and rigorous process. Today, we have better ways to ensure we remove barriers to election experienced by underrepresented groups. We continually review these processes, whether they apply to the election of fellows or to awardees, so that we can assess the diverse contributions of candidates. We know that diversity is more than gender diversity, and there is still much work to be done, especially around recognizing achievement across Indigenous scholars. I will come back to this later. Since our inception, we have had a steadfast commitment to building a fellowship that reflects the contemporary balance of disciplines and the breadth of the scientific community. This commitment is reflected in the Academy's new strategic plan, which I will also talk more about later. As a National Academy, we have a responsibility to model, promote, and influence best practice in the conduct of science, not just within our fellowship, but across the science sector in Australia. It is our collective role to set standards for science and to pursue scientific excellence in all that we do. In listening to fellows' presentations, you have already had a taste of the extraordinary scientific talent we have in our nation. I spend much of my time looking at nanostructures and observing the world at a different scale. So the opportunity to sit in this grand hall and hear about the wonders of our universe, how viruses multiply, novel ways to create clean energy, and the complexity and importance of the soil beneath our feet is truly mind-blowing. On Thursday, we'll hear from our awardees, including emerging scientific leaders. I have no doubt I'll be inspired again and reminded of why it is critically important for all of us to work to remove barriers that prevent the world benefiting from their creativity and discoveries. Science at the Shine Dome is one, if not the only, conference in Australia where you can listen to Australia's most distinguished scientists share their expertise across every discipline. Scientists of all ages and backgrounds, people passionate about shifting the frontiers of knowledge, come together to share, network, collaborate, and to inspire each other. Another reason I love Science at the Shine Dome is this place and all it stands for. The Shine Dome is the home of the Australian science, a bold, big concept. At its opening, it was described as unconventional and futuristic. It still is. To this day, the Shine Dome remains an architecturally significant building made greater by its occupants. Following the Shine Dome's refurbishment in 2001, then Prime Minister John Howard said of the building, in quote, "All of you will see it as a fitting physical reminder of the centrality of science and all that goes with it in the national life of our country," unquote. It's a fitting reminder of the importance of science. It has inspired many. It continues to inspire me and many others. More recently, newly elected fellow, Professor Arthur Georges, nicely captured the inspiration that the Shine Dome evokes for many of us. He said, quote, "My father realized that I had an interest in science. He was a politician, and he brought me to Canberra often, and one year he took me across to show me the Shine Dome. He explained that it was a center of academic life for scientists. As is anyone who sees it for the first time, I was really impressed. But back then, in 1968, it was really out of ordinary. That memory stayed with me forever, unquote. So may more politicians bring their children here, and I invite you to do the same. As the Academy celebrates its 70th anniversary, the majestic Shrine Dome celebrates its 65th. Discussions to establish an academy began in 1951 at a meeting in Canberra to talk about the future of science and technology in Australia. Sir Mark Oliphant said, when seeking to convince others of the possibility of an Australian academy, quote, if science for its own sake was to have coherence, there must be a means whereby that coherence could be expressed, unquote. Because of the great talent and growing capability of Australian scientists, Oliphant was inspired to establish a new scientific identity closer to home. As so he did. Oliphant became the Academy's first president. It truly is a privilege to be serving you as the Academy's 20th president, building on the work of those who have come before me. The 1950s were a time of post-war reconstruction when H.C. Coombs, a public servant and first governor of the Reserve Bank, spoke about the social reconstruction. It was a time when Australia built structures that recognized the value of evidence and institutions necessary for a stable democracy. Coombs spoke about the need to bring to bear the full power of the research and technology sector to meet our national ambition. The need for the research sector to step up to the national challenge. From this era were born institutions like the Australian Academy of Science and the ANU, where I spent most of my academic life. Not just established, but uniquely positioned to bring science to the service of the nation. Our 70 years since illustrate an enviable record of achievement in shaping Australian science, its system, and its people. In our current geopolitical climate, amidst the pandemic, and to steer our way through climate change, I put to you that the need for research sector to step up to the national challenge is as great as ever. It is this changing environment and the role science can and must play that has informed our strategic plan. The strategic plan identifies 3 priorities: scientific leadership, mobilizing and transforming the academy, and sustained strategic engagement. In terms of scientific leadership, the academy can and must continue to play a leading role in shaping the Australian scientific landscape. Just as it was our scientific leadership that led to the establishment of national parks in Australia, the establishment of national astronomy infrastructure that sees Australia positioned today as a world leader in this discipline, and the founder of SAGE, Science in Australia Gender Equity. It is our scientific leadership that means today we lead scientific cooperation efforts across the Asia-Pacific region via the International Science Council, regional focal point for Asia and the Pacific. Consider for a moment the value of this mechanism to enable science diplomacy in the geopolitical turmoil of our region and our time. In this leadership role, we have been able to make a significant contribution to the historic establishment of a Pacific Academy of Sciences. An academy designed to bring together the region's expertise and empower local scholars to be part of solutions in their region and unite as a voice for science. An academy designed to move away from a model that sees their region as a net importer of knowledge, rather drawing on the unique knowledge of its own people to provide the Pacific with agency and a voice in shaping its destiny. It will be launched next month in Samoa as part of the Commonwealth Heads of Government meeting. And I am so proud of the Australian Academy of Sciences' role in assisting its establishment. Early this year, the Academy's scientific leadership and sustained advocacy that secured protections for international research collaborations that are critical for our national interests. Australia's defence export control framework has undergone significant change to enable license-free movement of goods, technology, and services both within Australia and between AUKUS partners, that is the UK and the US. When first proposed, the framework and legislative change did not adequately balance security risks with open collaboration and created an overly restrictive regime for research collaborations between countries outside of the AUKUS agreement. The Academy strongly advocated for and achieved legislative amendments that provide protections for the license-free exchange of fundamental research between nations. In addition, the definition of fundamental research is now enshrined in legislation providing protections from possible future political maneuvers to change the definition without the parliamentary scrutiny. Frankly, the Academy held strong when others in the sector were willing to accept changes that would have placed at risk Australia's ability to engage knowledge vital to discovery research. These hard-fought amendments strike a balance between protecting Australia's national security while ensuring collaboration can continue to serve our national interests. At the heart of the second priority, mobilizing and transforming the Academy, is you, our fellows. Together, we have reviewed national committees so they are poised for action, able to respond to the changing needs of disciplines. Together, we have redesigned our governance structures, aligned our resources, and positioned ourselves to have the greatest possible impact. Together, we have recognized the need to significantly lift our engagement with fellows, ministers, early and mid-career researchers, industry leaders, teachers, students, and the broader community. Together, we are building a membership of the Academy that reflects the breadth of the modern scientific enterprise. You might ask, why are national committees, governance, engagement, and disciplinary breadth important? Because developing scientific advice and advocating for science to the highest offices of the land demands excellence in our fellowship, operations, systems, and structures. Because our ability to mobilize the unique assets of the Academy, the heart of which is you, our fellowship, is critical to bring science to the service of the nation. Because our ability to influence the journey of students studying science and maths and the teachers guiding them requires discipline and leadership. Because our ability to empower the next generation of researchers and remove barriers that hold them back is vital for our future. And because we must always ensure our independence remains uncompromised. This relies on a growing investment corpus sufficient to sustain the Academy's activities. We are ambitious for this nation. We are ambitious for science. We recognize we need to operate at a greater scale. And that is why we are redoubling our efforts to attract philanthropic support to grow our corpus and enable us to bring science to the service of the nation at the scale the current times demand. We can, we must, and we will. Our strategic plan also identifies a key challenge, namely to deliver sought-after scientific advice that influences Australians' actions and contributes to global science. That advice is drawn from your expertise. It is valuable because the Academy is non-partisan. It is valuable because we do not carry vested interests. It is valuable because we are not beholden to any single institutions. And it is valuable because we have no skin in the game except a better Australia. Our only interest is ensuring expert evidence is shaping the decisions and actions of our people so the nation advances and everyone benefits. It's a bold and challenging mission. Over the last few years, we have provided sought-after independent scientific advice in areas as diverse as artificial intelligence, Great Barrier Reef sustainability, the management of long COVID, national security matters, national environmental law, criminal justice outcomes, technological transfer, STEM skill development, and research infrastructure policy. But there are many pressing issues that require scientific advice. We have successfully argued the case for a wholesale review of Australia's outdated R&D system. The strategic examination of Australia's R&D system was announced in the May budget this year. This review is a first step to align national efforts across the whole of government, industry, universities, and philanthropy to create an environment where investment in R&D is coherent, strategic, and scaled. To inform this government review, the Academy is developing a 10-year plan to demonstrate how science needs to evolve to achieve our national ambitions. The plan title, Australian Science, Australia's Future, Science 2035, being led by Science Policy Secretary Ian Chubb, is assessing the capability of Australia's science system, its ability to compete and collaborate globally, and its contribution to the nation's economy, security, health, and quality of life. The Academy is convening experts from within our own fellowship and across the research and innovation sector to inform this initiative. This 10-year plan will be published next year. Just outside in the Jaeger room, you will find a booth where you can learn more about our work and contribute to it. Fellows, we do a lot, but there is more to be done to bring science to the service of the nation at the scale the current times demand. Our strategic direction and the changes we are undertaking are designed to position us and ready us to, in the words of H.C. Coombs, bring to bear the full power of research and technology to meet our national ambitions. We must, we can, and we will. You have heard our mission and our strategic objectives, and I am hoping that many in the audience are asking themselves, how can I contribute? Every part of the Academy benefits, and our impact is greatest when we work closely with you. There are many ways in which you can contribute, some more time-consuming, others less so. You can contribute by providing your expertise to influence policy and the Academy's activities, by serving on one of the 19 national committees for science which shape scientific disciplines, by nominating outstanding individuals for fellowship, for council membership, and for awards, joining the Academy's council, the highest decision-making body of the organization, by attending the AGM and having your say, by being active in fellows' activities in your state or territory, by actively engaging with and providing mentorship to early and mid-career researchers. They are our future. This year, we have over 100 early and mid-career researchers are here. Take some time to say hello to them. By representing Australia internationally at global gatherings or on the governing committees of global bodies such as the international scientific unions, by proposing ideas for and reviewing our science communication content, by supporting our education programs. This list is by no means exhaustive. Executive committee members oversee the Academy on your behalf and each carry portfolio responsibilities linked to the functions of the Academy. Let me put some faces to names. It is important that you know who the executive committee members are and who you can turn to at any moment to ask questions or have input. Please welcome to the stage Professor Ivan Morisic, the Secretary for Physical Sciences. Ivan, where are you? Sorry to make you get up from the seats, which are difficult to get up. Professor Bob Graham, the Secretary for Biological Sciences. Ivan and Bob also serve as Vice Presidents of the Academy. Professor Marilyn Anderson is the Treasurer. Unfortunately, he is unable to be with us today. Professor Francis Safarovich, the Foreign Secretary. Okay, she's there. And if you forget her name, she asks you to call her as Madam Secretary. Professor Ian Chubb, the Secretary for Science Policy. Okay, he has to leave. Okay. Professor Lynn Beasley, the Secretary for Education. Professor Jim Williams is a spokesperson for integrity. And we will soon be appointing a spokesperson for engagement. Please join me thanking them for all their contributions to the Academy. We also have in the audience council members. Ellen Anderson is here, and Julian Gale is there, and I've seen Margaret Shield earlier, and so Ian Wright, I've seen him somewhere. Okay, he's there, and who else is there? Did I miss anyone? Glenda Halliday was here earlier. Okay, good. Anyway, so please talk to them as well. And if you have any questions about the governance process of the Academy or any matters of the Academy as well. I'd also like to acknowledge the Academy Fellows who serve the regional chairs and co-chairs, some of whom I have visited recently on my tour around Australia. Regional chairs play an important role in bringing together fellows at a state and territory level and facilitating engagement with the broader Academy. I think Alison Rogers was here and somewhere, and Clitty Grice I've seen, and Clitty is there. And of course, Warwick Couch is there, and so the Victorian chair as well. So really, we want to thank you for your contributions and making sure that our fellowship is engaged at the state level and also bringing the challenges and issues raised by them to the Council of the Academy as well. That really helps us to be able to learn about what's exciting science is happening in the country and also learn about challenges faced by the fellowship and the broader scientific community as well. And thank you also for hosting me and warmly welcoming me and Anna Maria during our visits. Fellows, I conclude this part of my President's address by reminding you this is your Academy. The more we work together, the greater our impact will be. Ian Potter House across the car park is available for your use at any time, even when you are in Canberra on non-academy business. It includes office and meeting facilities, a fellows lounge, and a place to connect or simply rest a bag. The Academy staff are always available to assist. Colleagues, thank you for your time and attention today. I look forward to more conversations with you during the rest of the Science at the Shrine Dome. Maybe I can ask ExCom members to, instead of making them to stand, I know standing for 2 hours, 20 minutes is how painful it is, and I will ask them to go and please take their seats. Thank you, thank you. I'd now like to move to the launch of the Academy's Innovate Reconciliation Action Plan. At its heart, reconciliation is about strengthening relationships between Aboriginal and Torres Strait Islander peoples and non-Indigenous peoples for the benefit of all Australians. As a champion of our Reconciliation Action Plan RAP, I know that the Academy's contribution to the reconciliation will be most widely felt when we strengthen the participation of our fellowship in our journey. That's why I chose this occasion to launch the Academy's Innovate RAP to be amongst you, our fellows, and to share with you our learnings and to engage you in our next steps. The Innovate RAP is a significant milestone for the Academy, coming after the successful completion of commitments in our Reflect RAP. The Innovate RAP reflects the work, deliberations, thinking, and reflections of the RAP working group over the last 18 months. I thank them for their commitment and for challenging our thinking. I particularly thank our Indigenous advisors, Dr. Katrina Rook and Associate Professor Bradley Mogridge, and the Academy Fellows, Professor John Patrick, Professor Helena Rubinstein Dunlap, and Professor Trevor Lithgow, as well as Academy staff who participate in the RAP working group. To launch Academy's Innovate RAP, it is my pleasure to welcome to the podium Professor John Patrick, Fellow of the Academy. John has been a tremendous guide for me, sharing with honesty his engagement with the traditional owners and speaking about how his research has been enriched by Indigenous engagement. John will share a few words, then engage in a discussion with Indigenous early and mid-career researchers from the RAP working group and Academy's EMCR forum. Before I hand over to John, could you please join me thanking Anna Maria, Melissa, and all the entire Academy's team, really their hard work, and then also putting together such a great Science at the Shrine Dome. Please join me thanking them. Over to you, John, please.
Fellows, EMCRs, accompanying persons, and the Secretariat. Reconciliation is everyone's business. Jagadish, thanks for giving me this opportunity to speak to the fellowship. I wasn't quite sure what was needed here, so I've elected to talk about myself and my journey, and the message I want to deliver is that it's not an exemplar, it's not special. And as Jagadish has said, he has extended an invitation for us all to contribute to the reconciliation journey of this Academy. So I stand here not as an exemplar of what you might think you need to have achieved in this space. And in fact, there are many in this fellowship that are compelling examples that are making profound impact. For instance, Stephen Simpson, Jenny Brand-Miller, David James are developing innovative solutions to combat chronic diseases in Indigenous health at the University of Sydney's Charles Perkins Centre. Equally, CSIRO is taking a leading role in engaging with Indigenous knowledges and methodologies that undoubtedly involves members of the Academy's fellowship. And it'd be remiss of me not to acknowledge that past President Andrew Holmes and the CEO, Anna Maria Arabia, committed the Academy to a reconciliation action plan. This continues to be largely driven by Anna Maria and members of the Secretariat. I'll now just reflect on me, which sounds a bit sort of self-serving, but it's not intended to be so. And I have two messages, essentially, about reconciliation. First, it very much depends on an informed cultural awareness and capability. And in getting this invitation to speak, I reflected on my journey, which I believe started for me in the 1940s and 50s, growing up in a subsistence farming community located on the western shores of Lake Macquarie on unceded Awabakal land. Here, a flourishing past Aboriginal presence is rendered blatantly obvious by the numerous middens of cockle shells embedded in the soil profile. I didn't have to ask my parents what happened. It was clearly catastrophic. A decade later, exposure to Aboriginal culture was supercharged through fiery speeches by the late Charles Perkins. Symbolically, he was a wise man, symbolically delivered from the steps of the University's Sydney's Great Hall, the centre of intellectual activity. At the time, Charles Perkins was enrolled as a mature-age undergraduate in a BA degree, and in fact he was the first Aboriginal to graduate from an Australian university. My lack of courage blocked me from joining his Northern New South Wales Freedom bus ride in February 1967. This drew international attention, was mentioned in the New York Times, to the impoverished living conditions and palpable racism faced by Aboriginal communities in this otherwise great country. Another 2 decades, this is my journey, elapsed before opportunities arose to attend workshops on cultural awareness and capability run in my home university by an Aboriginal and Torres Strait Islander institute enabled by that university called Wallatuka that started in the mid-80s. An institute, and this is important, entirely staffed by Indigenous people. These workshops have become commonplace for organisations fulfilling reconciliation plan obligations. The other point I want to make about reconciliation is an obvious one, there needs to be a change to the status quo. There needs to be a change to the status quo. And you could say, well, John, what have you done? Very little, very little. I just pull some examples of what I've been linked with. I won't make any special claims, but the point I'm making, little contributions can mean so much. So, for an international conference convened in 1999, over 25 years ago, from about now, the organising committee that I served on secured Indigenous community permission, and I repeat that, we sought permission to fly the Aboriginal and Torres Strait Islander flags, as well as have an Aboriginal elder, Uncle Bill Smith, a proud, and I apologise, Bill, Anawan for my pronunciations, Woonara and Kamilaroi man, deliver a welcome speech to country. These speeches weren't given in that time. Welcome speech to country at a dinner that opened the conference. Uncle Bill's message, and this was profound, to the 250 assembled plant scientists was to walk together and share knowledge openly in a non-transactional manner to benefit community. And the impressive thing about this, that message resonated with the attendees. We didn't have a feel what the reaction would be. And in fact, resonated well into the future, meeting people globally that attended that conference. As Jagadish has mentioned, education is core to advancing reconciliation. And in this context, and a very modest contribution, mentoring several mature-age Indigenous and undergraduates through a system set up by the Wallatuka Institute at the University of Newcastle has been a rewarding experience. Similar comment applies to participating in successful Aboriginal summer schools for excellence in technology and science for high achieving, and it's so impressive, these young Year 10 Indigenous students. There is real hope out there. And finally, serving as a member of the RAP working group here at the Academy and the University of Newcastle has provided that sort of structure to provide opportunities to contribute to various reconciliation initiatives that, as an individual, I couldn't do. For instance, it's been an immense honour to be a member of and subsequently chairing the Academy's selection committee for the Aboriginal and Torres Strait Islander Scientist Award, and mixed with these wonderful young Indigenous STEM professionals. It's really uplifting. And Jagadish, I'll conclude by saying I'm most proud that you accepted the title without boxing gloves of being the reconciliation champion of the Academy, and more so agreeing that the selection panel for the Indigenous Award be completely Indigenous. I was happy to surrender that job and know that there was agency there for Indigenous people to review those applications. So, I'm not good at organisation and segueing, but in in launching the innovative RAP, so I've ended, you can now perhaps pay more attention, I've done my journey. RAP, President Jagadish outlined the Academy's vision for reconciliation and what has been done and what remains to be done. And this is really important. The level to which this enterprise succeeds very much depends on a growing engagement by the fellowship. And I think, and it wasn't my idea, I don't have many ideas really, it wasn't my idea over there, was to organise a panel of deadly Indigenous STEM professionals to have a yarn, and I was invited to have a yarn with them, yeah? So that's what we're going to do, to have a yarn with these two deadly Indigenous STEM professionals. They'll come up here now, obey, and we'll have a talk. And are we joined by... Oh, there he is. Okay, the third panellist. So could I introduce these good people? So we have Michelle Hobbs from Griffith University. We have Jordan Pitt from the University of Sydney, and none other than Happy Head, Associate Professor Bradley Mogbridge from UTS. Now, just before inviting these good people to introduce themselves, and that's where agency counts, could I point out they all have affiliations with the Academy. Melissa and Brad. Oh, I'm an old fellow, I get names. Sorry, Michelle. Michelle and Brad are recipients of the Indigenous Scientist Award from this Academy. Brad is a valued member of the RAP Working Group and Indigenous Scientist Award Committee. You might remember Jordan from last year. He co-convened the well-received 2023 Academy's Looking Back, Moving Forward public lecture series featuring Aboriginal and Torres Strait Islander researchers. He is current chair of the Academy's Early and Mid-Career Researcher Forum Executive. Okay, we'll have a yarn. So we've formulated, and these questions have been agreed by Michelle and Jordan, I think, but they have agency to manipulate them if they so wish. So the first question we thought would be useful is to give them a chance to introduce themselves. So what I'm interested in hearing is each of the panel indicating where they are from, their mob, their journey followed to become an Indigenous STEM professional, and the academic position they now hold. In addition, if they are comfortable, it would, I think, be valuable for the fellowship to hear about their unique barriers they may have encountered along the way as Indigenous STEM professionals. And Michelle, would you like to lead out with this? Because in a way, you've just entered this fray. Thank you.
Thanks, John. Good afternoon, everyone. So my name is Michelle Hobbs. I have connections to Bidgera, who are from semi-arid central Queensland area, and Dungutty, who are from Central Coast, New South Wales. So I'm a freshwater and a saltwater person. So it follows that I studied marine biology at the University of Queensland for my undergrad, and then moved on to a career in freshwater ecology. So currently I'm an associate lecturer and I'm still doing my PhD at Griffith University with the Australian Rivers Institute. So as John mentioned, I was also a recipient of the Aboriginal and Torres Strait Islander Scientist Award in 2023, which was a great honour and actually was a great boost for my profile and my career, which was, a wonderful experience. I guess in terms of my journey towards ecology, I chose ecology more because I was fascinated by plants and animals as a young person and a child growing up on a farm, and also because I had wonderful role models in my life. I saw my dad go on a journey from being a farmer to being a mature age student at university and becoming an academic for a career quite late in life. So I thought, well, if Dad can do it, then surely I can do that too. And also I had amazing female science teachers at school who encouraged me. So having those role models made me able to see myself in those positions. However, I never had Indigenous role models in science, and so for a long time it was quite difficult to picture myself being not just a scientist or a woman scientist, but being an Indigenous scientist, and what did that mean? I would say that I'm very fortunate to have had mentors and people encouraging me along the way, particularly Brad, who's here today. Smoothing the way for me, I guess, and encouraging me, including encouraging me to apply for the award that I won a couple of years ago. Well, I might leave it there and hand over to Jordan.
Yes, thank you, John. So I am Jordan Pitt. I am, Associate Dean Indigenous in the Faculty of Science at the University of Sydney and an applied mathematician. In thinking about this question, I've reflected a lot, I guess, because of the 70-year anniversary on the journey of my father. He was born in 1957, I guess as flora and fauna, up in Cairns. And so he was always a very bookish kid, actually. He really liked going to the libraries. But of course, doing that in Brisbane, where he moved down at some point, he got a lot of side looks and wasn't really encouraged to follow education, ended up leaving before finishing high school, and then going and join the military, but creating, I guess, a stable life for me to then follow through and be able to use some of that kind of bookish interests and be realized in me. So there's been a lot of growth, but also I guess it's moved in now. It's a bit harder to understand some of the barriers because it's a bit less obvious what they might be, and it's a bit more nuanced and heterogeneous for different groups as well, and the intersection of different diversity dimensions. I guess the thing that I always struggled with personally was kind of, the identity piece, and I guess kind of seeing where I fit in the story of science, because the way we usually talk about science doesn't feel really like it includes me. I guess that, and also a bit of like the pressure, right? So being good at maths and knowing from a very young age that, I was good at maths, but also like I was going to be one of the only Indigenous person that I knew that was good at it. And so there was always a bit of like that outside pressure to perform as well, like not from my father or my mother, but just kind of because of the circumstances of the society in which we live. Good mentors as well, and being very supportive has always been helpful.
Thanks, Jordan. Brad, a short comment. Brad, a short comment. Oh. Can't hear him. Can anyone?
Can't hear me.
Oh, no, can now.
Right, there we go. Yeah. I can hear you loud and clear. Awesome. We're on. Look, a short comment. I in the late 80s, I went from, high school to senior school and was generally encouraged not to do any science or maths. I chose to do some science. And, you know, as I look back at the last 20, 25, 20, 26 years, maybe more as a scientist, you know, I've sort of, like Michelle and no doubt Jordan, you know, it's been a lonely space. We struggle to find like-minded Indigenous scientists, and obviously that's a challenge in itself. But I suppose, you know, having a passion for it, you know, I started off in geology, but I found myself looking for uranium in a national park in the Great Sandy Desert, and that didn't fit too well. So I come home and shifted to environmental science, and then later on down to hydrogeology, and then soon found out, you know, the connection, you know, hearing some of the stories from the old people around knowledge and understanding of groundwater on the driest inhabited continent on Earth. And that's really driven me to sort of merge the two knowledge systems to come to solutions to the way we value and manage our water resources on, as I mentioned, the driest inhabited continent. And no doubt that old knowledge can play a part in developing solutions to the way we, you know, manage. And then also we can look for, you know, 234 years of fixing this place up, because it's copped a lot over the years, and, you know, and I think that's why I'm still in it. And yeah, hopefully we can work towards that. And I suppose with the Academy's RAP and advancing that, and also, you know, the national science priorities and statement actually pushing us and pushing the culture of science to shift. And, you know, as you mentioned, John, you know, we need to change it and shift the paradigm, and also and we need to be front and center. That's what I believe anyway. And our elders need to have direct input into developing solutions to fix this country up, because it's not too good at the moment. You know, when you think of the droughts we've had over the last 20 years, 2 of the worst on record, 2 1 in 500 year floods on the north coast of New South Wales. You know, they should happen every 500 years. They happen within a matter of months. We've had horrendous fires. We've had rivers drying up, massive fish kills. So, you know, like we're not in a good space. So I think Indigenous people, we're here ready to go. We've always been here ready. Let's say we're doing science before science was a thing and the Royal Society was even thought of. So let's with the RAP, you know, I think with the Academy support, we can move forward in this space. Thanks, John.
Thanks, Brad. In fact, you've covered 2 of my other questions.
Sorry, mate.
Give Michelle and Jordan a bit of a go at these. So you will all have recognised that the acknowledgement of country statement, but perhaps some of you mightn't appreciate country as core to Aboriginal and Torres Strait Islanders. This is manifested in a custodial ethic of caring for country that includes their unceded lands and waters, as well as plants, animals, ancestors, laws, language, culture and ethics. What I'd be interested in the panel to comment on is how they manage caring for country while simultaneously prosecuting a research program. And Michelle, maybe we start with you, because Michelle is just starting her journey in this area. Thanks, Michelle.
Yeah, you've said it really well. So caring for country, there's a saying, and it's not my saying, so don't quote me on this. It's a saying that's common to many different mobs, but I heard it from Uncle Blackman, who's a Butchulla man, who's a caretaker of Gari, otherwise known as Fraser Island. And he says, if you look after country, country will look after you. And that kind of sums up the Indigenous philosophy that we're all connected to country, we rely on it for everything, and it's our responsibility to look after it, and we take that very literally. So in terms of how that feeds into research, I've come to realise that we really need to challenge the status quo with how we do research, particularly when it comes to data pertaining to natural resources, environment, land, water, biological samples, and human data as well. Because The totem species that are involved in those samples, even the rocks and the soil, are incredibly important to culture, and Indigenous people have a direct responsibility to care for those relationships. So the way that I see research moving forward, and I definitely don't do this perfectly all the time because I've been trained in the status quo way of doing research. So the way that we need to move forward is by working with communities and co-designing research projects. That's so important because I think of science as a tool, and you only get the answer to the question that you directly ask from science. And we all know the importance of a really good hypothesis. But the thing with our hypotheses is that we all bring our own observations, our own perspectives, our own questions, and our own aims for the project. So when you give communities the opportunity to have a way in to these aims and these outcomes and these questions, you're giving them direct involvement and agency in being involved in the project, and for there being tangible benefits to the community as well.
Thanks. Jordan.
I guess the point I'll make is maybe the obvious one, is that the three of us are all involved in some way in environmental science. Now, obviously, Brad and Michelle are a lot more involved directly in environmental science, but even in my kind of research looking at applied mathematical models for the interaction of ocean waves and sea ice to understand climate processes, the perspectives and worldviews behind caring for country are very much core to why I'm interested in that work and why I think it will benefit people. I think there is a bit of a danger there, though, like in terms of we could recast a lot of things as caring for country, and I think we should be a bit careful, and I think Michelle hit on the point that we are doing it in a meaningful way, in particular by engaging with the local place-based community whose country we're trying to care for as well. But otherwise, Michelle did an excellent job of covering it, so I didn't have much to add.
It's dangerous, including you, Brad, we'll be here all night, but perhaps we can sort of move on. So caring for country is predicated, of course, on Indigenous knowledges and methodologies, and that have been developed over the millennia from time immemorial, and are and this is something that you use my words, being recast. They're not static, they're evolving. And I think it'd be useful for the fellowships to just get a sort of, and we'll start with you, Brad, an insight into what are Indigenous knowledges and how are they created and engaged by Indigenous research methodologies to care for country? And maybe just using an example.
Yeah, that's a great question. Definitely knowledges are gained over, as you said, millennia. A lot of Indigenous knowledge goes back to day one or beyond the dream time, or when our ancestors come from beyond the Milky Way to do their thing on Earth and create a landscape for us before all the other gods, let's say. And those knowledges are gained through impact, observation, testing, retesting, and then I suppose you create let's say, stories, and I suppose our stories aren't seen as fact, they're seen as fiction, and they're put in the realm away from science. So I suppose our observations are connected to our knowledges, that are connected to our stories and our songs and our dances and which then engages in the law of the land. So not the law of the people, but the law of the land. And I think having Indigenous scholars go through the process of creating Indigenous research methodologies is doing it our way. And I suppose, there's potentially a way of coming up with a methodology in a Western sense, but I suppose sitting down by the riverbank, creating an Indigenous research methodology, which a lot of our elders would not understand, but when you start talking about their connection to country and their understanding of country, their understanding of weather systems, their understanding of sky stories, the movement of the stars, the movement of species, and the turning up of species, a flowering of a plant, that observation is linked into the methodology, and then you can actually start understanding country itself. And so in those research methodologies then come from an Indigenous point of view. And, like even thinking through my research over the years, like sometimes I sort of wonder why I do research, because if my mum can't understand it, then why am I doing it? And, she left year 8 to clean white people's houses, as many Aboriginal women did in the 40s and 50s, 60s up until the 70s, So, she didn't have that level of literacy, sorry. So, having it in a form that she can understand, I think I'm doing my job, but then I won't get published. So that's a real challenge for Indigenous scholars to enter this space and start, having their research published. And then, if it's coming from Indigenous research methodology and from country and from the people and from the stories, I'm doing my bit. And I think, when the previous question, I just want to add that, if us as Aboriginal and Torres Strait Islander people first, but also then scientists and mathematicians and engineers and technologists, if we do it wrong, we don't work in Aboriginal affairs again. So it's in our best interest to do it right, and it's right. We do it right for the people, and we do it right for country. So in that regard, these research methodologies have to be sound from an Indigenous point of view. I could talk for days, but I shut up.
Thanks, Brad. And that is actually a really useful segue into this real challenge of blending Indigenous and non-Indigenous knowledges and methodologies, which is just starting to be considered and happening. And to me, one interesting thing, and it's been mentioned by some of the new fellows, One Health. One Health is actually caring for country, so it cares for you. So there's actually a platform on which one can place thinking about how these knowledges can be blended. And Jordan, I'll turn to you because I understand the EMCR communities discussing this very thing. So your comments would be valuable.
Yeah, I guess for me, what I see as the relevance of EMCRs is kind of the new and exciting critical mass of Aboriginal and Torres Strait Islander PhDs and people undergoing their PhDs, and with a wealth of kind of, I guess, Western science training and Aboriginal and Torres Strait Islander traditional knowledges as well. I think it's an incredibly exciting idea, and I guess we're in Canberra, so I also see it as a bit of a broader thing about reconciliation of the nation and a bit of nation building to build, to bring everything together. I guess what I would say is that there is a bit of a danger here as well. Like, we're not trying to find, with Indigenous knowledges, like needles in the haystack. The whole haystack's important, okay? So we can't really separate out the science and choose bits that we want to do. And that's certainly, like, for instance, I guess what I'm saying is what you might think about as being important in your scientific research might not be what community thinks is important and what they want to preserve. And so that's something to be aware of, and when trying to engage to blend these things. And the other part is just having genuine, trusted and deep relationships with communities in which you're trying to interact with, so that you can actually properly blend these things, and in a way that benefits them with that reciprocity and benefit sharing. And something that I'm trying to do more and more of as well is trying to build relationships that cut across different levels of the institution. Like, our individual researchers will always be the leads in the research that's going on, but we also need ways to engage, for communities to engage with the institution at a different number of levels, so that we can make sure that their voices are being heard right across the institution, and we're not playing the game of, well, I don't have any power to do this. Yeah, that's kind of what I see.
I know we're running out of time, yeah, but There was a request about getting a report card from these good people on where they see the academy. Can we do that, or should we bring? Thanks. So, as you all probably know, you know because Jagadish has said this, but you might have known this before, the Academy is on a reconciliation journey and progressed from 2019 to this point. Now, when the Reflect RAP was launched, Brad, you gave a little speech, if you remember, and it's published. It's on the record, Brad. And you in that speech, you offered a list of aspirational outcomes for Indigenous STEM professionals. And very briefly, because we are sort of using time here, just outline, give us a report card on where we are at against those aspirations you listed. Fearlessly, I must say.
Yeah, thanks. I think there's definitely been a great improvement in this space from the Academy. There's been courage in moving forward, especially to the Innovate. And, you know, when you think back from the original RAP, you know, it wasn't moved straight away, and the Academy acknowledged that it wasn't ready to move to Innovate until now. And so, you know, that's an honest conversation that the Academy's had with itself. So I think that's great strength and opportunity that I've seen in the Academy and the leadership. But I have a further list of demands. Is that all right? So increasing Indigenous fellows is always a big one. Currently, we're at 0.017%. So that's a big challenge. Elevate the opportunity to implement the National Science Statement, you know, as an academy, and also the priorities, allowing Indigenous knowledge to be seen as an equal, having courage to allow Indigenous scholars to lead projects through the academy, the leadership, the council, the fellows, the president, own it and lead by example, the RAP. And I think, you know, that's been a big improvement. And, you know, we've seen the Academy grow in this space. And one last recommendation. 22 national science committees. I'd love to see how many Indigenous members of those 22. And I did a quick look through, and I saw one in the astronomy. But also then I'd love to see an Indigenous Knowledge National Science Committee. That'll do me.
Yeah, I think that's it. Thanks, Brad. Can I leave it to you guys too? Yeah.
Firstly, thank you, John. You are always so gracious and so humble with your contributions. I know I have learned an awful amount from you, and you've been with us since our very first step. So thank you for your commitment and in illustrating and modelling to other fellows what we can achieve when we have more and more fellowship engagement in our reconciliation journey. We've always known that our Reconciliation Action Plan and its recommendations and our work will have the furthest reach and the furthest impact when we have more and more fellows involved. So I hope today excites you or makes you want to become more involved in our Reconciliation Action Plan work and to do what you can in your respective states and territories, laboratories, and in your fields of expertise. To Jordan and Michelle, thank you. And to Bradley, he's left, but if you can hear this, or I'll send him a recording. We have had many, many hard discussions, and I have needed to hear them all. We have needed to hear them all. And I actually welcome with open arms the recommendations and the demands that he puts going forward. I think there are some tremendous suggestions in there. Jordan, you're leading the way with the Early and Mid-Career Research Forum as its chair. And I'm looking forward to the transformations that you bring. I know the EMCR Forum always model for us ways of doing things differently and often better. So we look to you to learn. So thank you. And Michelle, as an awardee and involved in much of our work, thank you for your contributions. I'd also like to acknowledge Katrina Ruck. She's been on our working group, our Reconciliation Action Plan working group. She couldn't be with us today. She's delivering a presentation in another part of Australia. But she too has given us some very confronting and important advice that we have needed to hear. So thank you for your contributions, and thank you for so beautifully launching our Reconciliation Action Plan, Innovate. I hope we can fly through it. I hope we can achieve all of the things and get on to this stretch, the next level. But one step at a time, we need to walk before we run. So with that, I will now move to close this event because, not the event, just the day. There's 2 more days to go. Just down the way in the car park is that wonderful marquee, and it is ready and waiting for all of you for our spring soiree. There's a drink for us to share. I do need to share a little bit of knowledge with you. Many people have asked me about this collage, this logo. It's our 70th anniversary logo, and it was put together by our graphic designer, Leah Albert. And in it, and if you look closely and study it, you will find many things. And in there, you will find science and technology references from every state and territory in Australia. The 3 fellows at the bottom there are Sir Mark Oliphant, our first president, Dorothy Hill, our first female president, and Sir Ian Potter, whose wisdom and financial skills and philanthropy helped shape the Academy. We wanted to put all of the fellows up there, but we couldn't. So we chose the early days being our 70th anniversary. But all your work is represented, everything from CubeSats to state and territories to flora and fauna and different types of science. So a lot of thought went into that as our 70th anniversary logo. With that, I will close the day. Let's have a drink together. Let's continue networking and collaborating. Thank you all for a marvellous day, and we will reconvene tomorrow morning for the presentations from our 2024 fellows. Thank you.